सुप्रभात। आपसे ही इस संस्था की पहचान है, आपसे ही इसका मान है, सम्मान है, और जहां हो आप जैसे अनुसंधान करता वही अखिल भारतीय आयुर्विज्ञान संस्थान है। शरीर माध्यम खलु धर्म साधनम का दिया जिसने भारत को वरदान है, परिपूर्ण किया चिकित्सक और चिकित्सा से विज्ञान जय जहां जय अनुसंधान है, जहां जय अनुसंधान है। जय अनुसंधान, विज्ञान, वंदन, अभिनंदन। अभिनंदन एम्स की इस धरा से जहां बैठे आप जैसे फनकार हैं, फनकार जो जीते जीवन आरोह अवरोह में, फनकार जो जीते जीवन आरोह अवरोह में है। स्वागत इस शोध दिवस में, स्वागत शोधार्थी शोध समारोह में।
A very hearty and cheerful morning to one and all assembled here today. We, Iman Dondapat and Vikas Kumar, on behalf of the entire research community, would like to extend our warm welcome on the occasion of the second research day being organized at All India Institute of Medical Sciences, New Delhi. To begin with our today's proceedings, I would like to request our Honorable Director, Professor M. Srinivas, and Dean Research, Professor Rama Chaudhary, to kindly come onto the dais. We would request Professor Rama Chaudhary to deliver the welcome address.
Good morning, everyone. Respected Director, Sir, Professor M. Srinivas, Dr. Subrata Sinha, our Dean Academics, Dean Examination, Dr. K.K. Deepak, Dr. Vineet, and many of our senior faculty members. It's so nice to see them. Dr. Mahapatra, Dr. Kusum Verma, Dr. T.P. Singh, Dr. Neena Khanna, our former Dean Academics. So indeed, it's a day of celebration. After 2-3 years of COVID pandemic, we are physically meeting here today. So aim of this historic event is actually to connect students, researchers, as well as faculties with each other and create a platform where we can actually talk about translating the research. So taking it from bench to bedside. We all know that AIIMS has a mandate, and one of the important limb of that is research. So this research day is actually going to help each one of you to really know what is going on in my next lab. We are so busy, we don't even know what my colleague is doing. So time is now to collaborate, make team efforts, and continue working in an area which is very, very important in these days. It was in COVID pandemic, we understood the importance of research. In a very short time, we were able to make diagnostic kits in India. We were able to make our own vaccine, and India was on global stage. So I think with that, I would really like to welcome all of you on today's second annual research day. And I'm very happy to inform you that there has been almost 400 posters, more than 12 devices which are on display. There is a special poster session on sustainable development goals. There are also encouragement given in the form of research awards, which we will be doing it today, and not only for 2021, also 2020. And the whole idea of this event is that we can really advance research in the best possible way. As you are aware, AIIMS has been on the top front as far as research is concerned. In fact, the global survey has shown that it was third amongst the total, research institutions all over the world in 2016 survey. I would like to acknowledge the untiring efforts of all the faculty members, students, especially our Associate Research Dean, Dr. Ahuja, for their unwavering support. And also, CMET, we have created a new unit in research section, which is clinical research unit. And we also have CMIE, which is about entrepreneurship. We will be listening more about it in the afternoon panel discussion. So I would like to extend warm welcome to each one of you on this second annual AIIMS Day. And I'm sure this will be a great platform for connecting, discussing, and working together to find answers to the scientific problems in medicine and beyond. Thank you so much.
Thank you, ma'am, for such encouraging words. I will now request our director, sir, to deliver the audience with the opening remarks.
Thank you. Well, I say respected my teachers. I see some of them here. Dean Academics, Dean Research, all the faculty members and researchers. First of all, I would like to congratulate the team, which has done an excellent work. I think we should give a big hand to them. And my request for you would be that, if possible, keep it for a week or so. I don't know how many conferences are going to take that space. And ask the other people to come, at least from the Delhi hospitals and the colleges, research institutions, to come and have a look, at least at the level of the senior residents and the PhD levels, because they will get the connect that, this kind of research is happening. He is a person who is going to help me, how I'm going to, get the help. And then interaction between our students and those students, because that will go a long way, because what we are doing should not be in the silo. Only we are, publications and department and then annual reports. And then the thrust would be, which is there. I can see the thrust from the bench to the bedside. I think that is something we should extend it and then give a wide publicity that the people are connected between the institutions. And that will also help us, because as a senior resident, I used to struggle. Some of the people are here, they know. We used to go to the other departments, steal the rats, even the PBS we used to steal from the clinical departments. Space is an issue, timings is a problem for the clinical people to research. And then going to other institutions, we used to go to the JNU, NIHW, ICGB, NIN. I think a formal collaboration we need to enhance. It is there. Dr. T.P. Singh is here. It is there, but I think we need to enhance there so that, if it comes from the head of the department and at the level of that, it will bridge that gap more, and the strength will be more between the people to do a lot of collaborative research. Because gone are the days where we can do research within our own labs. I think we should come out of that thing. Mera equipment hai, mera lab hai, mera hi hai, koi aur nahi aayega. As long as we have this tendency, I think we'll be just doing publications which have no views. And we will not be able to give justice to the cost-benefit analysis for the things, because everything is being seen now. The kind of money which was flowing before to the research has definitely come down. We need to struggle to get that. And then if it has no implications to the clinical services, patient maybe prognostication, diagnostics, or therapy, and it's not going to be sustainable. And I'm very happy that, sustainable development goals have been taken up. A wonderful initiative, ma'am. I think we must give it to the ministry and the other people also. The community medicine and everybody should put a hand and then push this to the ministry and all, because they think that, our research is just for the publication. I think that's something which we must push it to the ministry and the people who are making policies, maybe the Niti Aayog and our ministry, that let them come and see that how we are also part of the SDG development goals and everything. I think with this, I once again congratulate the team, which has done an excellent work. And unfortunately, we are not giving the time. I think we must make it mandatory that faculty, including the director, will go speak to them. Let us spend the time. See, what is the encouragement at the end of the day for these researchers? They're all working after 4 o'clock, middle of the night, burning their midnight oil libraries and all that. I think some amount of research encouragement has to happen from us also. Make it mandatory that people will go, see, give ample time. अब clinicals का end नहीं होता है. And I think, there's no end for that. Patient तो always होता है. But at the same time, we have all the domains. Patient care is there, but the research, teaching, learning, and the other things which are part of our good governance and transparency and everything which is being a focus for us now, I think it's very important that we give some time to these people and encourage them. See, you look into that. When we go to them, they're so happy to share with you. Yesterday I could see that, for some time when I went there. I think we need to encourage them. The doyens are here. Please help us also and our youngsters that, give kind of encouragement to them, inspire them, so that we move forward to do more of our research which has relevance to our own patient care and relevance to our own country. That's something which is very important for all of us. With this, I once again congratulate the team. A wonderful job, and a big hand to the people who are presenting there, and the posters are there. Thank you very much. Thank you very much.
धन्यवाद सर, कार्यक्रम की शुरुआत को अपने आशीर्वाद शब्दों से सुसज्जित करने के लिए। अगले सत्र के लिए डॉक्टर सौविक मन्ना। डॉक्टर सौविक।
A very good morning to all of you. On the occasion of second Research Day of AIIMS, on behalf of Research Section, it gives us immense pleasure to welcome you all to the scientific session, Ignited Minds Best Papers. The first platform presentation is by Ph.D. students. Each presentation will be of 7 minutes, followed by 2 minutes for discussion. For this session, I would like to welcome the Honorable Chairpersons onto the dais, Professor Kusum Verma and Professor T.P. Singh. Professor Kusum Verma was formerly Dean Academics and Professor and Head Department of Pathology, AIIMS, New Delhi. Madam is currently consultant at Sri Gangaram Hospital, New Delhi. Professor T.P. Singh was formerly Professor and Head Department of Biophysics, AIIMS, New Delhi, and is also Distinguished Research Professor in Biotechnology. The first paper will be presented by Srishty Raman, Ph.D. student from Department of Anatomy.
Search for giving us this opportunity. It's always a pleasure to come back to my alma mater. I spent majority of my life here, and I'm looking forward to an academic feast by these 3 presentations of PhD. And now I think we'll start.
Say a few words. Okay. Good morning. I'm very thankful to Professor Rama Chaudhary, Subrata Sinha, Vineet, and the whole team. And of course, I was very delighted to hear wonderful words from our Honorable Director, Dr. M. Srinivas. I think this is the right way to think about research. Without research, we are nothing. So maybe this is not the time to talk on this. We will talk some other time. So we can start the proceedings.
Please start.
A very good morning to all of you. Today, I'm presenting my PhD work where we have developed a novel, visual, rapid, and cost-effective nanotechnology-based confirmatory diagnostic kit for the detection of cervical cancer cells from cytology or histology sample. This method entails an assay based on magnetic nanoparticles and quantum dots coupled immuno-nano-fluorescence, and thence is referred as MNCQDINFA. This kit, comprising the cervical cancer detection kit, was developed and was patented last year, August. Accounting for being the 4th most common worldwide and 2nd commonest nationwide, the disease, cervical cancer, has taken a heavy toll on human life. These facts are enough to mark the prevalence of the disease and the necessity to curb its dominance. The increased global awareness of the disease has instigated development and refinement of the screening and already available confirmatory diagnostic tests. The most prevalent amongst those are mentioned over here. With advanced research and explicit technology, the European Research Organization of Genital Infection and Neoplasia in 2014 stated that human papillomavirus infection is the sole etiological agent for cervical cancer. Although till date, around more than 200 types of HPV are known, but only a subset amongst them are referred as high-risk types. And amongst those high-risk types, HPV 16 and 18 are responsible for 70 to 80% of cervical cancer, while HPV 16 alone is responsible for almost half of all the cervical cancers. HPV infection sets the stage for the neoplastic and cancer stages, and based on these normal cytological changes which incur upon HPV infection, the cervical cancer has been staged into CIN 1, 2, and 3. So, in CIN1, human papillomavirus infects, internalizes, and persists in an episomal manner, while in CIN2, the human papillomavirus DNA integrates and transforms the normal cervical cells into cancerous cells. And thereupon, if the HPV infection does not resolve on its own, it then thereupon leads to invasive carcinoma. In the techniques I mentioned in the previous slide, HPV DNA-based detection techniques are able to detect HPV in all the 3 stages. But are those techniques able to differentiate between the infection and the transformed cancer cells? No. The other techniques in non-invasive screening techniques, which were mentioned in the previous slide, are the pap test and other cytological and histological techniques. Those techniques are able to detect only the later indirect cytological and histological changes which take place in the cervical cells upon HPV infection. Now, those screening tests are effective only in the later stages of the disease, and then they thereupon warrant further confirmatory biopsies which are invasive. So there, this MNCQDINFA, the assay, is able to detect the transformed cervical cancer cells rightly at CIN2 stage. And that is because of the biomarker of our choice, that is HPV E7. HPV transcribes almost 8 proteins, and out of those 8 proteins, E6 and E7 proteins are the major oncoproteins. So out of those E6 and E7, both these proteins, they are expressed by the cervical cancer cells only in the transformation stage. This E7 protein is the one which is responsible for initiation and maintenance of cervical cancer and was then chosen as the biomarker of our assay. Now this here we present an overview of our indigenous diagnostic kit for the detection of cervical cancer transformed cells. These are the constituents, and here we present a detailed description of how to carry out the assay in a time-based manner. One can see over here the negative and the positive observable difference is quite readily observed and can be easily comprehensible. So this can also be carried out in any low resource setting. Now, this is a detailed procedure. So when we get a cytology sample, we process it in order to enrich the protein component of the cytology. And then the enriched cell lysate is then incubated with the capturing antibody complex. These are then magnet-induced pelleted, and whatever the unreacted reactants, they are just washed off. Then the magnet-induced pellet is then incubated with the detecting antibody complex, and then again magnet-induced pelleting is done. And whatever the unreacted detecting antibody complex is there, it is washed off. And finally, the magnet-induced pellet is then dispersed and visualized in the presence of UV. So whenever there is fluorescence, we term it as positive for cervical cancer, and when there is no fluorescence, we term it as negative for cervical cancer. So once the detection procedure was finalized, we then went on to characterize the performance characteristics of the kit, for which the analytical sensitivity was tested using the purified and overexpressed E7 protein. So we tested a varied concentration of this standard E7 protein ranging from 1 microgram per ml to 100 microgram per ml using this kit, and we were able to find that the analytical sensitivity of the assay was like around 200 nanogram, and the observable fluorimeter results were also congruent to the observable results. Next, for the specificity, we tested many number of proteins whichever were available in the lab, and a few of the results are being mentioned over here. As can be seen, E7 was detected, only standard E7 protein was detected using this fluorescence MNCQDINFA analysis kit. Further, taken forward the analytical specificity, the cervical cancer cell lines HeLa and SIHA cell lysates and non-cervical cancer cell line NIH3T3 were taken, and they were tested using this kit with phosphate buffer cell line and E7 protein as negative and positive sample. So when these were tested again, the results were congruent as were the previous. Only SIHA cells, which have the HPV16 genome integrated into them, were able to express. Now, these were the antibodies which we used in the formulation of the kit, and when immunofluorescence staining was done, similar were the results. Only SIHA cells, which have HPV16 integration, were able to show fluorescence and showed positive results, which further confirmed our results for the assay. Now, finally, when MNCQDINFA testing was done in order to check for the diagnostic sensitivity and specificity, we took 25 cytology samples from the OBS-GYN OPD, and they were tested using the MNCQDINFA. So the results are portrayed onto the right. And for the gold standard, we went for immunofluorescence testing, and we used here commercially available in Vitrogen HPV16 E7 antibody. The results, as one can see, they are highly congruous. So when they were in the MNCQDINFA analysis and those of the immunofluorescence staining, when they were correlated, we were able to determine a diagnostic sensitivity and specificity of around 100% for both. Lastly, the proposed MNCQDINFA is a simple two-step and takes around 100 rupees per sample, is a very quick, 2 hours for a single sample to be carried out, and presents a very high specificity and sensitivity. Finally, to conclude, I would like to say that this proposed assay system is a novel combination of nanotechnology, molecular biology, clinical and medical biotechnology for the development and validation of detection technique for the detection of transformed cervical cancer cells, and may prove advantageous over the already known detection methods. Thank you.
Comment by myself. This is a very good, you've got a simple system. We have got commercially available systems which can detect E7 protein, but those are all expensive.
Yes, ma'am.
And has this been tested only in 25 samples as you, or have you done on a larger?
No, presently we have tested only on to 25 samples.
Because whenever a new test is developed, when the samples are small in number, you always get 100% sensitivity specificity.
Yes, ma'am.
The proof is you have to do it in much larger numbers.
Yes, ma'am.
And in this place, you are lucky. There's no shortage of samples with an active gynecology and cytology and pathology. I think this is a very promising, and with the cost of 100, you should go ahead and do it on much larger samples to show its sensitivity before it will be adapted by other people.
Yes, ma'am.
If not,
I will ask one if there is no other question. This E7 seems to be critical protein.
Yes.
Can it be a target also for, what is the function of E7?
So this E7 protein, it is basically it is a pro-mitotic factor. So as I mentioned, there was an E6 protein and E7 proteins. This E7 protein is a pro-mitotic factor because the cancer cells, they have to replicate and proliferate. So this is the one which then inhibits that retinoblastoma.
It's a protective protein. It's a protective protein.
It is an oncoprotein. It helps in replication, proliferation, differentiation.
No, it helps in cancer or blocks the cancer?
It helps in cancer. It promotes cancer.
So,
it is an oncoprotein.
So if function is blocked, then it could be a good target, is it?
Yes, sir.
Are there molecules to block its function?
Yes, there are, because this has to bind with the retinoblastoma protein.
Okay.
So there are drugs which have this capacity that they can bind to the retinoblastoma, and then its function can be taken up and not to cause cancer.
You.
Thank you, sir.
Sir, I had a small question.
Yeah.
Very interesting work. I just had a small question, probably out of ignorance. You said that HeLa cells are not detected. So is that good or bad? Because I think HeLa are HPV 18 positive. So
Yes.
because HPV 16 and 18 both cause cervical cancer. So if you're missing HPV 18, so is that good or bad?
Sir, ours is a test which is specific for 16E7. The antibodies which we have developed are specific for HPV 16E7. And since SIHA incorporates HPV 16E7 genome, only that is being detected. So it is a positive result because it showed specificity only for SIHA. If it shows for HeLa, then it would be cross-reactive.
Oh, but won't you miss the HPV 18 positive?
For that,
cancer
we'll have to
cervical
overexpress HPV 18E7 and then send antibodies generation for them and then incorporate into this kit.
cancers? Great. Thanks.
Thank you, sir.
Okay, thank you.
Thank you, Srishty. Our next paper presentation is by K.M. Renu, Ph.D. student, Department of Biophysics.
Very good morning to all of you. Today I am presenting here the 5-LOX protein is a blood-based biomarker and therapeutic approach for the targeting of breast cancer. I will go through the introduction, background, aims and objectives, methods and result, conclusion and futures aspect. So introduction, according to the WHO report, there is a 2.2 million women is diagnosed with the breast cancer in 2021 and 7.8 million women is living with the breast cancer according to the 5-year plan. And there is the several risk factors which are included the older age, family history, genetic mutation, early menarche stage and late menopause. So there is a 30 to 40% patients known their risk factor, but 60 to 70% patient there is not known their risk factor. According to the Globocon record, this is the pie graph of the all the cancer, and there is the 11.1% is the cases of breast cancer which is found. There is the several types of the breast cancer based on the origin and the hormone status, ductal, lobular and connective tissue, luminal A and basal light receptor, which is the triple negative breast cancer. So there is the till now there is the no clear cut value of the any particular protein which can be used as a blood based biomarker for the diagnosis, and this for the diagnosis of the breast cancer mammography is used, but this is showing the less sensitivity and specificity. So here we are trying to find out the particular biomarker for the breast cancer and targeting of the breast cancer. So here we will focus on the lipoxygenase, 5-lipoxygenase, which is an inflammatory protein which is enhance the proliferation of the breast cancer cell line invasion and metastasis. This is involving in an objective to find out the novel and more accurate non-invasive diagnostic marker for the detection of the disease and develop a new therapeutic modulator. Objectives of our study to evaluate the serum level of 5-LOX protein in the breast cancer and healthy control to design the therapeutic peptide modulator against the 5-LOX protein. So this is the 5-lipoxygenase protein, which is convert arachidonic acid into the 5-hettase, and these are involved in the mitogenesis and enhance the expression of the anti-apoptotic proteins. So methods, workflow for the finding of the biomarker for the breast cancer. We isolate the serum sample and we quantify the 5-lox protein in the serum sample with the help of SPR. Then we statically analysis in the 2 groups, patient and the control one. For the therapeutic target, we design a peptide modulator against the 5-lox protein, which is involved in the cancer promotion. And we check the binding affinity of this protein with the peptide with the help of the SPR. And then we inhibit the activity of 5-lox protein with this peptide. And here is the, we design a siRNA for the 5-lox protein, and we check the cell viability of the breast cancer. And then we check the expression level of 5-lox protein with the help of Western blot. So this is the demographic data of all my cancer patient samples and the healthy. This is the 30 samples, and this is the characteristic which is I use in the study. So firstly, I evaluate the concentration of 5-lox protein in the serum sample. So first we isolate the serum and then we immobilize the antibody against the 5-lox protein on the sensor chip. Then we pass the serum sample and then we quantify the concentration of 5-lox protein. So here we can see the difference. I got the breast 5-lox protein higher in the cancer patient, and this is the there is the control one. This is showing the low expression of the 5-lox proteins. So this is the ROC value of the previous data one. So we got the sensitivity 80% and specificity 83.3%. So we can see, we can find out here, it can be used as a blood-based biomarker for the diagnosis. So this is the 5-lox protein structure, and we design a peptide modulator against this activity and which inhibit. So we design a YWCS for which is have the capability to inhibit this peptide. So for the binding affinity for checking, we immobilize the 5-lox protein on the chip, and we run the different concentration of the peptide modulator. And so here we can see that this is showing the higher, when we use the higher concentration of peptide, it is showing the high binding capacity. For the this is the activity assay. We check the 5-lox, YWCS is inhibiting the 5-lox protein. So as we are increasing the concentration of this peptide, this is inhibiting the activity of the 5-lox protein. siRNA is designed for the particular 5-lox protein. So we design the siRNA and we check with the help of the MTT assay. So it as we decreasing the concentration of siRNA, it's showing the higher cell viability. So this is the Western blot, which is inhibit the we use the siRNA which inhibit the expression level of the 5-lox protein, which checked by the Western blot. So when we use the higher concentration, it's inhibit the expression level of 5-lox proteins. So conclusion on the basis of this study, we conclude that the 5-lox protein can be used as a diagnostic biomarker for the breast cancer, and YWCS, this peptide we synthesize in our lab, it can be have the inhibitory efficiency which further regulate the development of the breast cancer growth. Future aspect, we can use this 5-lox protein as a blood-based biomarker because it is showing high sensitivity and high specificity. 5-lox is a good blood-based biomarker, non-invasive biomarker, which can be used, and it is easy to handle, simple and cost-effective method. And YWCS have the anti-cancer capability, which can be further used as a therapeutic target in the breast cancer. Thank you. Any question?
Thank you, Dr. Renu. How good would be this as a biomarker to totally separate out breast cancer? You know, you have taken healthy controls.
Yes.
Have you taken other benign breast diseases? You know, the main problem always is benign breast diseases vis-a-vis cancer. So have you done that studies that, taken instead of healthy controls, benign breast diseases? Those have not been done.
Actually, we collected sample of all the 3 stages, first and second and third. So
No, that I understand. You have taken breast cancer cases.
Yes.
You have taken healthy controls. And you have not taken as a control
Yes.
group the benign breast
no,
disease.
ma'am. Yes, we.
And how much was the overlap between benign cases and the cancer cases? Because when you showed the scattergram, there was quite a lot of this thing, although your ROC curves gave you this thing. So do you think this can be used, or it needs much more studies?
Yes, ma'am, but it is showing the cutoff value now more than 4 nanogram per microliter,
Okay.
so we can use this protein.
Is there any question from the
Yes, sir.
audience?
I had a question.
Oh, okay.
Which was related to what Professor Verma asked, that what were your controls which you used to detect that serum levels were higher in breast cancer patients?
So same age group we use as a control, female only. Like
Were they suffering from any particular disease?
yes, sir.
Were they suffering from any
No,
particular
sir. Healthy without any suffering.
So, since lipoxygenase is an enzyme involved in prostaglandin synthesis, I think you should have controls which have some element of acute inflammation or chronic inflammation in them, because they will definitely show up increased level of lipoxygenase in the serum especially. So if you have asthmatics or some other acute inflammation, which I think Professor Verma also said, that you should definitely check them out as controls before saying that lipoxygenase is a breast cancer marker.
So that's why, sir, we collect the sample now. Healthy, those are not having
You are
any
not
disease.
getting my point. Any inflammation will have increased level of lipoxygenases. So acute inflammation, chronic inflammation is a predisposing condition for most cancers. So you will get false positives in a lot of inflammation cases. So
Yes,
you should
sir.
keep them as controls before you say that lipoxygenase is a blood-based biomarker for breast cancer.
But it is not more involved in the inflammation now. This
Have
is
you checked?
Yes.
Have you checked?
Yes, sir. This is not involved in the as
Have
the
you
inflammatory.
checked? Have you checked in serum of inflammation patients? If you have not, how can you say that?
Okay, sir.
That.
Okay, sir.
But with one small comment.
Yeah.
In any case, this could be a good target for you made peptides, but you made only one peptide. How did you make this peptide?
No,
against-
sir. We design more than 5 peptides, but those are having the more affinity.
They have
Those-
less affinity.
Yes, sir. Then we
This has
select.
the most affinity.
Yes, sir. Then we select this peptide.
But how did you design? What was your basis of designing peptides for this?
Actually, sir, my senior is also working, so I'm not independently design. So with the collaboration, we design.
Show the affinity.
Yes, sir.
You showed 10 to the power minus 8 binding affinity.
Yes, sir. We check the binding affinity with the help of activity assay.
Okay, good.
Thank you, sir.
Thank you, Renu. Our next paper presentation is by Apoorva Challa, PhD student, Department of Dermatology and Venereology.
Girls, female power. Female power. All girls.
Good morning, everybody. I am going to present my work on omics-based diagnostic markers of bacterial vaginosis in reproductive age Indian women. The work was done as a collaborative effort between the departments of dermatology, NMR and MRI facility, gynecology and microbiology at AIIMS, and CSR IGIB. We would like to state that the authors declare no conflict of interest. The work received funds from Intramural Collaborative Research Grant by AIIMS, and we would also like to acknowledge ICMR for granting me the Senior Research Fellowship Award. Lactobacilli are considered hallmarks of vaginal health, and they maintain vaginal homeostasis by production of lactic acid and hydrogen peroxide. Perturbations of the vaginal ecology owing to several endogenous and exogenous factors such as hormonal changes, menstruation, and sexual intercourse often lead to an increase in anaerobic pathobionts, which may lead to bacterial vaginosis. Bacterial vaginosis is often dismissed by women as a mere nuisance infection owing to the symptoms associated with BV, which include vaginal itching and malodorous vaginal discharge. However, as many as 50% of BV-positive women remain asymptomatic. If BV is untreated, it leads to increased susceptibilities to sexually transmitted infections, infertility, preterm birth, and spontaneous abortion. The existing diagnostic modalities either clinically evaluate symptoms using EMSIL's criteria, or laboratory-based Nugent scoring methods are used to evaluate the presence of various bacterial morphotypes. While the former is inadequate to diagnose asymptomatic infections, the latter requires microscopic facilities and skilled expertise, and is often subject to inter-observer variation. To address the existing limitations of the current diagnostic modalities, we decided to explore the potential of microbiome and metabolome biomarkers in BV diagnosis with potential for point-of-care test development. For this purpose, we collected vaginal swabs from 283 reproductive age group women in the age group of 18 to 45 years and assessed for BV using gold standard diagnostic criteria, which is Nugent scoring method. 40 BV positive women and 40 BV negative women were included in the study. Vaginal swabs were used for DNA isolation and sequencing of the V3-V4 region of 16S rRNA gene. Vaginal washings were subject to NMR-based metabolomics studies. Data obtained from these two methods were then subjected to machine learning analysis for identification of diagnostic biomarkers. The raw data helped us identify that the microbiome associated with BV is highly heterogeneous in comparison to controls, where controls are largely dominated by Lactobacillus species, most frequently observed being Lactobacillus iners and Lactobacillus helveticus. Statistical analysis identified that BV-associated samples exhibited a higher species richness and species diversity, and there was a considerable difference between the community composition of the two groups, as suggested by the beta diversity plot on the right. The most important features were Egathelia species, Snithia, Atopobium, and Dialyster, which were elevated in BV-positive women, and Lactobacillus helveticus, which was higher in control samples. Vaginal metabolome gave us an idea of the functional output of the ecosystem, and there was an increase in short-chain fatty acids such as acetate, pyruvate, succinate in BV-positive women, along with biogenic amines such as cadaverine and putrescine. Healthy control women, on the other hand, had higher abundance of lactic acid, glucose, and maltose. Statistical analysis identified that maltose, acetate, and formate were the most important biomarkers using univariate analysis. Multivariate analysis, which is depicted as the PLSDA plot on the bottom left, shows a clear segregation between the two groups. The important metabolites that contributed to this segregation were maltose, acetate, formate, succinate, and lactate. Overall, we identified that lactate and maltose were higher in control samples, whereas acetate and formate were upregulated in BV-positive women. Finally, the data was subjected to machine learning analysis, where the data was split into an 80 to 20%, where 80% of the data was used for training of the model and 20% was used to test the ML-derived biomarkers. The most important features were Snithia amni, Gardnerella vaginalis, Aerococcus christenseni, Megaceras species, and Snithia sanguinigens. The 5 species were used to construct the model, which exhibited an ROC-AUC of 0.9 and diagnostic accuracy of 0.83. Similarly, metabolome data identified maltose, acetate, and formate and lactate as important microbial-derived, ML-derived metabolite biomarkers, and the ROC-AUC of the model was 0.94 and accuracy of 0.88. To conclude, the microbiome-based model was good enough to predict BV with an accuracy of 0.83. However, the metabolome-based model performed better with a higher diagnostic accuracy, suggesting that the translational potential of metabolite biomarkers may be used for development of point-of-care tests. In the next phase of our study, we have proposed to use these metabolite biomarkers in the development of a multiplex paper strip-based colorimetric assay for detection of bacterial vaginosis. The idea is to develop a self-test kit that women can use by themselves to assess for any vaginal alterations. And the target are all reproductive age group women, who account for about 22.2% of our population. So both symptomatic women who hesitate to visit OPDs for clinical examination owing to the embarrassment associated with the symptoms or the hassles that they face in undergoing physical examination, and asymptomatic women who are probably at a greater risk for the adversities associated with BV can assess for alterations by themselves. early diagnosis and treatment may prevent sexual and reproductive adversities associated with this merely nuisance infection. Thank you.
You want to start? It's a
Let me turn it off.
important thing would be if you ultimately develop the paper
Yes,
test
ma'am.
for it. So it's a good this thing. Probably you need to extend this work to more cases and work on the development of.
As a part of our proposed work, we wish to validate in about 300 women to assess for the accuracy of this test.
Anybody from the audience? Yeah.
Thanks.
Hello. Yeah. Very professional. Thank you.
Thank
so I have
you.
one question because your essay is based on metabolites.
Yes, sir.
So what do you think the effect of diet is going to be? Because the diet will impact the secretion of lot of metabolites because all of these are dietary components, lactate, maltate, and couple of others.
Yes, sir.
Thank
So
you.
diet will have an impact, a role to play. And we also understand that glucose and maltose were higher in control samples. But however, at the same time, a lot of bacteria that are involved in the disease, increased production of short chain fatty acids are also seen. So like you suggest, diet will have a role to play. And we've not assessed for it in our preliminary data, but I think we would look at it.
Just a small question.
Yes, ma'am.
So, very good presentation.
Thank you.
Just want to add, can you elaborate what are the bacteria which are producing formate? That is my first question. And just a comment, the diet will not have direct involvement as of now because these are the core microbiota of the vagina. So it's not that by taking a diet, your microbiome of vagina will change in a day. Of course, the gut microbiome will. So that's the comment in response to the question which was asked. And thirdly, you have to really see that what are the metabolic end products you are talking about. You should also take a higher proportion of gut anaerobes and see what is the metabolite they are producing. So that will enhance your work, So maybe you can answer the first question.
Thank you, ma'am. So formate, as we know, Prevotella and Mobiluncus are known producers of short-chain fatty acids in the diseased condition, and both are associated with bacterial vaginosis. I think to answer Sir's question, there are studies which have correlated diet with glucose levels, one of the biomarkers that we saw in our study. However, maltose stood out as more important. So it might not have an effect immediately, but then it has been identified as one of the reasons why certain women are more prone to developing BV in women with higher starch diets. Studies have been done previously.
A short question. How specific is it, or is it a generalized indicator of vaginal colonization? Because the metabolites that you mentioned will probably be increased in any sort of colonization, or is it specific for, because any bacterial or fungal colonization of the vagina will produce those metabolites?
So they might produce the same metabolites, but we have correlated with the metabolites that we found with the bacteria that were associated with BV. And based on previous literature and our own findings, there seems to be a direct correlation. I wouldn't deny that there other conditions may not produce this, but the correlation in our study is evident. So
the controls that you used were healthy controls?
Yes, sir. So
So
women who did not...
Did you use any other diseased condition in which there is bacterial colonization? So just asking about how specific are your markers that you're saying?
So we've not done that. That is, I think, a point that we will consider.
Yeah, thanks.
Is there any question?
Extending to that questions only, did you associate or dissociate any comorbidities in these patients which you take as cases? And what are the other confounding factors as far as metabolomics is concerned in your case? Can you highlight on this?
Sir, we had excluded women who were receiving any form of treatment for such as diabetes and other comorbidities. Women who were receiving treatment for those were included. For metabolites, so we had excluded women who reported recent sexual intercourse, women who were on oral contraceptives, or women who were menstruating, all of which have a direct influence on the bacterial flora associated with the disease. But whether or not it has a direct influence on metabolites is something we haven't looked at. But yes, it does affect the flora, the vaginal flora, because sexual intercourse is known to increase concentrations of Snithia, and menstruation is known to be associated with the same species that are found in BV, all of which lead to transient fluctuations in the flora, but do not persist to develop BV. So that's one of the reasons why we wanted to focus on metabolites, because the functional output of the vaginal ecosystem is maintained by the flora despite the transient fluctuations.
Okay, there will be sole cases of bacterial vaginosis, or there will be comorbid associated bacterial vaginosis. So
Okay.
that would be a real challenging situations to make that this metabolite is going to make the dent in producing the biomarker.
Sure, so we'll keep that in mind.
Thank you. The final comment. Everybody is talking about biomarkers.
Yes.
Nobody is talking about developing drugs. Which is more important?
I think only if we detect an infection, especially an asymptomatic infection like this, can we talk of treatment. That is my first opinion of this. And based on the bacteria that we've looked at, we can also have targeted treatments, because most of women with BV tend to relapse within 3 to 6 months of treatment, because the entire flora is not affected by the current treatment method. So
Thank you so much.
Can I make one more comment, please? Okay. I think it might help if you have a control group with Candida and Trichomonas,
all right, sir.
Because those are common infections.
We looked at Trichomonas as a part of our study, sir, because it's one of the common infections that are seen with BV.
Okay, thank you very much.
Thank you so much.
The
Thank you,
best.
everybody.
Marks. Okay.
Thank you, sir and madam. I request Professor Puneet Kaur to felicitate the chairpersons with a Pachera plant as a token of our appreciation. The plant symbolizes prosperity and good fortune.
Look at this out. I think this is an exception, because all the people here are females here.
Thank you, sir and madam. The next platform presentation is by MBBS and MSc students. For this session, I would like to welcome the honorable chairpersons onto the dais, Professor A.K. Mahapatra and Professor Pramod Garg. Professor A.K. Mahapatra served as Director, AIIMS Bhubaneswar, and was formerly Professor and Head, Department of Neurosurgery at AIIMS, New Delhi. Professor Pramod Garg is currently Executive Director of Translational Health Science and Technology Institute, Faridabad. Sir is also Professor of Gastroenterology and Human Nutrition at AIIMS, New Delhi. The first paper will be presented by Ishan Gupta, MBBS student.
Good morning, and before we start, I thank the director, dean, and the faculty to call us. It's a wonderful opportunity, and research is a mainstay of medical science. And if you all look at the research in medicine, AIIMS stands to be third in the world. So I congratulate the students, faculty, and the scientists, PhD students, for their good work. Thank you very much. Pramod, you want to speak? We can start with the first paper.
A very good morning to all present. My name is Ishan Gupta, and I'm currently a final professional MBBS student. The topic of my presentation is prognostic implications of WT1 gene expression and flow cytometry in primary acute myeloid leukemia. So our research was funded by Department of Science and Technology, Government of India. AML is a heterogeneous disease characterized by abnormal differentiation and uncontrolled proliferation of undifferentiated myeloid progenitor cells in the bone marrow. Newer prognostic markers such as gene mutations including nucleophosmin 1, FMS like tyrosine kinase 3 and Wilms Tumour 1 have been identified. Wilms Tumour 1 gene is located on chromosome 11p1 and it encodes a zinc finger transcriptional regulatory protein which binds to DNA and is involved in cellular growth and metabolism. Recurrent somatic mutations in WT1 appear to occur in approximately 6 to 15% cases of de novo AML. These mutations cluster mainly in exons 7 and 9. In addition to the mutations, it has also been seen that WT1 gene expression can vary along with different occurrences of AML, and it has been seen that it can have significant reduction in many cases, showing morphological remission at the end of induction therapy in cases of primary AML. And enough characterization of these therapeutic targets and prognostic markers, especially WT1, has not been done adequately in the scientific literature. Therefore, WT1 gene expression may have a potential role as a marker of disease progression. So it was in this background that we decided to investigate the prognostic role of WT1 gene expression in patients of primary AML in combination with flow cytometric immunophenotyping. It was a prospective clinical research performed at our tertiary care center. So we collected peripheral blood samples and bone marrow specimens from 54 immunodiagnosed cases of AML. Morphological diagnosis was done on the basis of these specimens. They were subjected to flow cytometric immunophenotyping. They were diagnosed and the further classification of AML was done. mRNA was extracted from the peripheral blood or bone marrow samples, and the WT1 gene expression using real-time quantitative PCR was calculated. So this is the age and sex distribution of our patient cohort. So there were 3 pediatric patients and most of the patients were in the middle age group. We had 41 males and the rest were females. This is a peripheral blood film showing myeloid blasts, and these are myeloperoxidase positive blasts in the bone marrow aspirate. This is a representative plot of flow cytometry plots of a case of AML. So cells were gated based on side scatter and forward scatter, and it shows a positivity for CD13, 33 and 117, which are myeloid specific cluster of differentiation markers, and low positivity for CD34, which is a stem cell marker. We also did mutational analysis because we had to classify AML according to the WHO classification. So the AML ETO or the RUNX1, RUNXT1 mutation was detected in a subset of cases. CBFB MYH11 was also detected, and similarly PML RARA was detected by fluorescence in situ hybridization. These were classified as AML with genetic abnormalities. We also detected nucleophosmin 1 and FMS like tyrosine kinase 3 internal tandem duplication mutation. So in all our patients, treatment with induction therapy was started. A total of 11 patients relapsed or did not attain morphological remission, that is blast count percentage less than 5%, less than equal to 5% in peripheral blood or bone marrow after induction therapy. 39 patients attained morphological remission after induction therapy, and 4 patients died before completion of induction therapy. So they had to be excluded from the calculation of the WT1 gene expression. This is a PCR, real-time PCR amplification plot of the different samples. The straight line above 0 is the threshold line, and where each particular PCR reaction cuts the threshold line, the cycle value corresponding to that is called the cycle threshold or the CT value. So the double delta CT analysis method was used for relative quantification of the quantitative PCR. Delta CT values were calculated by normalizing the WT1 gene expression with housekeeping genes. And we found that the WT1 expression in the relapse group was significantly higher than in the complete remission group. Also, we found various correlations which included that the WT1 gene expression levels were inversely correlated with platelet counts, hemoglobin levels, and positively correlated with blast and flow cytometer counts, particularly percentage of CD34 positive cells. A previous study in 2018 has revealed that the WT1 gene expression can help divide AML patients into low, middle, and high risk groups. And the WT1 gene expression can be used to evaluate therapeutic response in middle as well as high risk cases of AML. Another study has showed that sequential monitoring of WT1 mRNA is of value for early detection of hematological relapse in AML in remission after chemotherapy or stem cell transplantation. And a recent study has highlighted that the status of WT1 gene expression before an allogenic hematopoietic stem cell transplantation is a strong prognostic factor for both overall survival as well as relapse risk. And that WT1 can also prove to be a reliable marker for monitoring the status of measurable residual disease after transplantation. So I would like to conclude by saying that WT1 gene expression is proposed to be a prognostic marker in AML. The combined usage of flow cytometry and WT1 gene expression contributes to an improved detection rate of relapse and may be used to monitor MRD status, assess the treatment efficacy, prognosis, as well as predict the risk of recurrence in cases of AML. Other factors which can be used for prognostication include blast count percentage, performance status of the patient, as well as other molecular markers. Hence, molecular analyses with wider panels in AML cases as well as prospective studies with larger sample sizes are needed to further validate these results. These are the references I used. Thank you.
We have 2 minutes for questions. Any questions from the floor, please? Nice presentation. Could you tell me, do you think the WT1 expression is the one which is correlating with the response rather than that the blast count and other abnormal cells are going down, and so the expression level will go down overall?
Actually, sir, the WT1 gene is in itself a part of the molecular pathway of the
That's right.
this thing.
So to say that WT1 expression will determine the response may not be correct. Your level will go down in those who have remission because those cells are gone, and you won't find expression.
Right, understandable, but the correlation is not always true. It depends on the transcriptional modifications which the WT1 may play a role, and then it can affect the blast count as well as the CD markers which are expressed.
What percentage of patients had this WT1 expression, the mutation?
Sorry?
What percentage of patients had this mutation, WT1?
So we have one thing is the mutation of WT1, other is the expression. How what is the CT value or what is the expression?
Out of 54, how many had the mutation?
So we did not do WT1 mutation. We did the expression study. So it was a study for expression of WT1. We did the analysis of mutations which are regularly done in AML. That is the AML ETO, CBFB, MVH11, and PML RARA.
One question. You yourself told that its limitation is small number. All 3 papers or 4 papers presented in the morning, this look like a preliminary study.
Right.
Why don't you start the study with proper protocol, taking a statistician and know that what is false positive, false negative, dropout cases, 4 patients died.
Right, sir.
This should have been taken into consideration. Otherwise, this paper cannot go to a very good journal with good impact factor because number of patients is small and no exclusion, inclusion criteria, no control group, nothing. It's just a analysis of data and without proper planning of a research study. I think you should have better planning.
So this is a preliminary study which we have done. We will include more patients.
Preliminary, we are talking a AIIMS research presentation. It cannot be preliminary. How long you can have preliminary study?
So we are including more patients because the median follow-up is also not as much. So there is also more time length for that.
Okay, thank you. Next one.
Any other questions?
Time
No.
is
Okay,
up.
thank
Time
you.
is up.
Thank you. So we move on to the next presentation.
Our next presentation is by Shivani Srivastav, MBBS student.
Good morning, everybody. This is Shivani Srivastava, 7th semester MBBS students, presenting the paper on comparative analysis of immunohistochemistry and MECHAN amplification status in pediatric neuroblastomas. A neuroblastoma is an embryonal tumor of neural crest origin and is the second most common solid tumor in children. Age, stage, and histological subtypes are the important prognostic parameters in neuroblastoma. MYC amplification is the single most important molecular genetic predictor of worse outcome. Thus, detection of MYC amplification itself moves a patient to high-risk category irrespective of the age, stage, or histology. ICMR consensus document lists FISH as an essential investigation only for stage 3 and 4 patient, while it is desirable in all other patients because of its lack of availability in majority of the hospitals in India. FISH is the gold standard test for detection of MYC amplification in neuroblastoma cases. However, it is not an automated technique as it requires trained personnel to perform it manually and interpret it. It requires considerable expertise and the equipment is specialized. The reagents and probes are also costly. Thus, FISH is not readily accessible in most diagnostic labs in middle to low income countries like ours. 2 studies have described MYC amplification in neuroblastoma in Indian context. First study described MYC amplification in 21 of the 103 whole cohort patients, of which 3 patients were found to have 14% amplification. However, they did not correlate MYC status with other parameters. The other study performed FISH on 56 FNAC samples and found amplification in 48% of them and more frequent in the undifferentiated subtype. However, no correlation with age, stage, or overall survival was made. Immunohistochemistry, on the other hand, is a routine technique performed in most diagnostic pathology labs in India, relatively inexpensive, and pathologists are also well versed in its interpretation. There are only occasional studies in which MYC and IHC has been performed in neuroblastoma, and there is also not much data on its role as a surrogate prognostic marker for MYC amplification testing. We therefore conducted this study to evaluate immunoexpression of MYC in neuroblastomas and correlate the results of immunoexpression with amplification by FISH to assess if MYC and IHC can serve as a low-cost surrogate marker for MYC amplification, as I've earlier described the feasibility of performing MYC amplification. The neuroblastomas which had adequate tissue were taken up for FISH and IHC analysis. A normal signal pattern for IHC interpretation is 2 orange and 2 green signals per cell. More than 10 MYC signals and a ratio of MYC to CCP probe of more than 4 is considered as amplification. And nuclear staining of moderate to strong intensities in more than or equal to 50% of the tumor cells was considered as immunohistochemistry positive. 38 cases of neuroblastoma were included in our study. Most of our study participants were below 6 years of age with a slight male preponderance. Undifferentiated histological subtype was the predominant one. Nearly 3/4 of the patients for whom stage data was available had metastatic disease. 11 out of 38 cases showed MYC amplification by FISH. These images show amplification with numerous orange signals and a normal MYC with 2 orange and 2 green signals. There was no significant difference in MYC amplification by age, stage, sex, and also histological subtype. However, amplification was only found in undifferentiated and poorly differentiated subtypes. Immunohistochemistry staining showed 10 of the cells to be immunopositive for MYC. The negative staining cells either had poor to moderate staining. Here are the staining patterns shown below. Positive, negative in less than 50% and more than 50% cells. We obtained concordant results in 29 of the 38 cases. Discordant cases included 9 FISH positive IHC negative cases and 4 FISH negative and IHC positive cases. Cohen's-Kappa correlation statistics showed moderate agreement between FISH and IHC. Compared to FISH, IHC had low sensitivity and low positive predictive value. Only few studies till date have compared MYC and IHC with MYC amplification, and none in the Indian context. Most of these studies showed higher concordance, but these were performed with an autostainer while we performed it manually. One study also showed significant association between MYC and IHC positivity and significantly shorter event-free survival. However, we could not comment on the outcome data because of our lack of clinical data. Santiago et al. Performed similar test in their study setting and found IHC to be cost-effective strategy and could be used as a low-cost surrogate marker for MYC amplification by FISH. However, we did not obtain the same results. MYC amplification was identified in 29% of the neuroblastomas in undifferentiated and poorly differentiated subtypes. Immunopositivity was present in 27% of the cases with moderate agreement. It was positive only in half of MYC amplified cases and few non-amplified cases. As the sensitivity of IHC was low, it cannot be used as screening test to perform FISH. False positive cases by IHC may lead to patients unnecessarily receiving higher dose of chemotherapy as a positive predictive value is low. Although specificity and negative predictive value are fairly high, it is not desirable that even a small proportion of patients with amplification miss out on receiving an escalated chemotherapy as they are expected to have an aggressive outcome. To conclude, MYC and IHC is not an appropriate surrogate marker for determining MYC and amplification status in neuroblastomas. Lack of sufficient clinical information and outcome data had been a limitation of a study. Nevertheless, we suggest that establishing a few centralized facilities in country to perform gold standard FISH to determine amplification status is a better alternative than seeking a low-cost surrogate immunohistochemical marker. Adequate budgeting for such prognostic interventions requires data to support planning. A national neuroblastoma registry will aid in planning for such centralized MYC testing facilities. Thank you.
I think I should congratulate her as a medical student she has presented, and it is the responsibility of all our faculty to involve medical student in some degree of research. So, any question from the audience? I think give her a big hand and ask question. Question from the audience.
Shivani,
Yes,
you said FISH is the gold standard. Any other technique to look for the MYCN expression or mutation? Because ultimately it will relate it to some genetic alteration.
Yes, sir.
So it is possible that you are comparing it against FISH as the gold standard, which may not actually be the most sensitive or specific.
The studies that were published have proven it as a gold standard, and that is the reason it has been the sole test for detecting MYC amplification for all our neuroblastoma cases. I don't think other options were explored.
Presented two series, 210 cases and 94 cases, and your is 37 cases.
38
so you
cases.
have to improve your research.
Yes, sir.
What do you plan?
How
will you improve your research?
We are actually expanding our sample size to include more cases, more retrospective and also prospective cases. The work is still going on in the laboratory, sir.
Dr. Sinha has a...
Yeah, just
the number of cases is only one. But what are other limitations?
Yes, sir.
Number is a very good, very important limitation. But what are the other limitations in your study? You did not compare with any standard other things, ne? See, FISH was not compared with any other gold standards, ne?
there is no other.
So there is no,
We took it as a
there is no
gold standard.
comparative analysis with other methods.
Yes.
Dr. Sinha, please.
Just a question that, ultimately, when a neuroblastoma gene is amplified, you would be expecting that it acts through the protein. So it sort of seems common sense to expect more protein in your IHC.
Yes, sir.
I mean, is there any methodological issues in IHC which could be used to improve the catch, so to say, of the protein? I mean, this is thing. I mean, you did your routine IHC, but
Yes.
are there ways by which you could sort of improve the detection limits or have it more sensitive than they? I mean, theoretically speaking, I know it's not available, but
Sir, it was our cutoff to take it as what cases to be taken as positive. We only took 2+ to 3+ in more than 50% of the cells to be positive. If we lowered our cutoff, maybe more cells would have been showing positivity. No, actually, sir, we didn't this time.
Thank you, Shivani.
Thank you.
Thank you, sir.
Move on to the next.
The next presentation is by Anjali Gaur, Department of Biochemistry, MSc student.
Okay.
Good morning, everyone. Myself, Anjali Kaur. I am a MSc student from the Department of Biochemistry. I'll be presenting my work on development of CRISPR-Cas12a based detection assay for clade C HIV-1. So I'll be starting with the brief introduction about CRISPR-Cas system. CRISPR is a highly diverse adaptive immune response of a bacteria that mainly utilizes the two main components. First is the guide RNA that is complementary to the target, and second is the Cas. Cas is the CRISPR-associated protein that mediates the cleavage to the target. So in this study, we have used the CRISPR-Cas12a system because it specifically recognized the single-stranded and double-stranded DNA as a target. And once it's recognized and bind and cleaves the target, it leads to the non-specific cleavage of single-stranded DNA molecules that are present in the vicinity of this enzyme. This special feature of this enzyme is used for the detection purpose. The rationale of this study is the early detection of HIV-1 is important for treatment, initiation, and prevention of transmission and disease progression. The current method of detection relies on real-time PCR and ELISA. However, these methods cannot be used for point-of-care nucleic acid detection. So the CRISPR-Cas-based techniques are highly specific, sensitive, cost-effective, and can be used as a point-of-care detection in low-resource setting. There are many studies on CRISPR-Cas-based assays, but till now, there are no reports so far for the detection of HIV using this assay. So we aimed to develop a CRISPR-based detection assay for detection of HIV-1. So in this study, we have designed the three major components. The first one is the guide RNA designing. As I earlier mentioned, guide RNA is complementary to the target and it also confers the specificity to the assay. So we downloaded the 86 HIV-1 clade C sequences, aligned those sequences and made the consent sequence out of those. Now the region with the maximum conservation is used for the guide RNA synthesis. This is the guide RNA synthesized. Here the red colored region is the Cas12 binding site of the guide RNA, and the blue colored region is the region that is complementary to the target, that is Pol protease region of the HIV-1. Further, we designed the primers against the Pol region that we used for the RPA. RPA refers to as recombinase polymerase amplification. It's an isothermal amplification. Here, we are amplifying the target at a constant temperature of 39 degrees Celsius for 30 minutes. Further, we designed the fluorescent probe. It is a single-stranded DNA probe that has at the 5' end a fluorophore attached to it and 3' end has a quencher attached to it. When the target is recognized by the CRISPR-Cas complex, parallelly the probe will cleave and the fluorescence can be detected. This is the brief workflow of the assay. So we took the blood samples of 16 HIV-positive infected individuals. We extracted the viral RNA, performed the cDNA synthesis, and amplified the target sample by RPA, and performed the CRISPR-Cas12a based detection assay. Now this is a one-pot assay. Here we are having in a single tube the amplified target, the guide RNA, the Cas12 enzyme, and the fluorophore. If the target is recognized and being cleaved, parallelly the probe is cleaved, fluorescence we have detected in the three different modes. First is the visual detection that can be used as a point of care diagnostic purposes. Second is the UV detection. And third is the fluorescence measurement, which can be used as a semi-quantitation. Now this whole assay is performed at 37 degrees Celsius for 15 minutes. Now, initially we standardize our CRISPR-Cas12 based detection assay on in IMC, that is an infectious molecular clone, HIV clade C positive DNA, and with the different dilutions of a patient cDNA, 748. Where we found that as compared to NTC, we are able to detect the HIV cDNA. This is a UV-based readout. Further, this is the same image as shown earlier. This is we have taken the readout visually, where change in color from pink to green signifies a positive detection, which can be seen by our eyes. So further, we've successfully validated the assay in 16 HIV-infected individuals. This is the UV-based readout, and this is the visual readout of the same. Further, the fluorescence we have measured on multi-mode reader. Here also we are able to detect the significantly high fluorescence in all the 16 HIV clade C samples. Here, interestingly, this AIIMS 619 is a low viral load patient, and with our assay we are able to detect the fluorescence in this sample also. This signifies the sensitivity of the assay. Further, we validated the assay for the non-specificity by setting up the cleavage with the healthy individual, and we could not found any detection, non-specific detection in the sample. Further, we took another disease control, that is HBV, for the assay validation, and we didn't found any non-specific detection. So the summary of the assay is isothermal amplification followed by guide RNA synthesis is designed against the Pol region, is successfully validated on 16 HIV-1 positive samples. The readout is taken on 3 different modes: visual, UV and fluorometer. No non-specific detection was observed. Hence, we conclude that the Cas12-based detection assay has been successfully developed for HIV-1. The assay in this study has been patented, has been filed for provisional patent by AIIMS. The future aspect of this study is the CRISPR-Cas12-based detection assay can be used to develop into lateral flow assay and quantitation of viral load. It can also serve as a potential tool to discriminate between different clades of HIV-1. Further, I would like to thank Dr. Kalpana Luthra, my supervisor, and Dr. Subrata Sinha, my co-supervisor, and my lab members, as well as I would like to thank AIIMS for filing the provisional patent, and the Department of Biotechnology, Government of India, the funding agency. Thank you.
Excellent presentation, Anjali. Very nice work.
Thank you, sir.
Any questions? Yes, please.
Yeah, Anjali, while your work is CRISPR-Cas can be used in various settings.
Yes, sir.
I do not expect that it is likely to be used for diagnosing HIV infection. The reason is that we have excellent serological tests, and even the lateral flow, which just detects antibodies, all of them have a sensitivity of more than 99% and a specificity of more than 98.5%. So really speaking, the role that you could envisage for something like this would be in case you are able to quantitate. And that too, you will need to cover all the clades and all the recombinant forms. Without that, the commercial potential would be very limited.
Okay.
Thank you.
Sir, I would like to answer the question. So first thing is that the serological tests that are available for HIV-1, it detects the antibodies. HIV antibodies, which are not being detected during the initial stages of the infection. First, that is the disadvantage of. The second is the NACO guide. According to the NACO guideline, it has been said that for the children below the age of 18 months, you cannot perform the serological test. You have to go for the NET test. So in this CRISPR-Cas system, we are able to utilize the both advantages of the both assay. First, point of care assay, where you could visually detect the detection can be done, and the second is you are highly specific, and even we found that in our study that this assay is highly specific for clade C. So that is the advantage, and you are detecting the nucleic acid. Despite
sorry. Anjali, thanks for the confidence of presenting your paper, but I would just like to say, do you have any other studies on other clades? As correctly pointed out, if you won't include all the clades, just because clade C is, common in our place, but you still have a heterogeneous population within the country. So you cannot just, so what are the other studies related to other clades on this?
We haven't checked, correctly pointed, we haven't checked for another clades. We will do, we have to standardize that. But the reason why I'm sure about that it's detecting the HIV-1 clade C is that guide RNA is highly specific. If there is any mismatch in the target and the guide RNA sequence, the enzyme would not act. The enzyme would not cleave the target, and the detection would not happen. So if we want to detect the different clades as well as the different types of HIV, we need to design more than one guide RNA. So guide RNA that is specific for HIV1 clade C, and that guide RNA that is specific for HIV2.
Thank you. Thank you, Anjali. That was excellent. We will close this session now and move on to the next.
Thank you, everyone.
Thank you.
I request Dr. Kalaivani to felicitate the chairpersons. For the next session, I would like to invite Dr. Kamal Gulati to conduct the proceedings.
A very good morning. Now we have the first keynote lecture session, and I welcome the Honorable Chairpersons, Professor P.P. Kotwal sir and Professor S.K. Khare onto the dais to chair the keynote session. Professor P.P. Kotwal, formerly Professor and Head, Department of Orthopedics at AIIMS New Delhi, and Professor S.K. Khare, Dean Research and Development at IIT Delhi. I also invite Dr. Amit Mendiratta onto the dais to deliver the first keynote lecture on artificial intelligence in healthcare. I request Professor P.P. Kotwal sir to please introduce the speaker. Thank you.
Good morning, everybody. I would like to congratulate Dr. Rama Chaudhary and the team for organizing such an excellent program. And I would also like to thank her for giving me an opportunity to participate and witness this academic program. I have the great pleasure to introduce Dr. Amit Mendiratta for this first keynote lecture of this program. Dr. Mendiratta is the joint faculty at the Center for Biomedical Engineering at IIT Delhi and AIIMS New Delhi. He has established the Center for Advanced Research and Excellence in Disability and Assistive Technology. He is presently developing new analysis and post-processing methodologies for quantitative biomedical imaging. He is a recipient of the prestigious Ericsson Innovation Award 2019 for his product robotic exoskeleton for upper limb rehabilitation. I invite Dr. Mendiratta to please deliver the oration. Thank you.
Thank you, sir. It's a real honor for me to come at this forum. So the topic I'm covering today is artificial intelligence in healthcare, and I have given it a subtitle of From Research to Clinical Deployable Solutions. So the story starts from human intelligence, and that's how the artificial intelligence will come into play, because everything we start from how human intelligence evolved, and that's how we automatize the machine for artificial intelligence. So I'll cover artificial intelligence, machine learning, types of machine learning, and their limitations for healthcare transformation in this world. So the features of human intelligence that we want the computers, algorithms, or machine to do are like humans can perceive and sense things. Humans can do logical thinking to reach a decision or a conclusion so that the final output can be delivered. Humans can self-learn the things by taking the decision, doing error and learn and correcting it, and improve the logic for better decision next time. They can compare many things, predict the future, understanding the past, and the advanced level of intelligence involves learning from the past to predict future, understanding the behavior or what other human person is thinking. That's what I want to predict. So all these are characteristic features of human intelligence we want artificial intelligence to actually imbibe. So artificial intelligence is nothing but science and engineering of making intelligent machines, especially intelligent computer programs in today's world. And when machines are designed in such a way that they can do the manual tedious tasks of human, they are called humanoid machines. And machine learning can simplify your job and day-to-day activities, and it's there in all of your hands today, which is in the form of Alexa, automated cars, or even Google Ads is an application of artificial intelligence. Machines can react like human, but are not there to replace. Trust me, it's a Hollywood gimmick, but that's not the reality. Let me show you an example over here. So this is a small girl who wanted to discriminate the lemons based on their size. And that's what she wanted to do with handpicking. But then she has devised a machine which is a gradient bar, and the diameter of the bar keep on increasing over a period of length. And she is able to cluster the lemons in different sizes. So this is an application of machine learning. It has made her job easy and simplified for the solution what she was able to do in a whole day's job. So this is nothing but a human intelligence designed for an automation. It is called rule-based artificial intelligence. You know what you need to do, you design it in the form of machine and deliver. When we don't know the criteria, what we need to do, and we still want machine to learn, which as a human I can't, we call it machine learning-based automation. So artificial intelligence is a bigger umbrella, which is making the machines intelligent. There are many subdomains, we'll come to it down the line. But machine learning is a small subcomponent of it, which is nothing but compresses of algorithms, statistical models, and tools to actually deliver the task. These are the mathematical operation, complex computational mechanics which goes behind the screen. And deep learning is a subtopic of machine learning, which is much more rigorous in computation processing, and for which we might not know what it's doing, but it still delivers the outcome. So we'll start with rule-based applications, because these are something which in healthcare has a tremendous scope of operation. So we learn from what human intelligence has already done over a period of centuries. So the expert knowledge is already available, and we try to take that expert knowledge, give them some mathematical operation, that is what we call rules, and these rules deliver the task and make the job easy. So these are very objective in nature, as you saw that the girl would have, the video which I showed you, the girl would have misplaced some of the lemons here and there, because starting at 8 o'clock in the morning and by 6 o'clock she will get tired. But the bars will not. They will still discriminate the lemons with as much precision as they were doing 10 hours ago. So these are objective in nature, they are fast. There are low error rates in rule-based systems, and they can automate the tedious manual task. And we have many rule-based machine learning algorithms running in your healthcare system today, such as screening OPDs. You enter the symptoms and it will tell you whether you belong to a medicine case, surgery case, and the system can be simplified. Emergency screening, image annotation, or marking the lesions, cancer, pneumonia. I will show you some examples down the line. Self-check kiosk, drug dispenser. Arogya Setu is a typical example of rule-based application, because when you log into the Arogya Setu, it used to ask you whether you have fever, cough, X, Y, Z, and then it discriminates whether you are symptomatic, asymptomatic, whether you should quarantine or no. These are typical expert knowledge, rule-based questions that has been put in the algorithm. Now, how does machine learning work? So it is as simple as you teach a kid, right? When a kid starts going to school and you say that this is a bird, how does a kid recognize that this is a bird? It actually tries to see that there are some features which can define a bird. Animal which has wings, animal which has beak, animal which can fly, it's a bird. And then over a period of time, it discriminates the types of beaks, type of feathers, and types by how it fly, and discriminate between different birds. So these are nothing but we call in machine learning features. Based on different features, the kid learns. Based on different features, the machine will learn. So you similarly give all that information to a machine, and then it try to make a pattern in it. So when you teach a kid, you basically give him task and it learns. And then repeatedly you take exams, right, to test whether it has learned or it is able to do what it delivered. Similarly, the analogy goes to the machine learning. There is something called training data. That is, you give the data with labels that this is what it is. and there is something called testing data. Once the machine is trained, you check the machine's efficiency to deliver the outcome. And there is called validation data, which is a completely different set of data which machine has never seen. And you try to see whether the machine is able to do the task with a completely unknown data that it has never encountered in its training or testing platform. So training, testing, and validations are three typical terms that I want you to introduce when you talk to anybody who does machine learning for your projects. There is a subcomponent called deep learning. It all starts with how brain works. So a neuron is in your brain do a complex task for all of us. It's a single neuron, 0 and 1, whether it will fire or not fire. But a combination of neuron with each other stores all the information in your brain and process and help you take the decision. Now imagine when these neurons are multiple, a lot of information can be processed by that single unit. And that is what is the concept of deep learning. That is how it evolves. And you must also see that in human brain, the memory or the tasks are stored by reinforcing different synapses to do a particular step or make a particular decision. And some of the synapses are retreat back if you want that memory to go away, right? And this formation and retretion of synapses is analyzed in deep learning as weights. We call them that the weight of this network is higher. That means this decision should be taken. A weight of this network is lower. This decision should be suppressed. And that is what mathematically it will do. So this is how a typical schematic diagram for deep learning algorithm looks. There is a input layer that you give the features. There is a hidden layer. We don't know how the network will perform. And then the output, what is you expect at the end of the process. Now this hidden layer can be multiple. As we know that as a human brain, there are many neurons which interact with each other. The more the complexity, the better it can learn. And the better it can go to a much micro level and take decision. And please note that I have written here, the network optimizes the weight, which is nothing but synapses in this neuronal network, for each relation to increase the output efficacy. It is same as like synapses work. It increases the weight of some of the pathway for a better outcome. It reduces the sum of the weights of the pathway to reduce the outcome. And that is how deep learning algorithm will typically perform. And what you get at in the end is there is an input. It does some advanced level of computation and you get an output. Now this machine learning as a full big umbrella can be divided into three subcategories for easy grasp. It's called supervised machine learning, unsupervised machine learning, and reinforcement learning. So supervised machine learning is a task-driven. We know what we want. We know what we want to deliver. There is a large input data, a lot of patient information, a lot of image, lot of signal that is required. And you know the output for some of them, and you train the machine over a period of time. Like, as I gave you example of bird. You give lot of type of bird images, lot of type of beak length, feather length, and it will learn that this is a bird and this is pigeon or this. So we'll come to the healthcare example for your easy understanding. But the supervised machine learning is very useful in predicting. There is a regression model that can go a long time. It can learn the characteristics of how the disease or cancer or pathology is evolving over a time, and it can model that and actually predict in future with some probability that what will happen down the line. Whereas unsupervised machine learning doesn't have that predictive value. They are not in regression in their properties. These are more cluster-based approach. You give input data and it can clusterize. Yes, no. A, B. COVID, yes, COVID negative. Pneumonia, yes, pneumonia negative. So these are like discrete decisions. It cannot do a continuous time series kind of analysis. But unsupervised data or unsupervised machine learning doesn't need you to identify and tell machine the features. These are some of the problems where you don't know how to make the decision and you want machine to decipher itself. How can I discriminate between different outputs? You tell me. I don't know as a human. This is much more advanced level of computation. Reinforcement learning is exactly like how we do. There is a system of reward and punishment. You expect something to deliver, and if it delivers in right direction, the machine will enhance the weights of that direction. And if it delivers in some direction where you don't want it to deliver, it will suppress that level of decisions. So giving you some examples of supervised machine learning, it's in categorical data like output. You put it in different baskets. Now there are some examples which I can quote directly here, like in your Gmail. Some of the mails will be labeled as spams. So this is nothing but how machine learning has learned that this mails has gone to multiple people. There are a lot of complaints, and this can be labeled as spam. So all of us use it. The cancer, put it in basket of benign or malignant image or hematological parameters, whatever data you mean, but the output would be benign, malignant. Grades of malignancy, chemotherapy response evaluation, whether the patient is responding to my treatment or not responding, and whether I can predict with the early changes that this will be a responsive or this will not be responsive. Because supervised, as I told you, it have a regression coefficient value as well. It can predict in time. So I can train that and see whether it works. Dementia, whether it is Alzheimer or frontotemporal dementia. COVID-19 is a very good example that in recent times we have seen a lot of applications in symptomatic and asymptomatic patients based on different aspects. Supervised machine learning as having a regression value, it's basically built a complex mathematical operation to decide what is the characteristics with respect to time for a particular parameter. It could be linear, as we all understand that linearly it increases. So I can predict in future without seeing the X value, what would be the Y value, because now I have got an equation for it. Or it can be something complex and non-linear, which I can't visualize. This is a display in 2 dimensions, but that could be a parameter in multiple dimensions, 5, 6 dimensions, which I can't put it on projector. You can't think as a human, you can't visualize it, but machine can. And that is what the beauty lies. So typical example of supervised machine learning, age versus dementia. With respect to age, how is the probability of progressing dementia? And if you add features of different Alzheimer's or frontotemporal or Parkinson's into it as a multidimensional point of view, it will perform still better because you are including more heterogeneity of population in your model. Infectious diseases has been explored a lot in artificial intelligence. Chemotherapy response evaluation, epidemiological models based on early to mid 2020, a model from Cambridge was published based on these models only, where the value of 89 came as the lockdown period to suppress the COVID effect. And all of you wonder why was the first lockdown placed at 90 days? Have you ever questioned yourself how the magic figure 90 days come? The first lockdown. It was based on a predictive model which was built in University of Cambridge in April, and that came out that more than 85 days would be good enough to suppress the COVID for further growth. Unsupervised machine learning, as I told you, has input data which is probably not labeled. We don't know how to discriminate it. As a human, I don't have any feature set that, okay, tumors looks like hyperintensity or hypointensity. I don't know, but I know there is a problem. And I want machine to decipher the parameters, how to discriminate between the 2 labels. So what it tries to do, it tries to make high intra-cluster similarity, that one cluster has a good overlap among itself, and low inter-cluster similarity. Now I can put it in the term of standard deviation or variability, that there is low intra-cluster variability, it's a tight cluster, or there is a high inter-cluster variability, the clusters are separated from each other. Now let me put you an example here. I have color-coded it so that you know that this is the output I am expecting. 3 different clusters. Now one is the tumor size in the x-axis and tumor aggressiveness in the y-axis. Now we know that there are 3 clusters. Now what I mean by low intra-cluster variability, the spread of each cluster has to be low, and high inter-cluster variability, the centroid or the mean of each cluster has to be separated from each other with a high distance. So mathematically, this intra and intercluster variability will be calculated, and a model will be built by a machine which will be like a complex function, that y is tumor aggressiveness, a complex function of tumor size, and it will have these two lines to discriminate between these two clusters. Now, whenever you get a new data point, it will try to just fit in this model. Where do I put this new data point in which cluster? As simple as that. Now, let me give you some more example. As we know, liver disease progress, we have a healthy liver, then we have steatohepatitis and cirrhosis, and sometimes the cirrhosis proceed to hepatocellular carcinoma. Now, let me put it in 3 different clusters. I want to classify based on some information, I don't know what, into healthy liver, cirrhosis, and hepatocellular carcinoma. The complex problem would be what cases of cirrhosis will actually proceed to hepatocellular carcinoma. I will not ask that question right now, but let's put it in 3 different clusters. I'll take the same examples like my earlier slide. We have 3 different clusters: healthy liver, cirrhosis, and hepatocellular carcinoma. Okay, I don't know what is feature 1 and feature 2. Feature means how do I discriminate between them. Now let me pose you some example, and I want some response. That would be really appreciated. Feedback is always helpful for learning, both for human and machine. If I say age versus height, how many of you would say I can discriminate between all the 3? Anyone? None? No, it's not a good feature set. If I say weight versus age, can you? To some extent, okay, high obese patient, old age, ठीक है, हो सकता है, but I'm not sure. Now if I give you, okay, LFT versus liver size, hepatomegaly, LFT. Yes, it has the information you can discriminate. Liver size versus texture of liver, nodules on liver, hard liver. Is it better than LFT versus size? Yes. Texture versus elastic properties, elastogram. So ultrasound and elastic properties, yes, definitely yes. So, and if I combine some of these together, that would be a much better feature set for me to discriminate. Agreed? Yes? So what do we learn from this part? Actually, machine learning or any artificial intelligence is nothing but mathematical operations. Even if you give it age and height, it will give you the output. Because engineers are very smart. They have designed the machine to give you an output. Now it's up to you to decide whether it is trustworthy or no. Right? So the take-home message is relevant features are absolutely essential for machine learning algorithm to execute the process and get a good outcome. If you put in trash as a feature, you will get trash as an output. But you will get an output. And if you don't know what is the output as a clinician, engineer will force, sir, this is the best output. But you have to tell, no, this is not a good feature. I want to get trained by something which is relevant to me as a clinician. Right? So this is the take-home message. Now let me pose you some example. Grade of breast cancer versus tumor size. Are you able to see 3 clusters? Easy. Human can perceive it. Right? I don't need machine for that. नहीं sir, 100 data होंगे तो हो जाएगा, आप 2 तो सही. Machine learning will do it better. I don't need it, but I need something for this. Can I see Parkinson's disease progressive stage versus voice and gait pattern in patient? Are you able to see some discrimination here as a human? Yes, no? Feedback, please. It helps me also improve. No, but machine can. Right? There is some overlap. Probably it's not as good as this. But yes, it is better than what we can do. Machine can. It can learn that there is some information in it that can improve me in clinical decision making. Now, number of days of fever versus body temperature. Anything. Anything you can actually imagine or think of, there could be anything. Machine can. Right? And that is the output you will get. There are 3 clusters, days of fever versus temperature. But is it giving you any information as a clinician? Whether it is relevant to you? Is there any hypothesis that goes that, okay, bacteria, viral, chronic fever, pyrexia of a non-origin, anything that can be sufficiently figured out that where it belongs? I don't know. But as an engineer, we can give you this. Now, whether it is clinically useful or no, that is your judgment. right? So don't blindly trust what is given to you, but a clinician input is always mandatory. Reinforcement learning. The algorithm system is based on reward and punishment. And objective is to maximize the reward and minimize the punishment. And that's how the repeated learning comes. And there are examples in literature for cirrhosis versus hepatocellular carcinoma, Parkinson's disease, grading for gait and voice, radiological diagnosis, prognostic monitoring for chemotherapy. And these are some of the examples I have taken from literature. Now, I will not spend time on this, but I really want to show you this, a hardcore device with machine learning.
You may think you have little in common with this robot, but it is controlled by living brain cells much like those inside your own head. Researchers grew a collection of around 300,000 rat brain cells in the lab. The cells quickly grow collections to each other and start communicating using electrical pulses. When the clump of cells was connected to electrodes, some parts of it were found to respond predictably to signals from the robot's ultrasound distance sensor. The output from these cells can be used to control the robot's motor circuits, preventing it from bumping into obstacles. Because the brain cannot leave the lab, it communicates with the robot wirelessly. The team plan to use their robot's brain power for new patterns of behavior in future. They hope there are enough similarities to teach us about unknown aspects of our own brain activity.
So what they did actually was they had a petri dish on the top and put 300 million neurons from rat and had multiple electrodes which were connected to IR cameras and motors in the robot. And with the reinforcement learning, the neurons were designed such that they learn that I don't have to hit the wall. And with repeated connections from different IR cameras as sensors and different motors and actuators, it tried to learn that I don't have to hit the wall. And this is the application of machine as a hard head device in your hand and learning, not a computer as an algorithm. Now, to summarize, machine learning, as we understood, has supervised, unsupervised, and reinforcement learning. These are the broad categories, but there are multiple algorithms which are under each category. Every algorithm has its own pros and cons. I'm not here to tell you each about algorithm, but there are multiple things which will perform differently, and we'll give you some examples. I just want to share some of the examples where this is a chest X-ray and a deep learning network has been built where it can actually tell you with a high probability. It is called heat map, that at which point the machine thinks that there is some abnormality. And you can correlate that this heat map fall over here. And it can say, okay, with 85% probability based on my training data, I can say it is pneumonia. It can say, okay, it is viral pneumonia, it is a fungal pneumonia, it is a cancer, it is tuberculosis. With some probability, it can reduce the probability and give you differential diagnosis as well. The more data you give, the better probable the machines can perform. Sometimes there are small subtle fractures as a tedious task sitting on the dark room from morning to night, the radiologist might miss. But yes, if there are tools which can actually screen the data for a easy reading and say, okay, look at this point, I feel there is something here. The machine is not designed to give a diagnosis, but give a hint. Just see here. If you agree there is something, mark it. If not, let it be. But it can prioritize or triage. So machine learning has application in healthcare across fields. Drugs, imaging, EMR record system, surgical training, prognostic monitoring, smart diagnostics, a lot of applications. Now, just 10 minutes, I would like to share some of our experience in our lab with machine learning. So there is a prostate cancer workflow clinically, and at some point, there is a radiological scoring that comes with pyretes, and the radiologist has to mark whether there is low or high grade pyretes. But before the lesion is marked, the segmentation of prostate has to be done. There is a high probability of prostate cancer in the peripheral zone, and with transition zone, there is a possibility of benign prostatic hypertrophy. So first the prostate has to be marked as a segmentation, and then machine learning that we have applied using some algorithms, and we have said that, okay, with 93% accuracy, we can score up to low-grade cancers, and with like 80 to 75% probability, we can grade the high-grade cancer just by reading images. So bone cancer is something we have been working on with team at AIIMS for 8 years now. So the typical response evaluation other than radiologist racist criteria is definitely pathology. The clinicians believe what they see as there is a chemotherapy response, yes or no. Now this is possible only if I take the biopsy or if I take the surgical resection and send some part of it for the histopath. Now can I do that non-invasively and judge that this patient is going to be chemotherapy positive, responsive or negative? If not, then probably I would like to change the treatment. That is the hypothesis. So we have designed a complex algorithm. I don't want you to pay attention to everything, but just a bit here. So with different sequences of MRI, which are sensitive to different set of information in the cancer, if we can figure out what is the active tumor percentage, what is the level of necrosis, and what is the level of edema in that tumor, I can tell if the necrosis is increasing in the tumor, the chemotherapy has done the job. Agreed? Yes, no? Yes? Yeah. So we have done that. So I'll show you 2 examples and then the cohort results. This is the baseline patient, and it was a 12-year-old female with osteosarcoma on the right femur. In the baseline, it showed that she has a necrosis tumor of 4% and edema of 40%. And after 3 cycles of chemotherapy, data was taken, and it shows that the necrosis increased to 91% and edema is 13%. And when post-surgery, the resection was done and the histopathology report came, it was 90%. So it closely matched what we actually predicted non-invasively before the resection was done. Another case where it was a 16-year male with right tibial osteosarcoma. The baseline necrosis was 1% and edema 27%. After 3 cycles of chemotherapy, the necrosis came to 8% and edema increased to 69%. And histopath shows the edema, the necrosis is approximately 5%, so close range. So when we put a cohort analysis and we divided the 2 groups into the response positive and response negative based on histopath, we saw that baseline was equal for both the clusters, and the algorithmic MRI evaluation versus histopath were close match. We were able to discriminate between response group and non-response group based on the necrosis that has evolved non-invasively before the resection was done. So it has possibly good applications in healthcare. Now, one last example is on contrast-enhanced CT. So we want to have some data which can be discriminated to healthy and diseased, and then cirrhosis and hepatocellular carcinoma. So we first has to segment the liver from the abdomen, and we designed an algorithm here which is called modified region growing, and there is a state-of-the-art deep learning algorithm which is available publicly for liver segmentation on CT. And this is how the 3D model of the CT look. Even the 2D slice you will say looks similar, and 3D is like this. But when it goes to pathology and cirrhosis, you can see that the deep learning has a poor performance in actually effectively taking out the full liver, but modified region growing and ground truth are quite mapping to each other. And that is what we see in the full cohort analysis. The output is conventional approach can be sometimes better than deep learning, whereas the machine learning community is moving completely towards deep learning as complex solution, but you have to probably start from simple solution and they can do better. So don't go with pre-notions that this is what I want to do, whatever would be the output I will accept. And our efficacy in classifying into healthy versus disease was 92, and then cirrhosis versus hepatocellular was in the range of 85%. Now, none of the machine learning talks is complete if I don't tell you that it has its own problems. So machine learning, we have a lot of medical data. We do feature extraction for supervised machine learning. And then there is a black box, which very few of us understand, and it gives you an output. Right? It works perfectly fine for healthcare. But the problem is you need lot of data for training. If I want to mark the lesion, I have to sit with the radiologist and ask him, please mark 500 data for me. He will say, I don't have enough time. But that is the limitation. I need it to be marked. Then only I can train the machine. Otherwise, I can't. There's a lot of features that need to be extracted. And manual feature crafting can be a tedious exercise sometime. We might not be able to do that. But that is the bottleneck. If we want it to deliver good, you have to do that exercise. The other aspect is deep learning, which is completely a black box. And you give the data, it do something and give you an output. No features, no training. But it does not require manual feature because you don't know what to do. It learns some higher order or hyperparameter features in complex architecture to deliver the output, but it works. It is based on logic. The decision is made that cannot be understood. So as a clinician, if you ask me why this patient is positive, I don't have any answer because it is not based on any feature which I can attribute it to. But there is something it has learned. So it is not explainable. I can't explain it or reason it why it is the way it is. And large amount of data is required for do deep learning because there is a lot of heterogeneity in population, disease progression, and everything need to be taught to the machine so that it gives good outcome. So I'll give you some example here. This is I have taken from the news and it's post from 4th Feb 2020. So what happened was the man decided to take a cart, put 99 mobile phones in the cart and decided to walk on the street with GPS on. So what will happen to the Google map? It showed there is a clog on the road and everybody started diverting away from the road. Oh, there is a clog there. But what was reality was this man was walking on the street alone. Now this is Google machine learning. It is not old. It is 2020. And then Google realized we have this bug. A human intelligence can actually fail my machine intelligence. And then they debug it and see that, okay, there has to be some other feature. So this is what happens, but you know the reality and you can correct it. In healthcare, the problem is sometimes you might not know the reality. Machine throws you something and you have to trust it. It's on your own judgment. So be cautious about it. That is what I only want to say. But it's a great area. After 2010, there is a spike in the increase in the literature on artificial intelligence, machine learning. And if I put machine learning, healthcare or cancer, I got only 11,000 hits. So there is a good area to invest your career in. So machine learning or artificial intelligence is a very complex area. But with my limited knowledge and shortage of time, I'm giving you exposure of only artificial intelligence, machine learning, and deep learning. There is another big ocean of artificial intelligence, cognitive, speech, computer vision, NLP, which has tremendous scope in healthcare, but it might need a full 1 or 2 day workshop to get exposed to everything. So the take-home messages are the rule-based systems are very useful in healthcare as they inherit the expert knowledge that has been gathered over centuries. And it doesn't need large data because it's based on rules. Low data or like 60, 70, 20 patients are good enough to test my approach because it's based on completely rules. Relevant features are absolutely essential for any machine learning algorithm to execute a good performance outcome. Trash goes, trash comes out. Good feature goes, something relevant will come out. Appropriate selection of algorithm is relevant for clinical problem. All algorithm might not work for all the problems. Each one has its own pros and cons. It's a hit and trial method for all of us. AI can learn feature in data that humans cannot sometimes perceive, like deep learning. So it has a huge scope in doing repeated tasks, endless time without getting tired, giving you the same efficacy in the morning and late night, continuously run, which humans cannot. So there is a scope for these to be put in the clinical workflow to optimize the resources. Sometimes you might be in a resource limiting setting and you want to prioritize where to put my resources. And the AI have scope in helping you optimize that. AI is not a solution. And it's definitely not solution for every damn problem you have in your life. It's a tool. It's just a tool like a screwdriver. Right? But it's a very powerful tool and it's a very powerful technology. You have to use it optimally for healthcare translation. AI is a double-edged sword. The output might not make any sense, but machine doesn't have common sense anyway. Right? It's for you to do judgment like how it performs. Needs a large amount of labeling data for training and testing. The ethical issues on AI and ML have just evolved, like who is responsible if I translate my AI algorithm in clinical decision making? Is the doctor accountable? Is patient accountable who give a consent? Is the person who designed the algorithm accountable? Or is regulatory compliance, CDSCO probably, I put a question mark, is responsible? Now there are many AI algorithms which currently run on your system a lot, which are with devices, right? If a device is running an AI algorithm, CDSCO has evaluated both the device as well as algorithm for its clinical translation. But there are many AI algorithms which are not run on any commercial device. As a person, I deploy it to you. You can use it as a third party. So there is no regulatory compliance yet. It's an area of discussion which all the stakeholders, including you and us, has to debate and probably come out as a solution. There is a geographical ethnicity, the population variability across globe. Now, we could have done studies in US, but whether that AI model trained on US population is applicable to me in India. We still follow the blood benchmarks even, which are inherited from Britishers, right? We don't even have good statistical analysis for some of our own markers. AI is still far. Whether it can be translated across globe, we have to question. So the future lies with when you merge human intelligence with artificial intelligence. The people who are empowered with artificial intelligence tool will optimize their time resources in a better way, but don't see that artificial intelligence is going to replace any of the clinical workflow. It's not new. It's the age-old technology which has been popularized in last decade. Flights with automated mode has been there for a while, but it never replaced the pilot, right? So, but you can get empowered with this. So I am just the face of what we do at IIT and AIIMS. There is a big team at IIT. There is a big team at AIIMS who has given me tremendous support what I present today. And I would like to acknowledge and thank them. I also work in area of neurorehabilitation. So I have received tremendous support both professionally and at personal level from faculty and staff at AIIMS. So this is the platform I would like to acknowledge and thank everybody in my journey. And if I just grab 2 minutes, I would like to end with a video. So just see this for 50 seconds.
Clearly, somebody in this room murdered Lord Smythe, who, at precisely 3:34 this afternoon, was brutally bludgeoned to death with a blunt instrument. I want each of you to tell me your whereabouts at precisely the time that this dastardly deed took place.
I was polishing the brass in the master bedroom.
I was buttering his lordship's scones below stairs, sir.
I was planting my petunias in the potting shed.
Constable, arrest Lady Smythe.
But how did you know?
Madam, as any horticulturist will tell you, one does not plant petunias until May is out. Take her away.
Sorry, madam.
It's just a matter of observation. The real question is, how observant were you?
Why did I show you this video? Did you notice anything in the video in 50 seconds? Very quickly. We don't have time. Yeah, so something with the clock. So some people have noticed color, right? So let me show you this video. Just try to see it again. Like what has changed during that 50 seconds, including the black coat of this detective. Try to see the changes that has happened and how many you can pick.
Clearly, somebody in this room murdered Lord Smythe, who, at precisely 3:34 this afternoon, was brutally bludgeoned to death with a blunt instrument. I want each of you to tell me your whereabouts at precisely the time that this dastardly deed took place.
I was polishing the brass in the master bedroom.
I was buttering his lordship's scones below stairs, sir.
I was planting my petunias in the potting shed.
Constable, arrest Lady Smythe.
But how did you know?
Madam, as any horticulturist will tell you, one does not plant petunias until May is out. Take her away.
Sorry, madam.
It's just a matter of observation. The real question is, how observant were you?
Right, so now let me show you it from a different angle. So there were total 21 changes that happened in 50 seconds, and we'll change the camera view.
Clearly, somebody in this room murdered Lord Smythe, who, at precisely 3:34 this afternoon, was brutally bludgeoned to death with a blunt instrument. I want each of you to tell me your whereabouts at precisely the time that this dastardly deed took place.
I was polishing the brass in the master bedroom.
I was buttering his lordship's scones below stairs, sir.
I was planting my petunias in the potting shed.
Constable, arrest Lady Smythe.
So why did I spend 3 minutes in showing you this video? It has a message. When I changed the angle of the video, actually I gave you a different data set. And your understanding of the content change. So like in AI, you need more data always to understand the full picture. More data you see, the better you get trained to judge the unseen, which might hit to you in the clinical scenario. Looking the AI through both medical as well as engineering lens. It's never that somebody has developed and you can deploy it. You have to take a judgment. It might have different viewpoints for all of us. And as I can say that many of you have witnessed 1 or 2 points, others must have seen two points, but there are total 21 differences. Treat each one of you were like one of the AI algorithm. For the same problem, each algorithm throws out some solution. And each algorithm will have different limitations. Now that's why I personally prefer in a sandwich approach. As I showed you in the liver, we separated healthy versus diseased and then split disease into cirrhosis versus hepatocellular. I could have done healthy versus cirrhosis versus hepatocellular as a 3-class problem. But then it's a complex problem. But when you split and go in a stepwise manner with different approaches, your accuracy is better. So see AI as a tool. The more data is required, and you have to open your eyes to that, because that is the way forward to go. With that, I would like to thank everybody present here and giving me the opportunity to share my thoughts.
I'm sure it was very interesting talk from Dr. Amit Mandiratta. So there may be many questions, I hope. So there, yeah.
I disconnected. Just
Hello.
Hello, sir.
Very nice, excellent.
Thank you, sir.
But I think the crux is the data.
Right, sir.
And that is a worry. Are we generating good data and making good data sets? And that focus is very much required.
Essentially, sir, because the large variability in your data would confuse the algorithm. That's why we plan to conduct studies with very focused data and then diverge into the interpatient variability, healthy variability, or complex variability. The data is the crux, rightly said.
We can take one more question because we are running short of
Yeah.
time. Please.
We are deeply pleased to listen to all this. But my question is that for medical science, the Bayesian learning, I think that has also a great role apart from what you have described. And also for simple procedures, we can easily make macros.
Correct, ma'am. Bayesian, I'm a big fan of Bayesian algorithm. So I'll just for the benefit of audience, I'll tell, right, when I showed you the cluster, I spoke about the centroid and mean, right? So I take the data as a variability of say mean and standard deviation. It's a one mathematical model of normal distribution to put my data. But I feed my algorithm with that only 2 parameters. Wherever, as if you see the Gaussian has a long tail going toward the end, there is a 1%, 0.1% probability it can be an extreme data that might hit your algorithm, right? But you don't want to cut that and say this is healthy. With 0.11%, it could be abnormal. So Bayesian is that approach who take into account the probability for attributing any data to any cluster rather than just mean and 95% standard deviation. Yes, there is a good scope, and I am a big fan of Bayesian algorithm in healthcare. We do work on that.
Just one question. Education 4.0 said that the technique will take over human brain. And Education 5.0 say human brain cannot be overtaken by...
Sir, let me put it in a very philosophical, it's a philosophical question. So I'll tell everybody that can AI beat human? Because you have been trained as an AI which was marked by a clinician. And that is the benchmark. Can you beat the benchmark? If you beat the benchmark and say that this is a patient with XYZ and clinician has labeled is no, it is PEQX. Whether you will believe clinician or the AI. So the AI has not reached the stage where it can actually beat the benchmark. But yes, if it beats the benchmark, it comes as a feedback to clinician to relook whether I want to evaluate that with a different mindset? Yes. But currently we don't expect it will ever beat that.
It's like God versus human.
Thank you. Thank you very
God
much,
versus
Dr.
who is
Amit.
first.
I think we'd like to close this session here. We are running
I think
behind time.
I've taken more time than allotted, but
Actually, really, let me share that this quest of data brought us to AIIMS 5 years ago when we have IIT AIIMS joint projects, and we are running currently 65 projects together with AIIMS doctors and IIT engineers, and 10% of that are in AIML. I think we'll have more to come in years.
So thank you so much for this remarkable talk about AI, machine learning, and deep learning. Needless to say that AI can be a game changer in the Indian healthcare context. And now I request both the chairpersons to kindly come forward to felicitate the speaker. Thank you, sirs. I request Professor Puneet Kaur ma'am to kindly felicitate the chairpersons with a token of our appreciation. Thank you, ma'am. Now I hand over the stage to Dr. Souvik. Thank you.
Thank you, Dr. Kamal. Now we proceed to the next scientific session, Innovative Research Best Papers. The first platform presentation is by resident doctors. Each presentation will be of 7 minutes, followed by 2 minutes for discussion. For this session, I would like to welcome the honorable chairpersons onto the dais, Professor A.B. Day and Professor Neena Khanna. Professor A.B. Day was formerly Dean Research and Professor and Head, Department of Geriatric Medicine, AIIMS, New Delhi. Professor Neena Khanna is Professor of Dermatology and Venereology at AIIMS, New Delhi. The first paper will be presented by Shravan Kumar Rawat, Resident Doctor in Department of Medicine.
So, good morning, everyone. I'm Dr. Shravan Rawat, Junior Resident in Department of Medicine.
Good morning. It's always good to come back to my MICA. I'd like to thank the research section, Dr. Raman Chaudhary and Vinita Huja to invite me for this. And I'm sure Dr. Day also echoes the fact that he's come. It's Ghar Wapsi for him also.
Now we'll have the presentation. 7 minutes. 7 minutes means 7 minutes. Okay.
So, my study topic is prediction of ventilator-associated pneumonia using a novel artificial intelligence-based decision support system. So, it was a collaborative effort of two institutes, AIIMS and IIT Delhi, under the guidance of Dr. Animesh Resar and Dr. Colin Paul, and these are the list of our co-guides. So, what is ventilator-associated pneumonia? It is a common complication that we see in a patient on mechanical ventilator for more than 48 hours. And it is a type of nosocomial infection which can lead to significant morbidity and mortality. The existing diagnostic modalities that we have is a clinical pulmonary infection scores, which includes several clinical and laboratory parameters like temperature, oxygenation in the form of PF ratio, leukocyte count, tracheal secretion, radiographs, and ET aspirate culture and sensitivity. we give scores from 0 to 2, and once the score is more than 6, we label it as a ventilator-associated pneumonia. So, as the CPIs depends on number of parameters like culture and sensitivity, whose reports comes after several days, which might be colonized, the evidence of tracheal secretion, labeling as purulent or non-purulent, which might be subjective, and the radiographic changes, which may have inter-observer variations, as we can see here in these two images, which is difficult to differentiate between collapse or consolidation. So, the diagnosis becomes difficult at bedside based on this parameter. Although the existing gold standard for the diagnosis is lung biopsies, it is an invasive procedure and difficult to perform. So we came up with this study which can accurately predict the diagnosis of ventilator-associated pneumonia. Our hypothesis was to integrate the AI-based decision support system with that of clinical, radiological, and microbiological data for early and reliable diagnosis of ventilator-associated pneumonia. Prime objective was to determine the diagnostic accuracy of AI in detecting ventilator-associated pneumonia, and also to compare the AI reading with that of conventional radiological reporting. And the proposed outcome was to calculate the sensitivity, specificity of AI-based diagnosis. It was an prospective observational study done in medical ward and ICU in collaboration with medical IT lab over a period of 2 years, and the sample size calculated was around 49 VAP patients. So, any adult patient on mechanical ventilator for more than 48 hours was included in our study. Pregnant lady or those who have denied consent was excluded. Also, the patient without chest radiograph done within 24 hours of suspicion of VAP or previously treated for VAP in the current intubation was excluded. This is the baseline model, that is the CheckSNet model, which uses input as a chest images and gives the probability of diagnosis of pneumonia. On the basis of this, we have modified our system to develop a newer algorithm, which is a joint deep learning model based on image data provided as a chest X-ray and tabular data in the form of clinical and microbiological data to develop a newer system. After the enrollment of patient, the several clinical parameters as mentioned here, like ABG, leukocyte count, bedside X-rays on alternate day was done, and CPI scoring was calculated. And the AI algorithm was developed, trained, and validated. After coming to the result part, we have screened 102 patients on mechanical ventilator, among which 65 patients was included. Total of 300 plus images was used for the analysis of artificial intelligence-based system. The total label data was divided into the training set as at 80% and validation set as 20%. These are the baseline characteristics of the patient. Among the microbiological culture data, most of it was maximum for Acinetobacter baumannii. This is how the AI system looks like. Here we put the image as a chest X-ray, and it gives out the results of various probable pathologies. But we are concerned here in the diagnosis of pneumonia. It gives the diagnostic probabilities and the prediction values as 0 and 1. 0 means VAP, 0 means no VAP, and 1 means VAP. Coming to the performance of AI VAP system, we have calculated the sensitivity and specificity based on the radiograph. It was 69 and 76% respectively, and the accuracy was 83%. But the joint model showed the accuracy of around 89%. Also, we have given the chest X-ray data blindedly to the radiologist without any clinical and microbiological data, and compared with that of CPIs, and calculated the sensitivity and specificity, which was 25 and 90% respectively. The third thing we have done was comparing the physician diagnosis with that of CPI system, and the sensitivity and specificity calculated was around 23 and 93%. Coming to the summary of results, the overall accuracy was compared. AI VAP system has much higher accuracy, that is 89% in compared to 70% of based on radiology and 66% based on physician diagnosis. So in the future, we can use AI to compare with that of gold standard diagnosis of ventilator-associated pneumonia. Also, we can use this AI VAP system to assess the response of the therapy which we give in case of ventilator-associated pneumonia. Concluding our study, as the sensitivity was quite higher and also the accuracy was higher in compared to the diagnosis based on the radiology and the physician, it can be used as a screening tool for the diagnosis. It can also reliably predict the CPIS values. So that's all from my presentation. Thank you.
Missed something, but you know, you say there's a physician-based diagnosis, okay, and to that you have and is more sensitive. The point is that in that case, what is your, I mean, I know these very difficult to have a gold standard of this sort, but how would you guess, what is your baseline on which you have seen this is actually, what your AI is getting is actually a physician, is your VAP?
Sir, in this case, we have seen either the physician has changed the antibiotics based on the diagnosis of VAP or not. On that parameter, we have compared with that of CPIS system with all the parameters.
Essentially, what you are saying is the therapeutic response is your gold standard. Yes. Is that, am I getting you right?
Yes, sir.
Okay.
You talked about lung biopsy at some point of time. But I thought HAP is quite a common diagnosis in any ICU. You are trying to make things rather complicated. Anyway, thank you. Thank you for the presentation. It's all right.
Thank you, sir.
Well done.
Our next paper presentation is by Anjusha Ranjit, Junior Resident, Center for Community Medicine.
Anjusha, your time starts now.
Should iron folic acid supplementation be replaced with multiple micronutrient supplementation for anemia prophylaxis in pregnancy? Good morning. I'm Dr. Anjusha Ranjit, Junior Resident from Center for Community Medicine, and I'm here to present the evidence from a triple-blinded randomized control trial. The Government of India has been supplementing iron folic acid in pregnancy since the last 5 decades. But the prevalence of anemia among pregnant women have more or less remained unchanged over the years. The reason for this could be the coexistence of other micronutrient deficiencies. MMS in pregnancy in recent years have gained popularity because evidences have shown a reduction in the prevalence of low birth weight babies associated with its use. Hence, we conducted this study to compare the effectiveness of MMS versus IFA for anemia prophylaxis among non-anemic pregnant women. We also compared the changes in serum levels of vitamin B12, folate levels, compliance and side effects between the two groups. For this, we conducted a community-based, triple-blinded randomized control trial in 2 PHCs in Haryana. We enrolled 200 pregnant women, more than or equal to 12 weeks of gestation, and having a hemoglobin of 11 to 14 grams per deciliter. The intervention arm received MMS tablets in the form of Unimab preparation containing 30 milligram of elemental iron, and the control arm received IFA supplements containing 60 milligram of elemental iron and 500 micrograms of folic acid as is recommended by the Anemia Mukt Bharat. The randomization sequence was generated by a person who was unrelated to the study by block randomization using variable block sizes. Allocation concealment was achieved by sequentially numbered sealed identical containers, and triple blinding was achieved by use of similar looking tablets and similarly labeled bottles, as we can see here. Telephonic follow-up was conducted every 4 weeks. A compliance assessment chart as seen here was provided to the participants at the time of enrollment. Anemia, iron deficiency, vitamin B12 and folate deficiencies were defined as per the WHO guidelines for the purpose of the study, and compliance was defined as the proportion of the total supplements that was consumed by the participants. And any participant who had consumed more than 80% of the supplements was considered to have good compliance. As we can see here in the consort flow diagram, 101 participants were randomized to receive MMS supplements and 99 participants to receive IFA supplements. At end line, we had blood samples for 94 participants in the MMS group and 91 in the IFA group. The two groups were comparable with respect to all baseline characteristics. Only 7.5% participants were lost to follow-up during the course of the study, and those lost to follow-up were comparable with those retained in the study with respect to most baseline characteristics. A modified intention to treat analysis, including all the participants for whom a blood sample was available at end line, was done, and the differences in hemoglobin between the two groups was found to be 0.8 grams per deciliter favoring the MMS group, and this was significantly higher in the MMS group. When adjusted for variables that were found significant in bivariate analysis, the differences in hemoglobin continued to remain significant at 0.72 grams per deciliter favoring the MMS group. The changes in serum folate levels, serum vitamin B12, as well as ferritin levels were significantly different between the groups, and all three of them favored the MMS group. As we can see here in the first part of the bar diagram here, the anemia prevalence increased in both the groups from baseline to end line, but the increase was much lower in the MMS group as compared to the IFA group. And also, the prevalence of other deficiencies, that is iron, vitamin B12 and folate deficiencies, decreased with MMS supplementation, whereas it increased with IFA supplementation. Of importance to note here is that around 60 to 65% participants in both the groups were vitamin B12 deficient at baseline. And this prevalence of deficiency increased to around 83% in the IFA arm that received no vitamin B12 supplementation during pregnancy. The compliance to the two supplements were comparable between the two groups, and it was high. Only 11 participants in both the groups had compliance less than 50%. And the adverse events profile were also comparable between the groups, but only 33% participants in the MMS group as compared to 46% in the IFA group experienced some form of adverse events. But this difference failed to reach significance. Both solicited as well as unsolicited adverse events were comparable between the groups, and none of the participants experienced any serious adverse events. We can see here that the changes in the levels of hemoglobin as well as the changes in the prevalence of iron deficiency favored the MMS group. The reasons for this could be that the other micronutrients in MMS may have enhanced iron absorption from MMS, and also these hematinic micronutrients may have itself led to a better hemoglobin response. The higher iron deficiency at baseline in the MMS group may have led to a higher iron absorption from MMS, and also there was slightly higher compliance in the MMS group, though this was not significant. A recent Cochrane review by Keats et al. Had shown that maternal anemia in the third trimester were comparable between the groups receiving MMS and iron with or without folic acid. The reason for this lack of difference may be due to that the earlier studies included anemic pregnant women, and 30 milligram iron may have been insufficient in anemics. We also saw that the changes in serum vitamin B12 levels as well as serum folate levels favored the MMS group. All the findings continued to hold in a per protocol analysis, including only participants with more than 80% compliance. But the prevalence of anemia increased in both the groups from baseline to end line. This may point towards the need for a higher dose of iron. Compliance was comparable, and the proportion of participants experiencing adverse events was slightly lower with MMS than with IFA, though not significant. Hence, we concluded that MMS containing 30 milligram of iron is not inferior to IFA containing 60 milligram iron for anemia prophylaxis in pregnancy and was at the verge of superiority. This provides early evidence on the beneficial effects of MMS compared to IFA in pregnancy on maternal anemia and micronutrient status, but there may be a need to consider higher iron doses in MMS for tackling the high anemia burden. This is the first study in India to assess the effect of MMS on maternal anemia and micronutrient status. Following this, a technical consultative meeting was hosted by WHO and BMGF to discuss the ideal dose of iron in MMS supplements. And for this, a large-scale multicentric trial is being planned by BMGF. Thank you.
Well done, but you didn't restrict to the time. What's the cost difference between the two?
There is a recent cost effectiveness analysis that was done. Based on that, the cost of 180 tablets, that is for one pregnant woman per pregnancy, comes to around $3.27 for MMS. Whereas
Can you speak in rupees? That's what is operationally needed, though.
Yes, ma'am.
A dollar is 80 rupees, 80 plus
240.
82 rupees. So.
Almost 240 to 250.
Okay, any questions from the house?
If there are none, I'll just ask a question. The MMS in pregnancy is already in practice now in many parts of the world.
Yes,
so
sir.
what's the novelty here in your study?
So this is the first study in India that we have done assessing MMS.
But after all, biologically, all human beings are same. India or China or Africa doesn't make much of a difference. Okay, thank you. Thanks. Just a minute. There's a question.
I have one question. Like, I am a faculty in College of Nursing. When we are giving this health education to pregnant ladies, we are telling them, like, for better absorption of iron, we need to have citrus fruit juice or anything. So was that included also? Because absorption of iron needs vitamin C.
Part of this presentation. It's a randomized control trial. Obviously, that's not being covered. Thank you.
Thank you, ma'am.
The next presentation will be delivered by Yallam Raju Saikosthap, Junior Resident, Department of Surgery.
A very good morning to all the respected judges, faculties, dignitaries, and my dear friends. At the outset, I would like to thank the research department for providing me this opportunity to present my study on 1-anastomosis gastric bypass versus Roux-en-Y gastric bypass for the remission of type 2 diabetes mellitus in obese subjects, a randomized controlled trial. Bariatric surgery has been the mainstay of treatment for remission of comorbidities and sustained weight loss in morbidly obese patients. Roux-en-Y gastric bypass has been the gold standard for the past 4 decades. But with the advent of OAGB, a novel procedure with technical simplicity but similar efficacy, things took a turn. We hypothesized that OAGB is non-inferior to RYGB in terms of impact of type 2 diabetes mellitus and other parameters like weight loss. So our primary objective was to compare the remission rates between both the groups as per AS/MBS criteria, a fasting plasma glucose of less than 100 mg per deciliter, in addition to a HbA1c less than 6% without the use of any OHAs or insulin. The secondary objectives included to compare the improvement in glycemic status in those who didn't achieve remission, weight loss, duration of either surgery, post-operative stay, including the complications. It was an open-label, randomized, non-inferiority trial commenced after an institute ethics approval at our center, AIIMS, New Delhi, with a calculated sample size of around 42 in each arm. The patients were recruited after meeting the following criteria. The inclusion criteria included patients above BMI of 30 kg per meter square with concomitant type 2 diabetes mellitus, weight loss, and type 2 diabetes mellitus not properly controlled by non-surgical measures, patients who had comprehension and commitment to follow up long-term and who gave an informed consent. Excluded patients were those who didn't consent or those who had a contraindication for GA. So this was the procedures in which in RYGB, we basically have more of a
restrictive pattern in which a small pouch is created near the GE junction of around 10 to 15 ml, and we create a BP limb of 70 centimeters and an elementary limb of 130 centimeters. Whereas OAGB has a single anastomosis in which a long tubular pouch is created. After the tubular pouch, we just take a loop around 200 centimeters and anastomose it. The study population was enrolled between 2017 and 21, 25 in the OAGB and 25 in the RYGB arm. And the RYGB arm had almost complete follow-up, whereas the OAGB arm had 3 patients lost due to COVID pandemic and 1 mortality due to COVID pneumonia. The baseline demographics were similar between both the groups, especially the BMI and the excess weight, which were comparable. Coming to the operative time, OAGB had significantly lower operative time by 40 minutes, whereas the post-op stay, complication rates and readmission rates were similar between both the groups. Coming to the weight loss, both the groups had a similar highest weight loss achieved in the first 2 years after surgery, as you can see in the BMI as well. And then there was a slight weight regain, but again, it was significantly lower as compared to where they started off. Coming to the baseline glycemic parameters, the distribution was comparable, though RYGB had a slightly higher HbA1c and mean duration. OAGB had a higher mean insulin requirement per day. So the remission rates were similar at all the 4 groups, but the highest achieved around 86% for both the groups. Even at 4 years, it was around 72% with a very good sustained remission rate, and the relapse rates were also comparable. The HbA1c also showed a similar trend with the least value achieved at around 1 year following surgery, after which there was a slight increase, but again, compared to a baseline, there was a significant fall in both the groups. And the similar trend followed in the fasting blood sugar as well, with the lowest rate in 1 year, and the 4 years rate was also comparatively low as compared to the baseline. This was the OHA and the insulin requirement, and as you can see, at 1 year, only 1 patient in the RYGB group still required all through the 4 years. The patient was a chronic diabetic over 30 years with an insulin requirement of 50, but she came down to around 10 units insulin requirement. And all patients showed improvement, none of them reverting to their baseline status, and most of them requiring only a single OHA, even at 4 years. So this was the relapse rate, 1 patient relapsing in both the groups at all 3 years. And it was comparable. The remission of other comorbidities, especially hypertension and OSA, was also significantly higher, especially in the first 2 years. Then there was relapse, which was associated with a weight regain, but again, it was comparable between both the groups at all the timelines. Coming to the discussion, this is the first RCT that has been conducted in literature on the impact of type 2 diabetes mellitus between both the procedures. However, Omega trial, which is the gold standard trial that has been conducted comparing these two, reported a higher remission rate of 60% in OAGB at 1 year following RYGB. But this could be explained by the shorter BP limb length they expected, 50 centimeters as compared to 70 centimeters in our study. The other study was by Almaki et al. It was the retrospective and the largest available study in comparing both these procedures. They showed a significant higher remission with OAGB as compared to RYGB at 1 year and 5 years as well. But their baseline population had a higher mean RYGB requirement and higher mean insulin requirement in the RYGB group, which was found to be significantly high. Hence, that could explain why they found a significant result. However, in our study, at all the timelines, we found that both of them were having similar rates of remission. Coming to the strengths of our study, this is the first study in literature comparing these two procedures in terms of impact of type 2 diabetes mellitus, as OAGB had been invented only in 2001. A single surgeon with vast experience of over 1,000 procedures performed all these surgeries. A detailed analysis of weight loss, remission of comorbidities, type 2 diabetes mellitus, and other were taken up with a good follow-up rate with only 3 patients lost in the OAGB arm. The study also reflected the outcomes and issues faced by the pediatric population, especially in relation to weight regain, relapse of comorbidities, and nutritional complications during the pandemic when there were limited resources. The limitations were the desired sample size could not be reached because, especially of the COVID-19 pandemic. The COVID-19 pandemic also had its implications in terms of the fact that the results, the weight regain and relapse of comorbidities affected both the groups. Maybe a superiority trial with a better sample size or a similar RCT achieving the desired can further substantiate the evidence if OAGB is more effective. To conclude my presentation, OAGB is non-inferior to Roux-en-Y gastric bypass in terms of remission of type 2 diabetes mellitus and weight loss with a similar complication rate.
Being a technically easier procedure with a shorter operative time and a shorter learning curve, it may be a preferred option as compared to OR-YGB in morbidly obese patients with type 2 diabetes mellitus. Thank you.
Thanks, Sai Kaustubh. So you are advising OAGB?
Yes, sir.
Okay. And you could not reach the sample size because of
the pandemic, sir.
So statistically or scientifically, how does it affect the result, in your opinion?
Sir, basically, when the sample size is slightly lower, there are chances of outliers, especially in both the groups, to affect the results, sir. In my case, there were around 25 in both the groups. And the people who didn't achieve remission in both the groups were all people who are having long-standing diabetes and this thing, which basically represents one end of the population. Okay. So it didn't cover the entire population.
Okay. Any question, comments, questions from the audience? Yeah. Yeah. You raise the hand. ऐसे ही पूछ लो. सामने आ जाओ, पूछो जोर से. Basically, how you defined remission of diabetes?
See, remission of diabetes, this was taken from the American Endocrine Association and
that's a standard process of defining.
Yes.
You agree, disagree? You have got any point with that?
1 year. And these patients, all these patients who have went into remission, immediately in the post-operative period, they don't require insulin or any support. Usually that happens.
Okay. Any other question? None. Okay, thank you.
Thank you, sir.
Thank you.
Thank you, well done.
Thank you, sir and madam.
Take a minute. I must thank Dr. Rama Chaudhary and Dr. Binit Ahuja for giving an opportunity to me to come over here and be a part of this. Thank you.
I request Dr. Yashdeep to felicitate the chairpersons with a plant as a token of our appreciation. The next platform presentation is by senior residents. For this session, I would like to welcome the honorable chairpersons onto the dais, Professor Y.K. Gupta and Professor Anita Saxena. Professor Y.K. Gupta is currently president of AIIMS Bhopal and AIIMS Jammu and was formerly Dean Academics and Head of Department Pharmacology at AIIMS New Delhi. Professor Anita Saxena was formerly Dean Academics, Chief of Cardiothoracic Center, and Head of Department of Cardiology at AIIMS, New Delhi. Madam is also currently Vice Chancellor of Postgraduate Institute of Medical Science, Rohtak. The first paper will be presented by Jaya Bishwas, Senior Resident, Department of Microbiology.
Good morning. Thank you to Dr. Rama Chaudhary and the team for inviting us and for putting up this show. Thank you. Just requesting you to the presenters to please stick to time. Thank you.
Good morning, everyone. I am Dr. Jaya Vishwas from Department of Microbiology, and the title of my presentation is In Vitro Susceptibility of FDA-Approved Drug Iravacycline versus Tigecycline in a Clinical Isolates of XDR Acinetobacter baumannii and the Study of Resistance Mechanism in it. So there is drastic increase of extensively drug-resistant Acinetobacter baumannii recently has been seen in India, which is more than 57.6%, led to very limited therapeutic options like tigecycline. And now we have also seen that then tigecycline non-susceptibility also has been associated with this overexpression of efflux pumps and presence of TETx gene in them. So recently FDA have approved iravacycline, which is also called TP434, with its modified D ring, remains unaffected by these two mechanisms like efflux pumps and the presence of TETx gene. So we just want to see that there is any difference in MIC value, which is minimum inhibitory concentration of tigecycline after interaction with this efflux pump inhibitor. If we are using, then there is any change with MIC is there or not. So the aim and objective of this study was in vitro MIC 50 by 90 of iravacycline in comparison to tigecycline in XDR Acinetobacter baumannii clinical isolate. MIC 50 or 90 is like minimum inhibitory concentration, the lowest amount of drug at which 50% of organism can be inhibited, and MIC 90 is 90% of the organism visibly can be inhibited. So we have calculated the MIC 50-90 for iravacycline versus tigecycline. Also we have compared the MIC 50-90 with the presence of CCCP, which is efflux pump inhibitor. And then we have calculated the prevalence of efflux pumps, molecular, and the TETx genes. The study was prospective, and it was observational. The cross-sectional, it is a cross-sectional study. The sample collection was from 2019 to 2021. Total number of samples were 196, which were all XDR Acinetobacter baumannii, which is like they are resistant to all group of the antimicrobials except just leaving one or two group of antimicrobials. The identification of isolates were done by Malditov, and the detection of XTR was done by Kirby-Board disk diffusion method and Broth microdilution method. And then we have calculated the MIC 50-90 with the help of Broth microdilution method. For Acinetobacter baumannii, CLSI doesn't provide any breakpoints. So we have used the FDA and UCAS criteria. In FDA, by FDA criteria, the susceptibility range for iravacycline is less than or equal to 0.5, and for tigecycline it is susceptible if the MIC is less than 2, or it is intermediate if MIC is 2 to 8, and it is resistant if MIC is more than 8. By UCAS criteria for iravacycline and tigecycline, the breakpoint is 0.5. After that, we have done this conventional PCR for detection of TETx gene. The primers are described here, and the multiplex PCR for detection of ADE ABC and ADE RS genes, which are R&D efflux pumps genes. So the result we have found, this is the graph showing distribution of MIC with iravacycline and tigecycline, and on the right side we have shown after use of efflux pump inhibitor what are the changes in the MIC. These and on the y-axis they have shown number of isolates. The blue bars are basically MIC after use of iravacycline, and the orange bars are basically after use of tigecycline. This is the break point at 0.5. So we can see here that the MIC with the tigecycline is greater, but with iravacycline it is in the lower side. So we can say that this iravacycline, which is a new FDA approved drug, which is having less MIC and which can be less resistant in these organisms. And on the right side, we can see MIC 50 and MIC 90. With the use of tigecycline, the MIC is around 2 with all the isolates, and after use with CCCP, the MIC reduced to 0.5 on the right side, and with iravacycline, this is not that changed. So we can see that iravacycline can overcome the efflux pump effects. Now, this we have total collected 196 samples, in which with the tigecycline, the MIC were maximum samples were resistant, and with iravacycline, 91% isolates were sensitive. This is the result part. Here, the detection of TETx gene. In first picture, they have shown the 16S rRNA. So that can showing that these all isolates were XDR, Acinetobacter baumannii. On the right side, this gel picture is showing the presence of TETx gene. We have found 2 isolates which were having TETx gene. Now coming on to the these 2 isolates which were having TG cycline MIC, which were 4. With the use of CCCP, the MIC range were reduced, but with iravacycline, the MIC were not affected. These were like in one sample it is maximum, one sample it is more than 1 which is resistant, and another sample it is with 0.12 which is showing this is sensitive. This is the detection of R&D efflux pump genes. Around 34, we have done 40, we have detected R&D efflux pumps in first 46 samples, and these samples, 34 samples were identified to have R&D efflux pumps. And these 4 isolates with the TG cycline, the MIC were 4, which is more than 0.5. Actually, these are resistant. With the use of CCCP, these MIC were reduced. In the presence of efflux pumps, we can see that iravacycline is having MIC around 0.5. So it is a effective drug in the presence of iravacycline. Now coming on to the discussion part, AST profile of isolates of Acinetobacter baumannii collected from ICU showed reduced susceptibility. We can use, in the presence of these resistant mechanism, iravacycline is effective. And then in the presence of efflux pump inhibitors, that this mechanism can be reversed. And by concluding with this, iravacycline has a lower MIC in comparison to TG cycline in XDR Acinetobacter baumannii. It can overcome the effect of efflux pumps and TETx genes in resistance. And the efflux pump inhibitor has potential to decrease antibiotic resistance, but specific efflux pump inhibitor should be explored. And then, sir, studying a new resistant mechanism like TETx gene should be helping in the future so that we can overcome these XDR Acinetobacter baumannii. Thank you, sir.
Thank you very much. Do you have any slide, or can you tell us what were the limitations of your studies?
Ma'am, actually we have to do further studies because we have found just 2 isolates like TETx gene only in 196 samples. So we need to do further studies. We have to include more samples to explore that there is presence of other resistance mechanism are there or not, ma'am.
Yes, please.
I was presenting a paper of the infection neurosurgery. I analyzed for 4,000 data and found 3% were the Acinetobacter. Till that time, there are only 3 papers in the world literature, 1947 to 48, I think. There is no study in the world literature after 1950. That is the time I talked to Aarti and some people in microbiology who started working on Acinetobacter. That is the time I first time understood how the Acinetobacter is creating problem in our ICU. Then we had 3 Ph.D. between 1994 and 2000, I suppose, Aarti Kapil. I was asking her when she is going to retire. Thank you.
I will just compliment this study because antibiotic resistance is less like a silent pandemic. And I, being the principal advisor to WHO and antimicrobial resistance, see much merit in this. The important thing which we must also see that the MIC is one, but the blood concentration which is achieved, which is usually higher than MIC, is another point when you translate this into clinical practice.
Yes, sir.
It is also important that when you see CCCP or this efflux pump inhibitors, how much they are effective with the tigacycline. So can the tigacycline will be as effective as iravacycline in some cases? And the next issue would be, of course, is the beyond this scope, but the accessibility, and yes, which sir. Dr. Rita and her team is working here. So thank you very much. Excellent work.
Thank you, sir.
The next presentation is by Nidhi Gupta, Senior Resident, Department of
A very good afternoon to everyone. The title of my oral presentation is Impact of Triphala Formulation on Potentiating Lenalidomide-Induced Cell Death in Multiple Myeloma via Impairing Multiple Pathways, a Complementary Integrative Approach. So as this presentation deals with the disease called multiple myeloma, so just with a brief introduction about this malignancy, which is a neoplastic plasma cell disorder characterized by the clonal proliferation and the accumulation of malignant plasma cells in the bone marrow. There are numerous treatment regimes for this malignancy like melphalan, immunomodulatory drugs, proteasome inhibitors, but despite these treatment regime, most of the multiple myeloma cases, they relapse after treatment and many cases are also refractory. So therefore, like chemotherapy associated with various side effects, morbidity, chemoresistance and lack of effectiveness. So there is a necessity to identify an efficient, less toxic and cost-effective modality for the cancer therapeutics. And now in the Indian system of medicine, there are various herbal medications which could be a promising option for the cancer treatment as an adjuvant therapy. So in this presentation, in the work, we have used a herbal formulation called Itrifol A of the moon. It mainly composed of two components, Itrifol, which is a mixture of three components like Amla, Harad and Bahera, and the of the moon component, which is mainly used for the treatment for the diseases affecting the kidneys, spleen and disorders of the liver. So in total, this Itrifol A of the moon formulation composed of the following constituents, and this formulation, Itrifol A of the moon, is being used nowadays for various diseases involving the mental illness like melancholia and apprehension. But there are no studies as of now for any cancer, including multiple myeloma. So, as for any cancer, there are multiple hallmarks associated with it, and the highlighted red one are the hallmarks which we have considered in our study. So we worked, so before proceeding ahead, so we prepared the aqueous extract of this formulation Itrifol A of the moon, and we characterized it by LC-MS, and we found out of various components, catechin is found in the major proportion in this aqueous extract, and catechin is a flavonoid which has been known for its anti-cancer property. Then we worked on multiple myeloma cell line RPMI 8226. We used another cell line also U266, but result of only this cell line has been shown here. So then we assess the inhibitory concentration of Itrifol A of the moon and the standard drug lenalidomide in multiple myeloma cells. So as we can see here, so as increasing the dose concentration of Itrifol A of the moon, cell viability of multiple myeloma cells decreases at all the time points, 24 hours, 48 hours and 72 hours. So we chose two concentration, IC30 and IC50. IC30 is the suboptimal dose, 50 is the optimal dose, and for the standard drug there was no change in cell viability observed, mainly because of its immunomodulatory action. So we treated these multiple myeloma cells single with both these drugs and in combination. So first then we performed the cell cycle analysis. So as we all know, each cell has different phases like G1S, G2M, and in flow cytometry we got peak like this for each phases, and the phase before the G0, G1 phase is the cell death phase, showing the cells undergoing death. So as can be shown here, like upper panel is showing the single treatment of both these drugs and the lower panel is showing the combination treatment, and the highlighted one is showing the effect, most effective shown in the combination group, as can be seen here. Sub G0 G1 phase showing the apoptosis or the cell death phase is significantly higher in this combination group as compared to the standard drug alone at both the doses. Then cell death could be via apoptosis or necrosis. So we assess the apoptosis via annexin 5 FITC-PI assay, in which we can easily appreciate the increase in the early apoptotic cells and the late apoptotic cells in this combination group as compared to the standard treatment alone. So this is showing when the standard drug is given along with the Itrifol A of the moon is highly efficacious even at the lower doses. Then mitochondrial membrane potential was also assessed as a red to green ratio change determines the depolarization of the mitochondrial membrane, and here we can easily see the red to green ratio has been decreased, suggesting the depolarization of the mitochondrial membrane after the combination effect as compared to the standard treatment. So then we assess the oxidative stress, which is getting decreased due to the increase in the antioxidant as a measure of Tolox equivalent in the combination group as compared to the standard drug alone. VEGF, which is an angiogenesis marker, is also getting significantly reduced. IL-6, which is an inflammatory marker, is also getting reduced after combination. Then we assess the mechanism by which it is causing its action by assessing certain pathways, and we found all of these pathways, PFAC, STAT3, AKT, they are getting affected and they are significantly down-regulated in all the combination groups as compared to the standard treatment alone. So just to summarize, that combination of Pitherophyll of the moon and the standard drug is highly efficacious as compared to the single treatment. They are affecting the multiple myeloma cells via inhibiting the signaling pathways, mainly the multiple pathways like FAT, STAT3, AKT, and ER. And inhibition of these pathways inhibiting the proliferation, inflammation, causing cell cycle arrest at subgenode-driven phase, causing apoptosis, decreasing oxidative stress and angiogenesis. And ultimately, all of these points culminate to decrease in multiple myeloma. Hence, Pitherophyll of the moon formulation has shown potential as an adjuvant anti-tumor therapy against multiple myeloma cell via affecting multiple pathways. So for the first time, this Unani herbal formulation demonstrated its anti-tumor potential and is pharmacologically safe, and it potentiates the effect of standard drug by inhibiting multiple pathways, suggesting its novel adjuvant potential. And it also might reduce the side effect of the conventional drug and might improve the quality of life. But yes, for further validation, this has to be performed in vivo and in clinical trial. So I would like to acknowledge my Chief Supervisor, Professor Alpana Sharma from Department of Biochemistry, and Ayush, mainly CCRUM, for providing the funding support. Thank you.
Wonderful presentation. Any comments or questions on this? Yeah, please.
Yes, sir. So these are like suspension cell line.
Actually, that is the defect of the human trial when you extrapolate the monolayer culture drug to a human trial. The human being has a solid tumor and cells are encapsulated.
Yes,
and
sir.
in a monolayer, when you give drug, is much more effective than the human being in vivo.
Yes, sir.
So the spheroid culture, where you take a 3% agar gel and make a spheroid and give drug, that is much better.
Yes, sir, it's a very, very good suggestion and we'll try to incorporate it. So it's a preliminary study, so we have just identified whether it is having any anti-tumor potential or not. Then we can proceed ahead for further studies in xenograft models or in clinical trial or a spheroid culture.
Yeah, madam.
Thank you so much. Nidhi, fantastic work.
Thank you.
I just want to know, like, what happens when you withdraw this drug in the culture? Does it revert back to its original cancer properties? And what happens to all the parameters which you tested?
No, ma'am. When almost the effect could be seen after 24 hours itself, but the most effective was 48 hours. And when we removed the drug from the culture cells were already in their altered parameters, like they were having distorted morphology and distorted all the parameters.
So when you withdraw this drug, so they remain healthy, or they?
So no, ma'am, they are not healthy. Sure. They'll remain as it is.
Very nice.
Thank you.
I would congratulate you on this wonderful work, because see, all the anti-cancer drugs have a serious side effects, and if we can increase their efficacy by giving any adjuvant, reduce the dose, it will reduce the dose-dependent side effects as well.
Yes, sir.
What we have hypothesized that whether this may reduce the side effects or whether it will improve the quality of life, that is a very billion-dollar question.
Yeah.
Finally, the limitation in these studies is because this is not even Triphala.
Yes.
This is a formulation which contains the 3 components of Triphala, but many, many others.
Yes.
So that is the limitation. So, but it is a good basic start indicative study.
Yes.
And a lot of studies has to be done after that.
So in LCMS, we found catechin as one of the components. So later on, we can take only that component to see
Yes.
whether it is having the same effect or not.
Correct.
Thank you, sir.
A lot of studies. In fact, with curcumin, there are hundreds of publications on curcumin.
Which cancer people give curcumin? There is a patent for the publication and the paper writing. But show me one cancer where last 50, when I was in Lucknow, director of CDRA told me, we have done curcumin test in 1960s. Okay, but I said, show me the result. It's bench and basic research, not translate to translational research? Has it happened?
I would say, Dr. Mahapatra, basic research is the basis of ultimately translational
Absolutely,
research.
but we have been trying curcumin for last 50 years.
We
Where is
have-
the result?
I would not enter into this debate now, but I would encourage this.
Thank you, sir.
I think it's something that can go to bedside. So we can stop at this and go to the next presentation. Thank you.
The last presentation of this session will be delivered by Surbhi Jain, Senior Resident, Department of Pathology.
So, a very good afternoon to everyone. First of all, I would like to inform Dr. Surbhi, due to some mishappening yesterday in a family, she couldn't join us today. So, on behalf of Dr. Surbhi and with consent from my guide, and as the event organizer has allowed me to present this work on the title, EGFR mutation analysis using the cell-free DNA from supernatant of centrifuge pleural effusion, expanding the diagnostic and predictive role of effusion cytology. So, liquid biopsy is any type of sampling that involves the analysis of various bodily fluids other than plasma, like CSF, urine, saliva. That these specimens, they are valuable sources of tumor-derived elements like circulating cell-free DNA, exosome, etc. So, the majorly exploited component in liquid biopsy is the circulating cell-free DNA, which is composed of short DNA fragment derived from cellular release of either tumor or non-tumor cells undergoing apoptosis or necrosis. As liquid biopsy has proven to be of amenable use in various clinical settings, it offers several advantages as listed here. Effusion samples represent one such source of cfDNA that are easily available and can be repeatedly obtained by minimally invasive thoracentesis for diagnostic, prognostic, and predictive analysis of tumor, while the supernatant is usually discarded as a bio-waste. So, studies have shown that the cell-free DNA can be effectively extracted from these effusions with yield comparable or even superior to the matched plasma sample. In lung cancer, currently, the mutation testing on plasma-derived cfDNA has been studied extensively and is currently approved in two scenarios. First is lung adenocarcinoma patient with insufficient tumor tissue for baseline EGFR mutation testing, or in the second, EGFR mutant lung adenocarcinoma patient developing secondary resistance to tyrosine kinase inhibitors. So what is the need of this study? There is limited studies on the feasibility of testing effusion supernatant cfDNA for molecular testing. So the proposed hypothesis of this study is cfDNA extracted from these supernatant. They are suitable for predictive biomarker testing and aid in timely patient management with comparable diagnostic yield as the tissue samples. So the aim and objective of the study are to assess the yield and sensitivity and accuracy of EGFR mutational analysis on cfDNA extracted from malignant pleural effusion supernatants. So the methodology adopted is, this was a prospective study being conducted with collection of all pleural effusion sample received in cytopathology laboratory spanning a duration of 7 months. Those patient pleural effusions with metastatic lung adenocarcinoma with available matched tissue or sediment cell block or plasma were included in this study for further EGFR mutation analysis. Post preparation of cell block, the supernatant were used for cfDNA extraction and processed further for EGFR molecular testing using available real-time PCR IVD kit, and statistical analysis were done to compare the concordance rates between supernatant cfDNA EGFR status to the available matched alternative sample, that is FP cell block or plasma. So coming to results, a total of 298 pleural effusions were examined for preliminary analysis of diagnosis and evaluation of primary tumor origin sites, and further confirmed lung primaries were selected. Quantitative assessment of cfDNA yield showed significantly upregulated bars of 10 positive malignant sample in comparison to the 10 non-malignant one. So all these result bullet points I'll explain in the subsequent tabular and pictorial representation. So of these 21 malignant pleural effusions from primary lung adenocarcinoma patient, 13 met inclusion criteria with a matched tissue or plasma sample available for EGFR mutation testing with patient clinical information. 11 were treatment-naive patient as shown patient 1 to 11. And while 2 were the EGFR relapse patient after that relapsed after a certain period of post-TKI treatment. So matched tissue sample in these patients used for EGFR mutation testing include cell blocks in 11 samples and non-procedure matched lung biopsy samples and plasma in 1 each. 2 highlighted sample patient 4 and 11 they tested negative for EGFR mutation on both tissue and supernatant cfDNA. No false positive cases were reported, and additionally we tested some more negative cases as well. So the overall concordance of mutation results between the malignant pleural effusions and tissue plasma results was found out to be 92% in 12 out of 13 cases, and only 1 case that was found to be false negative. That is patient number 6. So, and the 2 post-TKI progressed patient harbored the founder mutation as well. And in addition to this, that I would like to point out here that in patient 13, the sediment cell block we tried to test for the resistant T790M mutation that we could not able to detect on the cell block, but we were able to detect it on the supernatant cfDNA. So these are some amplification plot of that post-TKI patient. The right-hand side plot that shows only L858R. That shows only L858R founder mutation is from the cell block, and the left-hand panel that is showing L858R along with T790M, that is the resistance EGFR mutation. So this is again a case that was found to be false negative. So despite the concentration was good enough, but still we could not able to detect. So I suppose that we would definitely test it with some sensitive droplet digital PCR technique. So these are again the summary. Coming to discussion. In our study, we tried to compare both the DNA from supernatants and tissue cell block sediments of pleural effusion for mutation analysis, and our results demonstrated 92% concordance rate. Neither the tumor fraction nor the quantity of the cfDNA extracted from the effusion supernatant emerged as variable that influence EGFR mutation results. In the single case that was false negative for EGFR mutation, that the reason is not clear, or that as I said earlier, that we can try for the more sensitive techniques to detect like ddPCR. So the study highlights that paired mutation analysis is essential in liquid biopsy samples. So there are further studies that show that supernatant of effusion samples, they are a source of tumor DNA and hold immense potential to play a major role in diagnostic pathology. Coming to conclusion, by expanding the spectrum of cytology for molecular testing, the cfDNA from malignant pleural effusion supernatant they serve as a valuable source for molecular testing, exemplified by the current study and in patients with inadequate tissue sample. So for future direction, we are further exploring the study for inclusion of other pleural effusion sample, not only lung, but for other samples also, by using some high throughput next generation sequencing technology and subsequent validation with a highly sensitive droplet digital PCR technology. Thank you.
Are there any questions from the floor? Yes, please.
Sir, very nice presentation and very innovative work. This is the circulating tumor cell and the cell-free DNA is the new buzzword in science, because we want to understand why there is complete remission after that there is tumor recurrence. And that one answer could be the circulating DNA and tumor cells which are embedding in the other tissue and producing recurrence. So, but you should have taken the non-malignant pleural effusion as control.
So that we also tested.
But you have not in none of the slides.
Actually, the presentation was submitted by Dr. Survey.
Yeah.
Yesterday I got
so
to know.
So if you take the non-malignant pleural effusion and if you find something that can give you false positive, except in this sample you can say false negative. You cannot talk about false positive rate. So that was one of the major limitations of this work.
Thank you, sir.
So wonderful work. Just one comment. You have a 1 out of 10 false negative.
Yes, sir.
If I convert this in as 10% false negative. Will that be acceptable in terms of
So right now the
predictability?
sample size is very small.
Yeah.
And second thing, we were not able to get the matched tissue sample along with that, the liquid biopsy sample. So because there were many samples, but we were not able to get the matched sample.
Yeah. So
So
as
that's
you
it.
said that perhaps a larger sample size will clarify this. What is the, whether it's a 92% concordance or maybe it may vary. But false positivity and false negativity will be a crucial thing. But this is of course the future direction in this. Thank you.
Thank you, sir.
Thank you.
Thank you, everyone.
Thank you, sir and madam. I request Dr. Yashdeep to felicitate the chairpersons with a plant as a token of our appreciation. For the next session, I would like to call upon the dais Dr. Vivek Dikshit to conduct the proceedings.
धन्यवाद डॉक्टर शौविक। आप सभी को नमस्कार। व्याख्यानमाला के इस द्वितीय सत्र में आप सभी का बहुत बहुत स्वागत है। इस व्याख्यानमाला के द्वितीय सत्र के चेयरपर्सन से मैं अनुरोध करूंगा कि वे कृपया मंच पर आने की कृपा करें। इस व्याख्यानमाला के मुख्य वक्ता डॉक्टर राजेश भाटिया, आपसे निवेदन कि आप कृपया मंच पर आएं। इस व्याख्यानमाला की अध्यक्षता कर रहे हमारे चेयरपर्सन्स डॉक्टर चित्रा सरकार, फॉर्मर डीन रिसर्च, प्रोफेसर एंड हेड ऑफ डिपार्टमेंट ऑफ पैथोलॉजी, डॉक्टर के दीपक, डीन एग्जामिनेशंस, प्रोफेसर एंड हेड ऑफ डिपार्टमेंट ऑफ फिजियोलॉजी। डॉक्टर चित्रा सरकार से निवेदन कि आज के मुख्य वक्ता का परिचय कराने की कृपा करें।
Good afternoon. Congratulations to Dr. Rama Chaudhary, Dr. Vinita Ahuja, and the entire research section team for organizing this excellent event. And on behalf of Dr. Deepak and me, it gives us great pleasure to be chairing this keynote lecture by Dr. Rajesh Bhatia. Dr. Rajesh Bhatia is the Regional Technical Advisor to Food and Agricultural Organization of the United Nations and Senior Technical Advisor to Infectious Disease Detection and Surveillance of International Classification of Functioning, Disability and Health, World Health Organization, WHO. He has previously worked as a regional advisor, communicable diseases, and director, Department of Communicable Diseases, WHO Southeast Asia Regional Office. Dr. Bhatia did his MD in medical microbiology and bacteriology from the Maulana Azad Medical College, New Delhi. He's an expert in communicable diseases, public health, and drug resistance. Antimicrobial resistance is a major and serious concern, as we all know, and Dr. Bhatia has been a proponent in adopting the One Health approach with coordination and cooperation between human health, veterinary, and environmental sectors. So Dr. Bhatia has been at the forefront to emphasize the need to identify significant research areas that can be taken up for instituting measures to prevent and contain antimicrobial resistance in the veterinary sector. We all look forward to his lecture on One Health, and it gives me great pleasure and it's an honor for us to welcome Dr. Rajesh Bhatia to deliver the second keynote lecture of the morning.
Thank you very much, Chair, for these kind words, and thank you, Professor Rama and her team, for giving me an opportunity to be here with all of you. Chairs, I will make two submissions before I start my presentation. First, my presentation is not research-oriented. So from that perspective, in this particular gathering and at this occasion, this may be considered as an outlier. But my second submission is that whatever I'm going to sell you today, I'm going to sell you a philosophy, I'm going to sell you a concept which is of One Health, and my request to all you, the researchers and the budding researchers, is to generate data, to generate evidence for the implementation of this concept or this philosophy. And as I come to the end of my presentation, you will recognize that there are several areas where important contributions can be made towards One Health, which is leading to the basic aim of all the professionals or medical professionals who are in this hall. What is our basic aim? What is the mission of our life as medical professionals? To protect health and to promote health, isn't it? Especially from infectious diseases in the developing countries. So allow me to start with that. I feel the role of health professionals in infectious diseases is to treat the patient, but that's not the only role. Our role is also to prevent the disease so that it doesn't spread to other people. Our goal is also to control the disease so that we lead to our ultimate objective of preventing the diseases and then through that protecting the human health and promoting the human health. Now, to carry this thing forward, the big picture, I'll bring it now to few specifics. I'll try to show you three representative infectious diseases, and I'm sure you all have seen this kind of patient. This is a patient with hydrophobia or the end product of rabies. This is a patient of non-viral hepatitis jaundice, which is due to leptospirosis. And this is a patient who is admitted with severe sepsis in the infectious care, ICU following a severe food poisoning. Now, these are three patients about which I would be talking in my next few slides and bringing home the point of One Health, which is a theme of my presentation. Now, what is common in these three diseases? You saw rabies, you saw leptospirosis, and you saw foodborne infection leading to severe sepsis. What is common in these three? They are all medical emergencies. They can be fatal unless they are treated or a immunoprophylactic given after an animal bite. But they are just three of more than 200 known zoonotic infections. That is the infections which transmit between humans and animals. It's not only from animals to humans, it is human to animals too. So they move between humans and animals. And these comprise a large number of diseases which we encounter nowadays. And I'll come to some of those pandemics and epidemics also later because of this concept. So in case you need to control these diseases, which is our ultimate objective, as I showed in my second slide, you have to work not only with the human sector, you have to work with the animal sector also, because diseases are originating from them. So my first carry home message is working together. And next 15-20 minutes, I will be repeating these two words around 5 to 10 times, because that is the essence of One Health, working together across sectors, across disciplines, across institutions, so that we can promote human health and protect human health. Now let's see these three diseases, representative diseases, one by one, so that I can still make my point much clearer. You see this picture around you, even in New Delhi or other parts of the country. You see this picture also, close contact, close relationship between humans and dog in this case, but of course in the rural area with all sorts of animals. And then through these animals we get rabies, leading to hydrophobia and the death. And see the number of people who die every year in this country from rabies. It's around 19,000, which means 50 deaths every day from hydrophobia in this country. This also includes around 20 million people who are bitten, and then they have to go in for post-exposure prophylaxis using a treatment which costs around 3,000 rupees nowadays. Maybe it would be increasing. And most of these cases and deaths are in the productive age group. My parents or my very young children may not be coming out and coming in contact with the rabid animals or the stray animals. But of course, the productive people who go out, they do come in contact with these kind of animals, and then they can contract disease. Our contribution to global rabies, 40%. Leaders, we are the leaders in the world in this contributing to the deaths due to rabies in the world. And then, of course, this is the face of zoonosis. This is the face of rabies. In case you have seen a rabid dog, those of you who have seen it, you can never forget it. Such a ferocious look. You can see the death there, and that this is the kind of animal which we have. So in case you want to control hydrophobia, you want to prevent it in people, you have to prevent it first in animals, especially in dogs in India, because 96% of our rabies cases are because of dog bites and few because of cat bites. We don't have the wild animal biting in India, or at least we don't have the data for that. So this dog, rabid dog, where does it survive? What does any living creature, including dog, would need for its survival? Food. Without food, it cannot survive. Where does it get its food? It gets its food from the garbage. In Delhi, you go to any dhaba, you go to any small nursing home, any small hospital, move around, and you will find a lot of trash outside. And what else you will find there? You will find the dogs there. The dogs are not attracted toward trash, they are attracted toward potential food. So they come there, they have their food, and that's where the dogs survive, that's where they breed, and that's why their number keeps on increasing year by year. And this is another scenario. So there is a human being, there are a couple of dogs, and there is trash or garbage where the dogs are feeding. Now this completes the epidemiology of rabies. If trash is there, dogs are there, dogs contract rabies, and they pass on the rabies virus to the human beings. So that's how the rabies epidemiology is completed, and you get this kind of patient. So now very simple common sense. You want to control this disease, what do you need to control? Only the person who has been bitten, who is the end of that cycle? Or you want to control it in the dogs? Or in case you want to only control in the dogs, don't allow them to have foods from somewhere. So you have to control it in the environment also. And this working with the environment people, working with the municipal corporation people, working with the veterinary sector, and working with the health sector, that working together is the essence of One Health if you wish to fulfill the mission of your life, which is promoting and protecting the human health. So that's the main point that I want to make in next 10-15 minutes again. Now let's go to the second disease that I showed you earlier, the non-viral hepatitis jaundice, leptospirosis. From where do we get leptospirosis? It's a bacterial disease. The bacteria proliferate in the water or in the water bodies, which have been contaminated by the urine of different animals. Now this is the cycle of leptospirosis. Rats, rodents in the urban areas, and the cattle and other animals in rural areas, they urinate in the stagnant water, and then the human being goes there. The skin comes in contact with that leptospirus bacteria, gain into it, go to the liver, and cause all kind of complications, clinical complications, and then they land up in a hospital for treatment. You treat the patient. Leptospirosis is easily treatable infection. That's not a big deal. Even the penicillin works wonderfully well in this disease. But then do you want to only treat? Again, coming back to my initial slides, we want to prevent it. We want to control it. So if you want to prevent and control it, you have to see to it that the animals do not get into water and urinate there. They don't contaminate those water bodies. And for that, again, you can't work alone. You need people from different sectors, from different departments, from different areas who are not directly related with us, but we need to work with them to protect human health. Now, this is the third patient now. The patient in the ICU with severe sepsis and not responding to any antibiotic. I'll come to that in next slide. But these are the common things by which the foodborne infections move. We call them 5F: food, fingers, feces, fomites, flights. All these 5F, they bring bacterial infection into your food. You eat it, the food chain gets contaminated, and the person may develop, not necessarily always, but may develop a foodborne infection, which in case of invasive pathogen, ultimately leads to sepsis and may end up with a person in ICU, which we showed you earlier. This is the number of bacterial genera which can cause foodborne infection, 31. And if you read the names on the other side of this thing, you must have heard all these very common names: Staphylococcus aureus, Salmonella, Campylobacter, Listeriosis. All these are common names, and most of these are transmitted through the agency of food. They come in the food cycle, and then they infect the human beings. The data given on the screen is from WHO. Now look at the number of cases, and look at the economic loss. It's $110 billion every year that occurs because of foodborne infections in this world. And the second part, which is perhaps more important, and which was referred to by Professor Y.K. Gupta in the previous session about the antimicrobial resistance, most of these pathogens are resistant to multiple antibiotics, especially the affordable ones. Now, whether it's a Staphylococcus aureus, you have an MRSA, or you have an ESBL producing pathogen, most of these would be having some sort of component, some sort of resistant gene, which will not allow them to be treated by using affordable antibiotics. So this antimicrobial resistance is now becoming the most important challenge to humanity in its fight against infectious diseases. This is number one challenge now. And in case you don't have the idea, the people at the highest political level in the world have been talking about AMR. Whether it is UN General Assembly, it is G7, it is G20, it is G77, it is OECD, it is ASEAN, it is SAARC, everywhere, simply because they are now recognizing the impact of AMR on human health, on animal health, and on human development. If you look at the Sustainable Development Goals, there is a political declaration which talks about antimicrobial resistance. So that's the kind of problem that AMR is becoming, and I'll show you next couple of slides what's happening in AMR and what we all can go by working together. That's the most important thing. Now, what is AMR? You know it. No one knows it better than people sitting in this auditorium. It is unresponsiveness to antimicrobial agents when given in standard doses, making it difficult to treat an infectious disease in humans. In previous session, we were talking about Acinetobacter. We have been talking about MRSAs. We have been talking about Pseudomonas since ages, especially when Professor Srinivas was here. I think he brought Pseudomonas onto limelight in India. So we have been talking about all these things. And what is all these things? All these things are resistant to common antibiotics. Resistance in bacteria is not induced by us, is not induced by antibiotics. It's a very important thing to understand. It's a naturally occurring phenomenon. It is genetic changes that all the living organisms continue to undergo. So that's not something which we can do anything to control, contain, or prevent it. But then what happens? All antibiotics have got the capacity to select out those resistant variants. And once those are selected out, the other variants, they just move out. And then these resistant variants become the dominating flora in that particular area. Do you think in your hospital MRSAs were always there? Initially, there were simple Staph aureus responding to antibiotic. Then somehow by mutation or from some other place, MRSAs came there. They had a better survival instinct. They were not responding to antibiotic. They were proliferating. So they became the dominating flora. Similarly for Klebsiella, Pseudomonas, Acinetobacters, all these things ultimately because of the hospital environment where antibiotic pressure is there in plenty, they survive, they become the dominating flora, and then they infect patients, whether it is through ventilators or through any other source, which we have been talking since morning in this hall. And this is not a sudden thing. It hasn't happened yesterday. It has been a long-term misuse of antibiotics. whenever you have 5 minutes, think about it. Where do you see use of antibiotics? At least in humans. I'll come to other sector also. Person sitting at home, chemist selling antibiotics without any prescription, without any knowledge, a quack in a village giving all sort of antibiotics and even gentamicin injections to people. Now, all these things have been going on for ages and ages and ages. It's not that these things started yesterday. And that's the reason that with accumulation of resistance in every bacterium, 1, 2, 3, 4, 5, 8, 10 resistant genes became manifested. And then we call them multi-drug resistant organisms in our language, but the common media and the people call them superbugs. And the superbugs have been because of the long-term neglect of the rational use of antibiotics. What you see here is only the tip of the iceberg, but if you look at the bottom, it has been going on for ages. And now we are at a situation where for many resistant organisms causing disease, we have practically no antibiotic. And if we don't do anything now, what is going to happen in next few years is here. By 2050, this is a report which was commissioned by the Prime Minister of the UK a few years back, and Lord Jim O'Neill chaired this thing, and they did a lot of studies, and ultimately they came out with some figures. By 2050, that means 28 years from now, if sustained actions are not taken to contain AMR, what is going to happen? There would be a mortality of around 10 million per year, which means more people would be dying because of infection with resistant pathogens than people dying of cancers and people dying of roadside accidents, putting them together, more than that. And then this also shows what would be the loss of food productivity, 7.5%. This also shows how many people would become unemployed. They will go into the poverty. This also shows how high would be the cost of treatment. And you know it. If there is a person who is not responding to conventional antibiotic, you do some more investigations, then you bring in more toxic and more expensive antibiotics. So there is a cost to the person, there is a cost to the society, there is a cost to the health system, there is a cost to the country. And then, of course, this is a global cost. And this is what is worrying the global leadership. The global leadership all across the world is worrying them. Now, let's see. We know it. Indiscriminate use or overuse of antimicrobial agents is the main driver of antimicrobial resistance. You all know it. But then it's also known the more we use the antibiotics, the more we lose them. But where do we use them more? Don't tell me in ICU or a medicine unit or surgical unit, no. We use them a lot in different sectors. We use them in human health, we use them in animal health, and now, shockingly, there is a greater use of antibiotics in agriculture. People are spraying it onto their crops. You have to go to the villages and to find it out what is happening there. And then, of course, from all these three areas, ultimately antibiotics find their way into the environment, where they again select out the resistant pathogens who again through the food chain gets into us, human beings. We develop foodborne disease, we develop sepsis, we land up in the ICUs. Now that's how the vicious cycle goes. Now the most antibiotics are used in the livestock and poultry. Not only in India, across the world. Europe has started taking steps, but everywhere else. Now you will say that the animals don't fall that much sick. The antibiotics are not given to animals only for therapeutic purposes. These are given to animals to promote their growth and to prevent them from getting infected and dying, because that's an economic loss. And to prevent that thing, there are only two options. Either you have a good sanitation, good hygiene across the farm or across the village, or you start giving the cheap alternative, which is antibiotics. You keep on pushing antibiotics into animals. Bacteria are almost same. Staph aureus, E. coli, Klebsiella, they are all same. They are selected out and then they move to the human beings. So we are using it there also. So that's the point I want to make, that all sectors which are using antibiotics, we need to work together with all of them if we want to protect human health. So we need to work with all sectors, only then we would be able to do AMR. And friends, just one thing, especially to the younger lot, 5 years down the line, you will find that environment sector has to play the most important role in containment of AMR. Human health, you all are converted, you know it, and the things will get amplified. Veterinary sector, yes, things are happening very nicely. Environment sector is still ignorant, or they are just saying, this is not our problem, which is not the truth, because in environment is the place where the resistance is selected out. So 5 years down the line, environment will have to play a leadership role so that AMR can be contained. Okay, so now again, Professor Vaike Gupta earlier termed it as silent pandemic. So I'm just echoing what he said, that AMR is a unseen, invisible, insidious, going on for years, but is a silent pandemic, which is going to create havoc for us. Now, the problem is, when there is a silent pandemic, as the name says, no one talks about it. It's silent. But when there is a pandemic like that of COVID, which is a noisy pandemic, last 1,000 days that we have been under the shadow of COVID, you look at the media, you look at people, look at newspapers, everyone is talking about COVID. Does anyone talk about antibiotic resistance that much? Not even 0.1% of that. So there is one a noisy pandemic and there is one silent pandemic. But the point that I want to make here is not only about AMR, but about the noisy pandemic also. The noisy pandemic is, again, a problem which is to be faced through working together, working with people. Now, what are these noisy pandemics? Let's see this. Now, these are from the current millennium data, only from 2000 onwards. We are not going back into the history of Spanish flu and Hong Kong flu and other things. Look at this. Every 3 years, you have one or the other, what WHO calls as public health emergency of international concern, or a big epidemic, or even a pandemic, like we had flu in 2009, and we had COVID just now. Now, every 3 years, now we are having a public health emergency of monkeypox. So 22 years, 7 public health emergencies every 3 years. What does it mean? It means that 2025, we are going to have another pandemic. Don't ever think that there will not be any pandemic. Pandemic is imminent. The only thing about which we do not know is at what time the pandemic will come up. But it's most likely, if you look at the pattern, every 3 years, we have to have one or the other pandemic. So this is what we are going to have. What do these pandemics do? They kill people, they cause misery, they cause social chaos, as we all have seen, but they also cause huge economic loss. Now, if you look at the bottom line, the last line that I'm showing is for the pandemic, COVID pandemic. IMF, the International Monetary Fund, has estimated a loss, economic loss to the world till 2025 because of this pandemic would be $28 trillion. And you know what is the economy of India? Our annual economy is 3 trillion. So just imagine that if we have no economy for 10 years, then we become equal to the impact of the pandemic due to COVID-19. Huge pandemic. And it's still continuing. No one is saying pandemic is over. 1,000 days are over, but pandemic is not over. It is still continuing. That brings me to the question. I'm talking of these important public health emergencies. Where do we work with other sectors? Now, this is the dynamics of disasters originating from wildlife. No, wildlife is in its own silo. It's working on its own. It has got its own ecosystem. But when we invade that ecosystem, then we are in for big trouble. Now, 850,000 viruses are estimated to be circulating in wildlife. 850,000, of which 20,000 almost are coronaviruses. And how many coronaviruses have we encountered till date is 7. COVID was 7th coronavirus that we have encountered. So we have a large number of coronaviruses itself, which are there in the wildlife, circulating very happily. But we don't want them to circulate happily. That's the problem. We want to clear the forest so that we can grow more food. We want to make our highways through forest. We want to have some wildlife from there. We want our livestock to go there and have their own food. Now, all those things bring us and our livestock in contact with the wildlife and in contact with the vectors and in contact with the viruses, which are already there in the wildlife, and then they are brought first to the villages or to the suburban areas, then they come to the urban areas, and then they erupt or explode in the form of epidemics or pandemics. Now, this is what this picture shows. This is how there is spillover, and then there is an amplification in domestic animals, goes to the rural population, goes to urban population. All the public health emergencies in current millennium have been because of this. So if you want to prevent pandemics, if you want to control them in the early phase, what do you need to do? You have to work with people who are responsible for wildlife. You have to work with people who are responsible for livestock health, especially in the rural areas, and you have to work with your own health sector, public health, hospitals, and all other related areas, and only then you can contain it right in the beginning stage. This is again a continuous process, but we need to be alert, we need to be strong to combat this kind of problem. So the message is loud and clear. If you wish to prevent and control rabies, if you wish to prevent and control leptospirosis, if you wish to prevent foodborne diseases, and many other zoonotic infections, around 200 or so, and you wish to prevent and control silent pandemic of AMR and detect early all those things which can erupt into a pandemic, you need to work together with other sectors. You need to work together. There is no other way out. This is the philosophy now which has been globally accepted, and I'll come to that little later. And so four important stakeholders are animal health, environment, agriculture. Most importantly and most neglected chairs are the communities. No public health action can be done efficiently unless the word public in public health means public has to cooperate. Whether it is COVID, whether it is AMR, whether it is rational use of antibiotics, use of safe water, use of safe food, ultimately actions are at community level, and there we have to reach out to them. We have to inform them, we have to educate them. Okay, so what does it show? It just shows that health of people is interconnected between people, animals, plants, and their shared environment. This is what we have been talking for the last 15-20 minutes. This is the philosophy of One Health, and One Health is a collaborative, multi-sectoral, and transdisciplinary approach at all levels, from the national level right up to the most peripheral level. And this is the demand of the day, that we need to work together so that we can prevent and control most of the infectious diseases, AMR, as well as we can contain the pandemics right in the beginning. Now look at the benefits of One Health. If you look at the One Health, at least 60% of all human diseases have originated from animals, 60%. So if you can contain it, this number 60 will not swell to 70. This may come down gradually to 50, or if you can eliminate some of the diseases. The food production goes down. So in case you can have a good One Health system, your food production and nutrition security can be maintained. And then of course your ecosystem, where in the past 30, 40 years, several emergencies have come up, can be controlled or contained. Now this is the thing which I showed you, which I talked to you earlier also, how we get into the wildlife and how big it is an economic problem, and what are our social issues, what are our economic challenges that we venture into the forest and into the wildlife and bring with us some of the viruses from there. This is the component of One Health. Someone asked me, do you want that Ministry of Health, Ministry of Agriculture, Ministry of Environment should be dissolved and they should be put together as Ministry of One Health? No, that's not the objective of One Health at all. Objective of One Health is very clear. All ministries should have their own capacity. Make yourself strong enough to combat these things, but work along with your other counterparts in different ministries so that best results are obtained. And I have got 5 C's for that. Increase your capacity, that is your first C. Then coordinate with others, collaborate with others, cooperate with others, communicate with others, and have commitment to this thing. And how these things can be done? To implement this thing, there is a formula of 3 Ps. There should be national policies, There should be national programs. There should be people who are working on it. And how these things work, the policies are converted into legislation, into regulations, and the programs. The programs are implemented through joint planning between different sectors, working together, once again, working together, joint development of strategies, joint development of operational plans and the implementation, and then of course, awareness among the community, advocacy at the highest level, and the participation of all people into it. Whatever I have said makes sense, but it's very simple to talk. It's very difficult to implement it, simply because we all, health department has been working as health department for ages, veterinary department has been working as veterinary department for centuries. It's very difficult to bring them together. And people don't recognize the economic benefits of working together. There's even a disagreement between professionals on the way forward in this thing. We haven't done sufficient capacity building jointly with other sectors, so each one understands the compulsions and the strengths of the other sectors. And there is even a lack of clarity about the definition, concept, and scope. And that is, friends, what has brought me here. That's what Dr. Rama Chaudhary wanted me to come here and talk to you about, so that we can at least come to a conclusion. Yes, we need to work together so that we can prevent diseases, protect the human health, and promote human health. Now, these are the key elements. I will not go into details of these. Anyone can make out this all logical thing. Policy, funding, programmatic approach, human resource development, capacity building, monitoring, evaluation, community involvement. But there is a FAO document. It was published just a year back on the National Network for One Health, which gives you all these things. Last but one slide, power of One Health. The One Health will give you means the entire global community, lot of benefits. The benefits would be you would be able to preserve and use your medical interventions, especially antibiotics, for a much, much longer period. The one worries us in AMR is that we are gradually sliding into post-antibiotic era. I think that we can delay or that we can prevent in case we can have a system by which we can see to it that our antibiotics are preserved, our nutritional security is preserved, our environment is preserved, our flora and fauna in the wildlife and in the rural areas are preserved. There are huge, huge benefits. They all come from this just by working together. And one thing perhaps you must have noted, I have not asked so far for any additional funding. I have not said that give us more money for this thing. That's what every program does. I'm not asking for more money simply because money is available to all the departments for their own capacity building. But what I'm asking is working in a different way, but working together. Communicate, collaborate, coordinate, commitment. I'm just asking for those kind of things so that people can work together and then bring. So friends, for you, a few carry home messages on One Health. I hope you can remember 2 or 3 of these. That would be sufficient. It's a validated, holistic approach. It's not something which is, I'm saying, globally it has been validated. It's a holistic approach. It looks simple and powerful, but it is extremely complex implementation process, and that you have seen. It warrants a multi-sectoral, multi-institutional, and multidisciplinary approach. There is a need for a policy shift to desectoralize. Desectoralize doesn't mean demolish one sector, but desectoralize means working within a sector. No, we need to work across the sectors. That's what One Health is talking about. This is the action where I want all of you to contribute. Generate data, generate evidence, undertake research, whether basic research, operational research, translation research, so that the policymakers have got the ammunition in their hands to say, okay, now we can go ahead with One Health. Because this is what should be the cornerstone of One Health, data and research-based or the evidences. Should not be on the basis of someone's whims and fancies, but should be on the basis of scientific evidence. And that's what you all can do by working together. Can you have some research projects with veterinary people and with environment people, maybe on AMR, maybe on some other areas? I think you have plenty of projects with IITs on information technologies. Why not to have some projects with the veterinary sector as well as with the environment sector? And of course, we need to make a change in the national thinking. This is just a beginning. Just a beginning. Even if 1% of you talk about it to other people, then there is a cascading effect and more people will know about it and some of them will start working on it. And this is a must for effectively treating your patients and preventing spread of AMR and other zoonotic infections. Friends, this is where our future beacons the concept of working together or the concept of One Health. Thank you very much for your patient hearing. Thank you very much.
Thank you, Dr. Rajesh Bhatia, for the wonderful talk and taking us to the society and social concern about the medical issues. And it's been a wonderful talk, as I say. We have a constraint of the time, so I'm afraid I will not be able to take any questions, but I'm sure he will be available on email or some other. So it's very important. So with that, we come to the conclusion of such an important talk. Thank you. Once again, we can clap for him.
Thank you, sir. Thank you very much. Thank you very much.
Over to the organizers. Thank you.
बहुत बहुत धन्यवाद आपके इस ज्ञानवर्धक उद्बोधन के लिए। मैं चेयरपर्सन से रिक्वेस्ट करूंगा कि आज के इस सत्र के मुख्य वक्ता का अभिवादन करने की कृपा करें। बहुत बहुत धन्यवाद। मैं डॉक्टर दीपिका से निवेदन करूंगा कि इस सत्र की अध्यक्षता कर रहे हमारे चेयरपर्सन्स का अभिवादन करने की कृपा करें। बहुत बहुत धन्यवाद आप सभी का इतनी एकाग्रता और ध्यान से इस सत्र में इस व्याख्यान को सुनने के लिए बहुत बहुत धन्यवाद। अगले सत्र में मैं डॉक्टर अर्चना और डॉक्टर टोनी से निवेदन करूंगा कि वे इस कार्यक्रम को आगे लेकर जाएं।
Thank you very much. We are going to move on to the formal inauguration of the second AIIMS Research Day. And while we prepare for that, I request you all to keep your mobile phones either in the airplane mode or switched off, please. Thank you very much.
We would like to now invite the dignitaries, our Honorable Director, Dr. M. Srinivas, Dean Research, Dean Academics, and Dean Examination onto the stage, please, for the lamp lighting ceremony to initiate the inaugural function.
I request the dignitaries to light the lamp. We request our Dean Research, Professor Rama Chaudhary, to deliver the welcome address to the audience.
कमल का
So good afternoon, everyone. Honorable Director, Sir, Dr. Srinivas, Dean Academics, Dr. Subrata Sinha, Dean Examination, Dr. K.K. Deepak, our Deputy Director Administration, Dr. Agarwal, MS, Dr. Sharma, and all the senior faculties present here. So I'm just going to give you some of the reflections. So we all know that the mission of AIIMS has been education, research, and patient care. Research is one of the major aspects of our institute's mission. And AIIMS research section has actually coordinated the various activities of extramural research projects which are funded by various national, international agencies. We have got TCRF, all of you are aware about it, Clinical Research Unit, and also Center for Medical Innovation and Entrepreneurship. This is the organogram of the research section. We have got good WHO collaborating centers of neonatology, diarrheal diseases, physical medicine and rehabilitation, cardiovascular epidemiology and prevention, genetics, maternal health, and oral health. Then the annual AIIMS Excellence Awards, we are all waiting for that. Many of you are going to get this award today. And we have awards in the AIIMS Excellence category for basic sciences and for clinical sciences. And in addition, we have basic and clinical sciences, especially in oncology. These awards will be presented today. A new award has been constituted, which is Dr. Surendra K. Chawla Best Research Paper in the field of cardiothoracic surgery. Also, there are many intramural schemes which were started, and these are meant more for the your younger faculties, assistant professors and associate professors at AIIMS. Grant is given for 2 years. We have also a lot of collaborative research projects going on. We also have undergraduate research mentorship scheme, and you saw some of the MBBS students presenting out of that scheme, their work. We also have inter-institutional collaborative research projects with IIT Delhi, IIT Kharagpur, THSTI, and AIIMS has collaborated with University College London, and a lot of projects were awarded in last few years. Then AIIMS endowment scheme is also there. This endowed chair is called Shobha Seth Wenger's Distinguished Chair in Gynecology under Meera Seth Corpus Funds. There is Infosys Chair in Oncology, and Infosys Foundation has donated money to AIIMS to establish one chair in cancer. We also give travel fellowships for faculties, which is 2 to 4 weeks, to gain advanced training in emerging areas of medical sciences and utilize them for to work at AIIMS. In addition, there are travel fellowships for the students and scholars. And if you see, more than almost 2 crores funds has been received from various extramural grants from DBT, DST, ICMR, and that is actually the fund which has helped so many of you to do very good research work. We also have intramural grants, and this is with AIIMS collaboration with IIT, THSTI, UCL, UG mentorship, and almost 17 crores of fund is given in that. In addition, during COVID, to intensify the research in the COVID area, the first call was given in March 2020. Research on SARS-CoV-2 and COVID-19, 46 projects were granted. Second call in May 2020, another 29 projects granted. And long-term health consequences of COVID-19, again in January 2021, 11 projects were granted that time. In addition, we had fear of mucormycosis. We had large number of cases, and a special call was given for mucormycosis and COVID-19. 26 projects were granted that time. So if you see the publications just in the field of COVID-19 research itself, you will find that 2020 we had almost more than 150 publications, 2021 almost equal, and 2022 already we have more than 60 publications now. So these are some of the various projects funded by various international agencies, and the research institution is contributing to top first 1% scientific paper based on impact factors and citations as per the UGC publication, and after Indian Institute of Science, AIIMS rank second actually, is the only institution with the overwhelming service component in addition to research component. For the Indian Institutes on Global List, I mentioned in my morning welcome address also that it is after Massachusetts General Hospital, Mayo Clinic, AIIMS Delhi is ranking third world over. So also the influence of AIIMS research on policy making, it has helped in making policies in National Blindness Control Program, Fluorosis Control Program, control of iodine deficiency, hepatitis B and C, testing in blood banks, tobacco control program, geriatric age group care, and national program for prevention and control of diabetes, CVD, and strokes. Influence of AIIMS research on policy making, and also in oral health care program, trauma care policy, intervention in high-risk newborns, zinc deficiency, emergency contraceptions, etc. So what is our vision towards 2022 to 2030? We heard about a very good lecture on artificial intelligence in healthcare in the morning, and that is the vision of AIIMS, as also mentioned by our director, that we should take artificial intelligence as much as possible and use it in healthcare and do some innovations. We have already seen some of the work presented by some of the students, so how we can really take it up. And another sector which is very important, we talked about AMR, just now we heard from Dr. Bhatia about antimicrobial resistance. So this is another area in which we are going to work, like bacteriophage research and therapy center, as an alternative to antibiotics, whether we can really use it as one of the therapeutic options for treating these infections. So these are some of the teams, and of course everyone is part of it. But yes, just to mention, we have CCRF team. This is a total research section team. And we are thankful to everyone for their contribution. So I just also wanted to thank, I'm sorry, the slide is not coming, but I really want to thank our earlier, all the past heads of the research section, and also the Dean Research, including Dr. Kotwal, Dr. A.B. Day, Dr. Mehra, Dr. Acharya, Dr. Mahapatra, Dr. Chitra Sarkar, and Dr. Subrata Sinha. And more importantly are the associate deans. And there I must mention Dr. Pramod, Garg, who is also here, And Dr. Vineet Ahuja is already continuing in this. So with this, I would like to thank each one of the, research team. And I think nothing is possible without the teamwork. This day has been only possible because of the teamwork. And I really thank each and every person of the research group and research team. Thank you so much.
Thank you, ma'am. Now we will move into the release of the research directory, its latest edition. And for that, I would request the dignitaries on stage to please come forward.
We also request the editor for the research directory this edition, Professor Rakesh Lodha, and his team to come forward to assist in the release of this directory, please.
Please move on the side.
Thank you. Thank you very much. Now we go on to the next segment, which is a much awaited segment, which is the announcements of the various excellence awards for research. So because of COVID, we are going to have the awards distributed for 2 years, 2 academic sections, that is 2020 and 2021. So we will start with the Research Excellence Awards in the Clinical Sciences category. And we will also project the papers that have been awarded. So for 2020, I'll start in the reverse order, as is logical. I request the director and the dignitaries to come forward to present the awards. So the certificate of recognition for research in a clinical study, the first certificate of recognition goes to Dr. Urvashi B. Singh for her paper on Mycobacterium tuberculosis polyclonal infections through treatment and recurrence. This was published in PLOS One. The second certificate of recognition is awarded to Dr. Bhavuk Garg, additional professor in the Department of Orthopedics. The paper was published in the Journal of Spine, and this is the modified posterior vertebral approach for resection in a spinal deformity. It was a retrospective comparative study. A third certificate of recognition is awarded to Dr. Sanjay Sharma for his paper presented and published in the Journal of the American Society of Radiology. This was on the normalized dual energy iodine ratio best differentiates renal cell carcinoma subtypes. The next certificate of recognition is awarded to Dr. Bishwarup Chakrabarti of the Department of Pediatrics for his paper that was published in Epilepsia on the development of a novel outcome prediction score for pediatric convulsive status epilepticus. Another certificate of recognition to Dr. Rakesh Kumar for his paper that was published in the Journal of Clinical Nuclear Medicine on PET or CT-guided tissue sampling in patients with a failed or inconclusive CT-guided procedures. Now coming to the third prize, there are about 4 third prizes. The first one is to Professor Radhika Tandon for her paper published in PLOS ONE, The Association of Cataract and Sun Exposure in Geographically Diverse Populations of India. The next third prize goes to Professor Shefali Gulati for her paper that was published in JAMA Pediatrics on the efficacy of the ketogenic diet, a modified Atkins diet, and low glycemic index therapy diet among children with drug-resistant epilepsy. Another third prize to Dr. Rohit Bhatia, professor from the Department of Neurology, for his paper that was published in the Journal of the American Academy of Neurology, The Effect of Yoga as an Add-on Therapy in Migraine. It was a randomized clinical trial. And another third prize to Professor Nand Kumar from the Department of Psychiatry for his paper that was published in the Journal of Brain Stimulation. This was a randomized, double-blinded, sham-controlled trial of repetitive transcranial magnetic stimulation in the treatment of negative symptoms in schizophrenia. There are 2 second prizes in the clinical sciences. The first one goes to Professor Samir Bakshi for his paper that was published in the Journal of Clinical Oncology, olanzapine for prevention of vomiting in children and adolescents receiving highly emetogenic chemotherapy. And another second prize to Dr. Deepak Kumar Gupta, professor in the Department of Neurosurgery, for his publication in the Journal of Spine, The Development of Validation of Finite Element Analysis Model of Craniovertebral Junction. And the first prize in the clinical studies for the year 2020 goes to Dr. Somesh Gupta, professor in the Department of Dermatology and Venereology, for his paper published in the European Academy of Dermatology, a machine learning based decision support mobile phone application for diagnosis of common dermatological diseases. There's a third prize also awarded to Dr. Rupa Rajan for her paper in clinical sciences. This was published in the journal JAMA Neurology, the assessment of botulinum neurotoxin injection for dystonic hand tremor. Now coming to the basic sciences category, the certificates of recognition, the first one goes to Dr. Vijay L. Kumar, professor in the Department of Pharmacology, for the paper that was published in the Journal of Biochemical and Molecular Toxicology, Metformin and Silymarin afford protection in cyclosporin A-induced hepatorenal toxicity in rat by modulating the redox status and inflammation. The next certificate of recognition goes to Dr. Sachin Kumar, who's scientist in the Department of Medical Oncology, for the paper that was published in the Journal of Current Problems in Cancer. Identification of differentially expressed circulating serum microRNA for the diagnosis and prognosis of Indian small cell lung cancer patients. The next certificate of recognition goes to Professor Seema Kashyap in the Department of Ocular Pathology for her paper that was published in Cancer, Immunology and Immunotherapy, Clinical Relevance of Comparative Expression of Immune Checkpoint Markers with the Clinical Pathological Findings in Patients with Primary and Chemo-Reduced Retinoblastoma. Next, Dr. Lata Singh from the Department of Pediatrics for the paper that was published in Cancer, Immunology and Immunotherapy, the prognostic significance of PD-1 and PD-L1 expression in uveal melanoma, the correlation with tumor-infiltrating lymphocytes and clinical pathological parameters. The next certificate goes to Dr. Uma Sharma from the Department of NMR for the paper that was published in Journal of NMR in Biomedicine, Abnormalities in Metabolic Pathways in Celiac Disease, investigated by the metabolic profiling of small intestinal mucosa, blood, plasma, and urine by NMR spectroscopy. There are 2 third prizes. The first one goes to Dr. Bebaswata Nayak from the Department of Gastroenterology for the paper that was published in the Frontiers in Oncology, Prognostic and Therapeutic Potentials of Oncomirs Modulating the mTOR Pathways in Virus-Associated Hepatocellular Carcinoma. The next third prize goes to Professor Sujata Mohanty for her paper that was published in Journal of Stem Cells Translational Medicine, Tissue-Specific Mesenchymal Stem Cell Dependent Osteogenesis in Highly Porous Chitosan-Based Bone Analogues. There are 2 second prizes as well. The first one to Professor Alpana Sharma from the Department of Biochemistry for her paper that was published in RNA Biology, Targeting of Stromal Versican by MIR-144-199 inhibits multiple myeloma by down-regulating the STAT3 signaling. And the other second prize goes to Dr. Jyotirmoy Banerjee, Associate Professor in the Department of Biophysics, for the paper that was published in the Journal of Frontiers in Cellular Neuroscience, the GABA-A receptor-mediated epileptogenicity in focal cortical dysplasia depends on age at epilepsy onset. And the first prize goes to Professor Kalpana Luthra for her paper in Nature Communications, broadly neutralizing plasma antibodies effective against autologous circulating viruses in infants with multivariant HIV-1 infection.
A very good afternoon. Now coming on to Oncology Awards in Clinical Science category for the year 2020. The second prize goes to Dr. Dipali Jain, Additional Professor, Department of Pathology, for the work entitled Spectrum of Uncommon Compound Epidermal Growth Factor Receptor Mutations in Non-Small Cell Lung Carcinomas with Treatment Response and Outcome Analysis. And this was published in Journal of Lung Cancer. The second prize is also shared by Dr. Brusabanu Nayak, Additional Professor, Department of Urology, for the study entitled Role of Circulating Tumor Cells in Patients with Metastatic Clear Cell Renal Carcinoma. The study was published in Journal of Urologic Oncology, Seminars, and Original Investigations. And the first prize goes to Dr. C.S. Bal, Professor and Head, Department of Nuclear Medicine, for the study entitled Efficacy and Safety of 2,5-ACPSMA 627 Targeted Alpha Therapy in Metastatic Castration-Resistant Prostate Cancer Patients, published in Journal of Trianomics. Now coming on to Oncology Awards in Basic Sciences category for the year 2020. Second prize goes to Dr. Nevete Lomi, Associate Professor, RP Center, for the work entitled Prognostic Impact of HER2 protein and pink-eyed dilution protein in uveal melanoma, published in Journal of Human Cell. The second prize is also shared by Dr. Deepika Mishra, Additional Professor, Division of Oral Pathology and Microbiology, for the study entitled Novel Use of Fluorescein Dye in Detection of Oral Dysplasia and Oral Cancer, published in Journal of Photodiagnostics and Photodynamic Therapy. And the first prize goes to Dr. Ritu Gupta, Professor, Department of Laboratory Oncology Unit, IRCH, for the work entitled RNA Sequencing Profiling of Deregulated MicroRNAs in CLL and Their Impact on Clinical Outcome, published in Journal of Blood Cancer.
We now move on to the Basic Science Awards for the year 2021. The certificates of recognition go to Dr. Chandra Prakash, Associate Professor, Department of Medical Oncology, for his paper, Quercetin Impairs HUR-Driven Progression and Migration of Triple-Negative Breast Cancer, published in the Journal of Nutrition and Cancer. The next certificate of recognition goes to Dr. Idiatullah, Additional Professor, Department of Biophysics, for his publication in the Journal of Biochemica at Biophysica Acta in General Subjects. The paper is titled Curcumin Rescue P53Y220C in BXPC3 Pancreatic Adenocarcinoma Cell Line, Evidence based on Computational, Biophysical, and In Vivo Studies. The next certificate of recognition is awarded to Dr. Siddharth Kundu, Assistant Professor in the Department of Biochemistry, for his publication in Journal of Helion, Mathematical Modeling and Stochastic Stimulations suggest that low-affinity peptides can bisect MHC-1-mediated export of high-affinity peptides into early and late phases. The final certificate of appreciation goes to Dr. Seema Sood, Professor, Department of Microbiology, for a paper, Molecular Characterization of Decreased Susceptibility to ceftriaxone and Genotyping of Neisseria gonorrhoeae Isolates in New Delhi, India. This was published in the Journal of Diagnostic Microbiology and Infectious Diseases. There are two third prizes. The first one goes to, there are three third prizes. The first one goes to Dr. T.C. Nag, Professor, Department of Anatomy, for his publication titled Aging of the Human Choriocapillaris, Evidence that Early Pericyte Damage Can Trigger Endothelial Changes. This was published in the Journal of Experimental Eye Research. The next third prize goes to Dr. Jyotirmoy Banerjee, Associate Professor, Department of Biophysics, and to Dr. P. Sarat Chand, Professor, Department of Neurosurgery, for their paper titled Altered Hippocampal Kynurenine Pathway Metabolism Contributes to Hyperexcitability in Human Mesial Temporal Lobe Epilepsy, Hippocampal Sclerosis. This was published in the Journal of British pharmacology. The next third prize goes to Dr. He's not here.
Is Dr. Sharat Chandra here?
The next third prize goes to Dr. Sujata Mohanty, Professor, Department of Stem Cell Facility, for her publication, Comparative Evaluation of Anti-Fibrotic Effect of Tissue-Specific Mesenchymal Stem Cells Derived Extracellular Vesicles for the Amelioration of CCL4-Induced Chronic Liver Injury. This was published in the Journal of Stem Cell Reviews and Reports. The second prize goes to Dr. Rupesh Srivastav, Associate Professor, Department of Biotechnology, for his publication, Regulatory B Cells Inhibit Osteoclastogenesis and Play a Potential Role in Ameliorating Ovarectomy-Induced Bone Loss. This was published in the journal Frontiers of Immunology. The first prize is awarded to Dr. Ritu Gupta, Professor, Department of Laboratory Oncology, for her publication, Branching Clonal Evolution Patterns, Predominate Mutational Landscape in Multiple Myeloma, published in the Journal of the American Journal of Cancer Research.
Not clinical.
We are now going on to the clinical awards in the clinical category, and there are 5 certificates of appreciation. The first one goes to Dr. Roshan Bhatt, Associate Professor, Psychiatry, for his publication, Mobile Methadone Dispensing in Delhi, India, Implementation Research. This was published in the Journal of Bulletin of the World Health Organization.
Is Dr. Roshan here?
Is somebody receiving it on his behalf? The next certificate of appreciation goes to Dr. Vaishali Suri, Professor, Neuropathology, for her paper, Gene Expression-Based Profiling of Pleomorphic Xanthoastrocytoma, highlights 2 prognostic subgroups, published in the American Journal of Translational Research. The next certificate of appreciation goes to Dr. Madhusudan, Additional Professor, Department of Radiodiagnosis, for his publication, Non-Invasive Staging of Liver Fibrosis Using 5-Minute Delayed Dual Energy CT Comparison with US Elastography and Correlation with Histologic Findings, published in the Journal of Radiology. The next certificate of appreciation goes to Dr. Chandan Das, Additional Professor, Department of Radiodiagnosis and Interventional Radiology, for his publication, Role of Diffusion Tensor Imaging in the Evaluation of ulnar nerve involvement in leprosy. This was published in the British Journal of Radiology. The next certificate of appreciation goes to Dr. Professor Neelam Pushkar, Oculoplasty and Oncology Services, RP Center, for the publication Mucous Membrane Grafting for Lid Margin Pathologies and Stevens-Johnson Syndrome, a randomized comparative study, published in the Journal of Eye, London. There are multiple third prizes. The first third prize goes to Dr. Anita Chopra, Additional Professor, Department of Laboratory Oncology, for her publication, Prognostic Relevance of the Expression of EMP1, CASP1, and LRP3 genes in Pediatric B-lineage Acute Lymphoblastic Leukemia, published in the Journal of Frontiers in Oncology. The next third prize goes to Dr. Anup Saraya, Professor and Head, Department of Gastroenterology and Human Nutrition, for the publication, Effect of Vitamin D Supplementation on Vitamin D Levels and Bone Mineral Density in Patients with Cirrhosis, a randomized clinical trial, published in the American Journal of Gastroenterology. The next third prize goes to Dr. Asuri Krishna, Additional Professor, Department of Surgery, for the publication of prospective randomized comparison of sexual function and semen analysis following laparoscopic totally extraperitoneal and transabdominal preperitoneal inguinal hernia repair, published in the Journal of Surgical Endoscopy. The next third prize goes to Dr. C.S. Bal, Professor and Head, Department of Nuclear Medicine, for the publication Novel Fibroblast Activation Protein Inhibitor-Based Target Theranostics for Radioiodine Refractory Differentiated Thyroid Cancer Patients, a pilot study in the Journal of Thyroid. There are 2 second prizes. One goes to Dr. Juma Shankar, Additional Professor in the Department of Pediatrics, for a publication, Delayed Administration of Antibiotics Beyond the First Hour of Recognition is Associated with Increased Mortality Rates in Children with Sepsis, Severe Sepsis and Septic Shock, published in the Journal of Pediatrics. The other second prize goes to Dr. Vishal Gupta, Assistant Professor, Department of Dermatology and Venereology, for his publication, Evaluation of Platelet-Rich Plasma on Hair Regrowth and Lesional T-cell Cytokine Expression in Alopecia areata, a randomized observer-blinded placebo-controlled split-head pilot study, published in the Journal of the American Academy of Dermatology. The first prize in this category goes to Dr. Manish Soneja, Additional Professor, Department of Medicine, for the publication Effectiveness of an Inactivated Virus-Based SARS-CoV-2 Vaccine, BBV152, in India, a test-negative case-control study, published in the Lancet Infectious Diseases.
Now coming on to Oncology Awards, Basic Science category for the year 2021. Third prize goes to Dr. Alpana Sharma, Professor, Department of Biochemistry, for the work entitled Inhibition of microRNA 144-199 promotes myeloma pathogenesis via upregulation of varicecan and FACSTAT3 signaling, published in Journal of Molecular and Cellular Biochemistry. Second prize goes to Dr. Sameer Bakshi, Professor, Department of Medical Oncology, for the work entitled PGC1A-driven Enhanced Mitochondrial DNA Copy Number Predicts Outcome in Pediatric Acute Myeloid Leukemia, published in Journal of Mitochondrion. And the first prize goes to Dr. Jayant Kumar, Additional Professor, Department of Biochemistry, for the work entitled Diagnostic Utility of IGF-2BP1 and its Potential Targets in ETV6 Run K1 positive B-cell acute lymphoblastic leukemia, published in Journal of Frontiers in Oncology. Now coming on to the Oncology Awards Clinical Sciences category for the year 2021. The third prize goes to Dr. Sanjeev Kumar Gupta, Additional Professor, Laboratory Oncology, IRCH, for the work entitled Proposal and Clinical Application of Molecular Genetic Risk Scoring System, MR Plus, for BCR-ABL negative pediatric B-cell acute lymphoblastic leukemia patients, report from a single center, and this was published in Journal of Leukemia Research. Third prize is also shared by Dr. Dipali Jain, Additional Professor, Department of Pathology, for the work entitled EGFR Mutation Testing on Plasma and Urine Samples, a pilot study evaluating value of liquid biopsy in lung cancer diagnostics and management, published in Journal of Current Problems in Cancer. Second prize goes to Dr. Rakesh Kumar, Professor and Head, Diagnostic Nuclear Medicine Division, Department of Nuclear Medicine, for the study entitled Role of Dual Point 18F FDG PET-CT in Diagnosis of Pancreatic Adenocarcinoma in Patients and with and without Concomitant Chronic Pancreatitis, Comparison of CECT and EUS, published in Journal of Pancreatology. And now coming on to the first prize, it goes to Dr. Supriya Malik, Assistant Professor, Department of Radiation Oncology, for the study entitled A Phase 3 Randomized Trial of Transabdominal Ultrasound in Improving Application Quality and Dosimetry of Intracavitary Brachytherapy in Locally Advanced Cervical Cancer, published in Journal of Gynecologic Oncology.
So we also have the Shobha Seth Wenger Distinguished Chair in Gynecological Oncology. So Meera Seth Corpus Fund donated money to the AIIMS to establish a chair in the gynecological oncology. This prestigious chair is awarded to a senior faculty from AIIMS who has outstanding track record in the field of gynecologic oncology. The applications were evaluated by the Scientific Advisory Committee of the AIIMS Endowment Fund, and the awardee of the chair is expected to contribute to the development of the specialty concerned. Her involvement in research and patient care would help in capacity building and inspire younger faculty. And this is also associated with a grant of approximately 6 lakh rupees, which may be utilized for consumables, minor equipment, and manpower. And for this year, it is awarded to Professor Neeja Bhatla, who is the head of Department of Gynecology and Obstetrics. Congratulations to all the awardees. We will now be closing the inaugural function with the national anthem. But before the national anthem is played, just a reminder to everyone that we will reassemble at 2:30 for the inauguration of the posters, exhibitions, which is going to happen at 2:30 in the foyer of the auditorium. Everyone is invited for lunch. For the faculty and the dignitaries, it would be arranged in the second floor of the cafeteria building, and for everybody else, there is packed lunches available in the quadrangle associated adjacent to the auditorium. And now we will have the national anthem. I request you all to stand. जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात
नामे जागे तब शुभ आशीष मांगे, गाहे तब जय गाथा, जन गण मंगलदायक जय हे भारत भाग्य विधाता, जय हे जय हे जय हे जय 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 हे.
Thank you. Please assemble at 2:30.
like categories like the undergraduate, junior residency, that is MBBS. So they had to got their postal work here. People who are doing senior residency working on today's even fellowships, the one of the PN programs, somebody is the MD medicine, and subsequently they will do cardiology, and thereafter we have further fellowships. So if you look at, there's a diverse talent out there. We have people who are clinicians with basic science, people who have basic scientists, people who are in public health, public immunology, and clinicians. So besides people are collaborating. So there are another award this time, there's a public choice. So every resident, every student, and every faculty would come and just scan the code and give a...
Almost
3,000 people. 3,000.
So everybody has brought their friends, but even then, a lot of people see the other posters that you can see. At least they've come. Choosing the awardee will be very difficult.
No, so he said that you can register with your email. So obviously in the same category, you cannot keep on voting for the same person. Then we have a selfie award. How many of your friends are getting a selfie with them? This is the, we have a serious approach. Besides that, this is just to generate more enthusiasm so that at least their friends come and take the undergrads. We have undergraduate, we have oral platform presentations as well as posters. These are all the senior resident, all the people who would be senior residents. So this is just, I would say, a fraction. It's 350. If everybody go to... Hello. Oh!
Sorry, I didn't recognize.
Sitting at 120.
You're tired now. Yeah, lots of students, yes. Almost 400 students. Yes, for school. आज तक के सब speakers भी हुआ है. Otherwise, yesterday they had all the teams here. Yes,
Then another thing that the ma'am said, I understand is sustainable development. These are the goals for
United Projected some of our work. Using them as part of the test. Yes, without chemistry, microbiology can't carry on.
He's the research director. much time you can go here. We'll go from this side.
We're doing, we operate in brain through the endoscope and also through the microscope. And this is a endoscopic training. First we made this model in which there is a, you can place and you can change the direction of the blades. And this was the first hardware that we developed. And we applied for the patent. And then the other activities of Indonesia like cutting and then moving of a biopsy concept, moving of another instrument. And then we made some articulating arms in these. And in these, the endoscope, and we can actually plan out the complete trajectory of the surgeon. This is basically to train the surgeon and a number of iterations. Then this was our latest one, which whatever the surgeon. This is Dr. Raman, he is a postdoc from IIT Delhi. He is my student. Dr. Singh was here. He was our combined student. And you can see that he's picking this design. But there's another camera which is capturing. And there's a software which is based on artificial intelligence, deep learning regression, which estimates the rank loss and even how the ring is moving and all that. And the PhD student who's designed this is presenting the same right now in France actually. Abstract has been selected. This has got a US patent, US and Japanese patents. We presented yesterday with BIRAC. We got one commercial partner who will help us to commercialize. But our main plan is if this can be incorporated in most of the medical colleges. So I could realize that a lot of nursing students are very interested in a lot of medical students. Because I usually train surgeons. And you can actually know where the lateral ventricle, third ventricle is, and the chambers of the heart and things. How the heart chambers and all the see all the heart chambers. The good thing is that we've all made them all through CT scan, MRI, segmentation, and then 3D printed them, and the material has been directly in correspondence with the biomechanical properties for reiteration. So that is one. This was the COVID lung, which was, we were inspired by Dr. Mary. We made one for our teaching what exactly was the COVID. So these are our 3D printed instruments that we have designed. And these are some multifunction. Normally, our biopsy concepts are like that. And we were having this is actually a microscope. We operate also under the microscope. Then we realized that we do a lot of microsuturing. This microsuturing is on 4O. This is the thickness of the number increases and the suture size decreases. 8O, 9O, and 10. 10O you can actually hardly see with the eye. And they have to be, and then
so how small is the needle?
The needle
4 mm, 6 mm.
Then because the pressures of the animal ethics committee. When I was a resident 30 years ago, we used to do it on rat and guinea pigs. Because of the management, that was the course that. Then the animal ethics says the animal ethics you cannot kill so many rats and guinea pigs. So we ultimately had a structured program. We did it on elastic sheets, then tubes. But the main question was how to design this algorithm. What was your architecture?
So basically the idea is after these this is an effectual design. So we have designed the Immuno-Nano-Fluorescence architecture wherein the search and the other the things that Sir would have thought while giving that score. Now when a human is giving score, it is not that he will just see these 5 things and give a score.
So these are the
नमस्कार, आप सभी का इस द्वितीय सत्र में हार्दिक स्वागत है। व्याख्यान माला के इस सत्र में हम अपने चेयरपर्सन से निवेदन करते हैं कि वे कृपया मंच पर आएं। इस व्याख्यान माला के मुख्य वक्ता हैं डॉक्टर पी बलराम। हम आपसे भी निवेदन करते हैं कि आप कृपया मंच पर आने की कृपा करें। आप इंट्रोड्यूस करो, मैं सिविल।
I welcome Professor P. Balram. I welcome Dr. Randeep Guleria, Dr. Subrata Sinha, and Dr. Rama Chaudhary. I request Dr. Randeep Guleria to please introduce Professor P. Balram.
Good afternoon. I think we should start this session. We are right on time. It's indeed a great honor to have Professor P. Balram, Director of Indian Institute of Science, with us. His introduction is already there on the slide. You can see that he is the Chair Professor, National Center of Biological Science, Bangalore, and former Director of Indian Institute of Science, Bangalore. He has done a lot of pioneering research, and his research interests are basically around understanding of the structure and function of voltage-gated transmembrane peptide channels, design and synthesis of model peptides, particularly disulfides, and developing fluorescent probe methodology for examining membrane structure. So it's indeed an honor to have you here with us, sir. And today we're going to be talking on reflections of science in the age of coronavirus. I think, again, a very important topic, considering that the last 2.5 to 3 years have been very challenging for us because of this novel virus. And it has really affected science in many manners, both in terms of the type of research that was being done and how it also literally shut down some areas of research because the funding all shifted towards research on COVID. So without much ado, it will be indeed a pleasure to have you give your distinguished lecture on the reflections of science in the age of coronavirus.
Okay. Can you put on the first slide? It's here.
Yes.
Yeah. Thank you. Good afternoon. It's a real privilege for me to be with you on Research Day, and it's also a special privilege for me to be introduced by Dr. Guleria. I'm very grateful for this invitation to come and speak. The only thing I must confess is I've been retired for some time. And the question that I ask myself is, what can I say to you that is really new? Then I realized that I don't have much to say to you by way of new research. So I thought the one thing that I could do really is to reflect on science in the age of the coronavirus, because over the last 2 years, our lives and our thinking and every action of ours has been dominated by the coronavirus. On this slide, I have the foundational pillars of 2 of the disciplines in which I have worked over my career. One is chemistry and the other is biology. So what I have really is Mendeleev and his periodic table as one of the foundational pillars of chemistry, and Darwin and his ideas on natural selection and evolution, which are particularly relevant for the coronavirus, on the other side. In the middle, I have the various images of the coronavirus which have appeared in the literature. The coronavirus has, in fact, over the last 2 years, been an artist's delight. It's been a public health nightmare, but people have imagined what the coronavirus would look like with many, many very attractive depictions of the coronavirus. One might ask, does the coronavirus belong to chemistry or does it belong to biology? And what I have on this slide is what you will find in every biology textbook, the tree of life. The tree of life, of course, has bacteria, archaea, and eukarya. There's a last universal common ancestor right at the node, which nobody knows anything about. But viruses are, of course, excluded from the tree of life when we teach students. Viruses, the question really has been, are viruses living or are they simply crystalline chemicals? It turns out viruses spring to life only once they enter a host cell. And so their ability to do biochemistry is somewhat limited as compared to even the most primitive of bacteria. A word about chemistry. One of the best definitions of chemistry that I've read was by the biochemist Arthur Kornberg, the man who discovered DNA polymerase and fired the first shot of the biotechnology revolution. Kornberg in later life called chemistry the lingua franca of the medical and biological sciences. It is, of course, one of the sad facts of medical education that chemistry often does not find a very prominent place in the education of clinicians. You might also ask, if you're going to reflect on science, what is science? Most of you have a very well-defined profession. But if you are doing scientific research, you wonder, what are you studying? Science, of course, in its broadest terms, can be defined as the study of nature. Now, if you tell somebody that science is the study of nature, the next question that they ask you is, what is nature? And the best definition that I found of nature is in volume 1, the very first issue of the journal Nature, where the biologist Thomas Huxley was asked to write the editorial. Huxley did not write the editorial. Instead, what he did was he translated from the German an essay by the German poet von Goethe, in which he said, "Nature, we are surrounded and embraced by her, powerless to separate ourselves from her and powerless to penetrate beyond her." The coronavirus, if you ask me, was in fact truly a force of nature. I want to tell you a few things about the coronavirus, but I'm going to tell you the background. Like most ordinary people, I had never heard of the coronavirus until it appeared. I don't know how many of the physicians had heard about the coronavirus. I suspect not too many of them had heard about the coronavirus until it actually was upon us. And then, of course, in the last week of March of 2020, we had the lockdown. That was our first experience with lockdowns. And for the very first time in my life, I was locked up in this little room with a computer in front of me and nothing else to do. The Prime Minister had said that you can't venture out. I remember famously said, the Lakshman Rekha is the front door of your residence, so don't cross it. And so we stayed inside. So the questions in April, May of 2020 asked by observers innocent of virology were the following. What are coronaviruses? When did science discover the coronavirus? Who first isolated this class of viruses? And then, most importantly, what did science learn from the SARS and MERS episodes of the first decade of the 21st century? The coronavirus pandemic, COVID-19, is not new. It had already appeared before. We've seen the SARS-CoV-1 virus, we've seen the MERS virus, but yet we'd forgotten about them. Now, of course, I have a number of cartoons over here. The one I like best of the cartoons decorating this slide is the one at the bottom right, which of course tells you about banks. Before COVID, if you entered a bank with a mask, of course, you were thought of as a robber. And during COVID, of course, you can't enter the bank without a mask. Now, the age of the coronavirus is defined by these images. Very soon, by April of 2020, these had appeared on the internet, they'd appeared in the newspapers. Policemen in Secunderabad, auto rickshaw drivers in Chennai, chefs in Italy and in France, all of them All this while, many people were dying in Italy. Nevertheless, they found some dark humor in decorating Easter eggs with the coronavirus. So I searched for a definition of a virus. I should know one, having studied biology, but I thought, let me still look. And the best definition of a virus that I found was in this book called Aristotle to Zoos. It's subtitled A Philosophical Dictionary of Biology. It's authored by the English immunologist Peter Medawar, one of the best writers of science, and he calls a virus a piece of bad news wrapped up in protein. And sure enough, that's what it is. The nucleic acid is wrapped up with protein. We're all familiar with the spike protein. And of course, the only difference in the coronavirus is, in addition, it's got a phospholipid bilayer, so it's an enveloped virus. If the coronavirus is an enveloped virus, this is the kind of detailed picture we have of it. You can see the lipid bilayer in blue. You can see the coronaviral spike protein projecting outwards. And then, of course, inside you have those little blue circles, which are the interior proteins of the virus, which then hold the nucleic acid in place. Now, I thought I can't come to an audience of clinicians without quoting William Osler. Many, many years ago, William Osler said that soap and water and common sense are the best disinfectants. During the pandemic, we had lots of soap, reasonable amounts of water in most places, but the one thing which was in very scarce supply was common sense. Now, why should we study the coronavirus? There are two pieces of advice, and I show both of them on this slide. The first one is from the Godfather 2 movie. Al Pacino, as Michael Corleone, says, keep your friends close, but your enemies closer. Very early on in the pandemic, every government had declared the coronavirus as a public enemy, and therefore we must now learn everything there is to be learned about the coronavirus. But again, since I'm in an audience of clinicians, I must quote from Abraham Flexner. It's the Flexner report of the early part of the 20th century which revolutionized American medical education. And it is that revolution in American medical education, with its enormous emphasis on research, which is what you are actually celebrating today, it is that emphasis on research which has really driven the biomedical enterprise in the West over the last century. Flexner said, the plague is upon us. One cannot well study plagues until after they've run their course. So what I really like to suggest is that our understanding of the coronavirus is just beginning. The pandemic has been officially declared to have closed, and therefore we might worry, what have we learned about the coronavirus and what is it that we will learn in the future? But my first task, which I set myself in April of 2020, was to find out who discovered the coronavirus. By that time, the BBC News and the National Geographic had put the picture of the lady here on the extreme right, June Almeida, who took the first electron micrograph of the coronavirus, which I show you here. Now, if you look at that electron micrograph, you can see where the coronavirus actually obtained its name. You can see it looks like the corona with the spikes sort of sticking out here. The people who published this paper in 1967 were David Tyrrell and June Almeida. David Tyrrell, for those of you who haven't heard his name before in the context of virology, is the man who did some of the most fundamental work on the viruses which cause the common cold. And he did all this wonderful work in the 1950s and the 1960s, immediately following the Second World War. So I went and read this paper, and then I found this paper was a wonderful paper. It had these micrographs, but I wanted to know where did the sample actually come from. And so what I did was, when you read a paper, and this is for the students who are here, sometimes the best information in papers is buried in the figure legends and in the acknowledgements, and nowadays in more modern times in the supplementary material which nobody ever reads. Here I found that this picture was of a strain called 229E, and it then thanked Dr. D. Hambre for the 229E virus. So I made a few telephone calls and asked someone, people who were in virology, have you ever heard the name Dr. D. Hambre? They told me they never heard of this name, so I thought it's time for me to go back to the literature. So if you go back to the literature 1 year earlier in the Proceedings of the Society of Experimental Biology and Medicine, this paper appears, a new virus isolated from the human respiratory tract. What Dr. Hambre, and from the journal I realized that it was a lady, from the name Dorothy. So this made me even more curious because there were relatively few ladies in American science in the early 1960s. So I needed to find out what Dr. D. Hambre looked like, and if I were going to give a talk, would I be able to put her picture on the slide just as I've put the pictures of the other people. So now my research consists of trying to find a picture of Dr. D. Hambre. But you must read this paper, because this tells you what clinical research can be. This was done at the University of Chicago, and in the university infirmary, the people who would come in mostly would be young students with colds. So all you did was to take nasal secretions from them and then try to culture a virus from these secretions, and that is how she discovered the new virus, which is the coronavirus, which is called 229E. This is the first coronavirus to be demonstrated as a human pathogen in the 1966 paper. It's also, the size of the virus is determined as 89 millimicrons. Today we will say 89 nanometers. And in later years, it's actually been found to be associated with acute respiratory distress syndrome, which is what we talk about now in healthy adults. There are case reports which have appeared. She also established that the nucleic acid in these viruses was RNA by treating them with DNA synthesis inhibitors, and they didn't work, and therefore it was an RNA virus. David Tyrrell, using electron microscopy for the first time, also estimated the size. And I was really struck by this. They say 80 to 100 nanometers, and Dorothy Hambre here has said 89 nanometers. My respect for her experimental abilities then went up orders of magnitude. In 1968, this appeared. It's not a paper, it's not a letter, it's something written by the editor in response to a letter that he's received from a group of virologists, which includes D. Hambre, which said that a new class of viruses has been discovered, which are called the coronaviruses. But by the time the virologist named this formally, her name had disappeared. This made me even more curious. And so I then did what anybody should do nowadays, is to search every possible database in the literature. You can search the PubMed database, you can search the Google Scholar database, and what I found was that she was a very productive scientist, beginning in bacteriology in 1941, and had a publishing career which spanned 3 decades from 1941 to 1972. Yet she remained a completely unknown scientist, and there was no photograph available. So I won't tell you what I did. At this point, I began to search, and finally I found there were some papers which had been deposited in an archives in Arizona. And now, since I had nothing to do by the end of April of 2020, I wrote a letter to the curator, an email to the curator at the Arizona archives, asking, do they have this collection? And they were very happy to receive a letter from someone. They were also locked down. And one young lady at the archives then said, as soon as the lockdown is removed, I will go and search this box. We've never looked at it. She first did, and then she found that Dorothy Hambre was an extraordinary photographer who had made lots of pictures of the American Southwest. By this time, I knew she'd also written a book called Rhinoviruses, which I found, and therefore she was, in fact, an expert. Yet nobody knew anything about her. Finally, the archivists became so interested in this because they went even during the lockdown and found that they were photographs which they had never ever looked at, had not been developed. They did it and then produced the picture. So today, if we had, I don't show you all the pictures that I got, that's the discoverer of the coronavirus. And today, if I were to give a lecture to students, I would say these are the 3 discoverers of the coronavirus. A word here. A woman who has begun with a Ph.D. in the late 1930s, that means did her Ph.D. during the years of the Great Economic Depression in the United States. And she worked her way through the Second World War and eventually retired from the University of Chicago as a research associate. So this will tell you how far women have come in the last 60 years. What do you do with literature research of this kind? This is historical research. All that you can do is write it up. And where will you publish it? So I published it in Frontline, which is a magazine. And so they were happy, they took it. And then to my utter surprise, eventually I was interviewed by archivists in the United States on email, and my articles were reproduced in a newsletter in a small town in Colorado, which has a total population of 1,000. I'm very proud of this publication, simply because the only reason they picked it up was Dorothy Hamre, having found the coronavirus, decided that it was time to retire and went and settled in this town and died here. So then they did put on their newsletter that the discoverer of the coronavirus lived in our town. I was also asked the question, why were you doing all of this? Reason I did this was I had nothing else to do in the lockdown. I'm not a clinician, and I got COVID in the very first phase. I could not now prescribe chloroquine or azithromycin to anybody, so I could at least find who the discoverer of the coronavirus was. So in this interview, I did tell them that's where Uray is, and this is where Flagstaff, where the archives is. And I said it is a tribute to the power of the internet that the road from Uray to Flagstaff passes through Bangalore. It also seems to me that in some strange way, the virus has led us to its discoverer by turning the world upside down. So I do hope that she will in fact appear in some future textbook of virology. But I was in correspondence at this time with a man who wrote one of the first papers on the coronavirus as a young student in the early 1970s. That is Professor Ken McIntosh at Harvard. So I asked him by email, I was corresponding with him, and then he'd written an article in Pediatric Infectious Disease about recent advances in coronavirus discovery in 2005. So when all this ended, he said, he read my article, and then I rather liked this. He said, thank you so much. And he said he was ashamed that he didn't try to find out more about Dr. Hamre, although he did know her name, because that's where the original coronavirus sample really came from. But for those of you who are interested, you must read this book called The Cold Wars. It's about the wars against the common cold, which still afflicts all of us. Now that the COVID-19 has disappeared, we are back to catching normal colds. And David Terrell did all this work, and all done with volunteers, healthy volunteers, and you put nasal secretions from sick people up the noses of healthy people and ask the question, are they going to catch cold? If they catch cold, you harvested more secretions, and then you had more raw material from which to get viruses. But Terrell is very important because he wrote a chapter in this book on medical microbiology. And I brought this along particularly because I was in correspondence with Professor Raman. And he said in this book, coronaviruses cause acute, mild upper respiratory infection, and in brackets, common cold. With this, the virologists lost interest in coronaviruses. Those who fund research lost interest in coronaviruses. And that's why we've really suffered during the COVID-19 pandemic. Research should have been continued. In 2003, SARS surfaced, and I have only one slide on this. That pictures Carlo Urbani, probably a physician who really deserves a place in the history of medicine, because he actually identified that this was a new infectious disease, persuaded the government of Vietnam to impose the most extraordinary quarantine measures, controlled the disease at that time, but quickly contracted the disease and died a few weeks after he really identified the disease. 8,000 infections, 800 deaths. But it is this virus which has given us everything that we know about SARS-CoV-2. Much of the work on SARS-CoV-2 depends on the work that was done on SARS-CoV-1. This is a paper from the Journal of Virology which shows you the architecture. The architecture of these two viruses is very similar. The structures of the proteins involved had already been worked out. And this is why science proceeded very fast during the 2020, 2021, 2022 period. Today, many students think that this flood of papers in Nature, Science, Cell, and Lancet is because scientists have done something extraordinary. It is because they have worked on the back of the knowledge that was gained by studying SARS-CoV-1. Today, of course, the pandemic was officially declared in March of 2020. Already in 2020 in Science, the papers had appeared of the receptor binding domain of the spike protein complexed to the angiotensin converting enzyme receptor. So we have all these atomic level structures which have since appeared. As medical students, some of you might wonder, why do you need to spend so much time getting these atomic level structures? What is it that you learn from these? I can't tell you what we learned from these or what we hope to learn from these, which is a more accurate way of putting it. But I can tell you that the most important thing about atomic level structures is that they are made up of atoms. And atoms are important. Richard Feynman, the most quotable of physicists, in volume 1 of his famous lectures on physics at Caltech says, if in some cataclysm all of scientific knowledge were to be destroyed and only one sentence passed on to the next generation of creatures, what statement would contain the most information in the fewest words? He asked this question in the backdrop of the Cold War between America and the Soviet Union in the 1960s, because you're always worried about a nuclear catastrophe. Today we are once again worried about a nuclear catastrophe, and you might wonder what will happen if everything is destroyed. He says, I believe, he answers this question, it is the atomic hypothesis or the atomic fact or whatever you wish to call it, that all things are made up of atoms, etc., etc. Remember why chemistry is important. You are a bunch of chemicals, everything is around you a bunch of chemicals, and you cannot now find a single material around you which is not a chemical. So however much the Tata salt advertisement which we see night after night on the cricket matches, which promises you salt which is chemical free, but still has 80 minerals in it, is simply nonsense. So too are the sort of advertisements that are put out, for example, by Patanjali, which will say that I'll give you a phenyl which is now chemical free. Everything contains chemicals. Now a little bit towards the end of protein sequence variation by random mutation and natural selection. I'm sure Dr. Guleria is probably still getting questions on whether a new variant will now sweep through the population. So we can ask the question, what has the coronavirus done? It's been a free-running experiment. We could not have done such a wonderful experiment in a laboratory. Large populations have been infected. These populations have vastly different genetic profiles. Their immune responses are different. And the virus has evolved in this environment. I take you back in time to a paper written in 1970. This is written by the famous evolutionist John Maynard Smith. He called this paper, just a page in Nature, Natural Selection and the Concept of Protein Space. Then he talks about mutations. He takes the four-letter word, word, and says, let's make a single change. He gets war, another change, go. Another change, gone. Another change, gene. So if you've mutated word into gene, we've done this without changing. Every word has a meaning. And he says this is how protein sequences evolve. If mutations don't cause deleterious damage to the protein, it will survive and will find a function. And eventually mutations, either neutral mutations or functional mutations, will slowly get selected for. In biology, Darwin is everywhere. So natural selection is at the heart of the selection of coronaviral variants. This is an essay written by the Nobel Prize-winning scientist who worked on bacterial evolution in the test tube, Frances Arnold. She titled her essay, The Library of Maynard Smith, My Search for Meaning in the Protein Universe. In fact, what the coronavirus has been doing, it's been searching for meaning in response to environmental pressures. We create many environmental pressures. Vaccines are one of them. No vaccine is the same as some other vaccine. Your immune systems create a pressure, and it is in response to these pressures that variants are actually selected out. Now, the SARS-CoV-2 spike protein on which everybody concentrates has 1,273 amino acids. That is, it's a sentence of 1,273 letters. We can change each one of these the way Maynard Smith did. The number of possible sequences is 20 raised to 1273, which is astronomical. It's 10 raised to over 1600. It's a number that even the scamsters cannot imagine. It's such a large number, it's impossible. Now, it is this spike protein which actually acts as a Trojan horse, binds to the cell surface, welcomed into the cell by membrane fusion. So like the Trojan horse, it gets the enemy inside. Now, as the pandemic progressed, what can I do? I can only analyze sequences. Where do I get data? I can't get Indian data because it's a secret, and certainly not a retired professor. But of course, the National Center for Biological Information is available, and the Americans were depositing sequences, dates of sample collection, everything's given, and so there were over 6 lakh sequences. The major irony of this is that I've just come this morning after chairing a meeting of the Indian Biological Data Center where the INSACOG data has just been deposited. But it wasn't available at that time, and you can see with the help of 2 of my colleagues, one of whom's retired and the other one's a postdoc, we spent our time on computers, email, and the telephone discussing this and putting together data. Now, of course, these figures can be understood. I try to simplify them. I'm a protein chemist, and therefore I like to look in terms of single amino acid replacements. I don't like to look in terms of pangolin lineages, which are a set of abbreviations which I can't understand. So it's the pandemic was driven by a single mutation, D614G, an aspartic acid changing to glutamic acid at one position. The Wuhan virus mutated, adapted to human hosts, and afterwards the pandemic went. Along the x-axis here, days since January 1st of 2020. So just days. So if you look here, you will be coming here to 2021 at the end. Now, of course, if you look at the American data for variants, the American data is beautiful because the Americans had a free-flowing experiments. They were free with their data. They allowed everybody to mix. So people got all kinds of variants. And so you can see over time the rise of the alpha variant and its fall, the rise of the delta variant and its fall. And for someone trained in chemistry, they look almost like perfect titration curves. They look like you can actually find a crossover point. At this point, for example, you might ask the question, is there evidence for viral recombination in people who have been infected with both strains? But here I show you something which is important toward the end of my talk, a mutation at position 681. This is very close to what is called the furin cleavage site, and it's a proline going to histidine or a proline going to arginine. In the alpha variant, it was this. In the delta variant, it was the R. And now again in the Omicron variant, we are back to the H again. So one can study infection dynamics with this data over time. Most of you probably are familiar with this, so I will not actually belabor this point. But to just tell you that as time goes on, you can see this. You can see variants coming and going. And this, of course, is the dramatic rise of the Omicron mutant at this point. And you can see, now if you superpose this on infections and deaths and hospitalizations and so on, you will find that is much less when you come to the Omicron. So by this time, you've got mutations which are not doing too much damage to the host. I'll cross over this, but to tell you, this analysis of mutations sometimes is helpful in looking at antibody responses and in trying to see which antibody binds at which position. Has the virus evolved a mutation which will help it to evade the antibody response? But since I was going to reflect on science, and this is largely for the students who are getting into research now, science has transformed in a way over the last 50 years that is completely unimaginable. The Omicron mutant was discovered in South Africa in November 23rd of 2021. It was not published. It was tweeted. And the mutations were tweeted. And 2 days later, because I was following this, a correction was also tweeted on Twitter. So if you found the arginine, and it was originally listed as a lysine, you have to correct it to an arginine. You have to be following Twitter. But I want you to worry about two things. One is the date, November 23rd, November 25th. There are 30-point mutations, 2 deletions and 3 insertions. Till Omicron came, there were no insertions. Now the question, of course, is where did the insertion come from? And sure enough, by December, within a few weeks, there are already bioinformatics people now suggesting that this unique insertion has actually come from recombination with coronavirus 229E, Dorothy Hamray's original discovery. Now this means that there have been co-infections with the old coronavirus, the new coronavirus, and new variants have sprung up. There are others, of course, in the literature, because all this analysis is ongoing, which says that maybe it jumped from humans to mice, had these mutations, jumped back, and so on. But one doesn't really know. But the dates are important. November 23rd of 2021, on 27th of December 2021, a paper appears in Science, which is now a structure of the Omicron spike trimer, complex to the angiotensin converting receptor soluble domain, and an anti-Omicron antibody. So you can see that the pace at which science is being done in the West is unimaginable compared to the pace at which we can actually do anything. And this is actually a very worrying kind of trend. Before I conclude, I only want to draw your attention to one last fact. Where did the coronavirus come from? Did it come from bats to human beings through an intermediate animal host? Was it classical zoonotic transfer? Or was it a virus which was being investigated in a laboratory, which had now escaped from the laboratory environment and eventually adapted to a human host? This is now from a paper appearing in 2022, 19th of May, and I draw your attention to the name of Jeff Sachs. He's an economist. Lancet originally appointed him as the chairman of what they call the COVID force or whatever it is. He resigned and so on. But he's now pointed out something which had been pointed out much earlier, that the furin cleavage site, which is necessary for coronaviral membrane fusion with the host cell, is completely unique to SARS-CoV-2. None of the bat viruses so far have that. And it turns out that the mutations which are there, the furin cleavage site, I've listed it here. This is the mutation which drove the Delta and the Alpha and the Omicron sequences. And you can see this complete cluster of highly basic residues, arginine, is now known from experiment, enhances protease cleavage, which is necessary for membrane fusion. So where did this come from? We do not know, but the paper by Sachs now points out that this sequence corresponds to the subunits of an epithelial sodium channel. And people have been doing experiments where they've been trying to engineer viruses for what you call humanizing the coronavirus. And this has been done in American laboratories, and it has been done in Chinese laboratories. We don't know where the coronaviruses came from. I don't think we will ever know, because geopolitics and other factors will ensure that we never know. Now, it could have come from natural evolution or laboratory creation. And when I think about this, I'm reminded of a poem that I read when I was in school, where William Blake wondered about the lamb and the tiger. He noted that both the lamb and the tiger, and indeed all animals, have bilateral symmetry. The lamb looks beautiful, this tiger also looks beautiful, but the tiger also looks fearsome. And he says, what immortal hand or eye could frame thy fearful symmetry? Did he smile his work to see? Did he who made the lamb make thee? So he is a religious person, so he simply asks this question, was they created by divine intervention?
we might ask the same question about the coronavirus, because it also has the fearful symmetry that Blake imagined. Now, I worried about all of this when I was locked up in the lockdowns, and so I think I did all my research sitting at a desk. And John le Carre, who writes some of the best spy novels I've ever read, said survival is an infinite capacity for suspicion. And he also said a desk is a dangerous place from which to watch the world. A few months ago, I was asked to give a talk to a group of engineering students. And you know now that the only thing that is more difficult than talking to medical students is to talk to engineering students. And there was a listener, and she wrote me an email after this because she was on the management board of the institution which was conducting this online lecture. And she sent me a mail. She said she introduced herself as a poet. And she said that, I never knew all of this was there in science, but she said maybe there is poetry in chemistry or something like this. She sent me a mail a week later, and she said, go and read this blog. It's on oncology. And she drew my attention to something written by someone she knew, which I'm going to show you on the next slide, because I thought this is Research Day, and I should really show you something. This is by an oncologist at Columbia University, Azra Reza. She is the oncologist of Pakistani American origin, and she's written a book called The First Cell, and she's been a practicing oncologist for a long period of time. And then she says, despite all the research that has been done over the last 50 years or so, how she obsesses over what I've done wrong and what the field is doing wrong collectively. And she's written this book called The First Cell, which really asks this question, which really is the first cell which triggers a cancer? And this, of course, is akin to asking the question, which is the first cell which has led to life on Earth, for instance? We don't know. And so there are many questions which have evolutionary implications, which I think come out when you read something like this. Lastly, I must quote Lederberg, writing in the Journal of the American Medical Association in 1988, in the aftermath of the AIDS pandemic. And he wrote this article called, an editorial called Medical Science, Infectious Disease and the Unity of Humankind. And you must read him. He says, human intelligence, culture, and technology have left all other plant and animal species out of the competition. We also may legislate human behavior, but we have too many illusions that we can, by writ, govern the remaining vital kingdoms. The microbes that remain are competitors of last resort for dominion of the planet. The bacteria and viruses know nothing of national sovereignties. In that natural evolutionary competition, there is no guarantee that we will find ourselves the survivor. And this was Lederberg in 1988, and so that's not too long ago. He's not a man whom you can quite ignore, and therefore I think you must think about it a little bit. The one thing that the coronavirus has taught us is that two of the most important preoccupations in Indian life, one is religion and the other is politics, that both religion and politics retreated before a force of nature. Lastly, I must quote from an Indian, the first Director General of the Indian Council of Medical Research, very distinguished. I never had the opportunity to meet him, but I've always been told by very senior people that you must read things which he wrote, Dr. Pandit. And what Dr. Pandit, in a convocation address at King George's Medical College in Lucknow in 1956 said, are our medical students being educated in the true liberal tradition of a university? Are they exposed to the intellectual ferment and humanizing influence of a university education? Are we sending forth through the portals of our universities men and women whom we call physicians in the time-honored meaning of the word? Physicians who, in addition to recognizing accurately and treating effectively human ailments, also possess the imagination, resourcefulness, and ideas to deal with unexpected situations. COVID-19 presented us with an entirely unexpected situation. He asks a question: do our physicians have an intelligent understanding of man in relation to his environment? If I were asked today to paraphrase Dr. Pandit, I would only say, do our scientists have an intelligent understanding of man in relationship to his environment? Thank you very much for having me here, and the only acknowledgement that I wish to make is to the two institutions which have nurtured me throughout the course of my professional career, the Indian Institute of Science on the top left, and the institution which has provided me some shelter in retirement, the National Center for Biological Sciences. Thank you.
बहुत बहुत धन्यवाद महोदय आपके ज्ञानवर्धक और प्रेरणादायक उद्बोधन के लिए। मैं डॉक्टर रणदीप गुलेरिया से निवेदन करूंगा कि वे हमारे आज के मुख्य वक्ता महोदय का अभिनंदन करने की कृपा करें। बहुत बहुत धन्यवाद।
I now request Dr. Deepika to please felicitate Dr. Randeep Guleria.
बहुत बहुत धन्यवाद। अब हम आगे के सत्र में बढ़ते हैं पैनल डिस्कशन के लिए, और मैं डॉक्टर कमल से निवेदन करूंगा कि वे आएं और इसके विषय में हमें बताएं।
Thank you very much. A very good afternoon. We now proceed to the panel discussion. I request Professor Rakesh Agarwal sir, Director, JPL Puducherry, to please come on to the dais to moderate the session. We wish to convey our sincere gratitude to Professor Rakesh Agarwal sir for kindly agreeing to moderate this panel discussion at a very short notice. We requested him only 2 days ago, and he was very kind to spare time from his extremely busy schedule to travel to Delhi for this panel discussion. Thank you, sir. I also kindly invite onto the stage Dr. Rajesh Gokhale, Secretary, Department of Biotechnology. Our Honorable Director, Dr. M. Srinivas sir. Changing this. Okay. Yeah. So I request Dr. Rajesh Gokhale sir, Secretary, Department of Biotechnology, to kindly come onto the stage. Our Honorable Director, Dr. M. Srinivas sir. Dr. Chitra Sarkar, former Dean Research and former Head, Department of Pathology, AIIMS. Dr. Balram Bhargav, sir, Chief Cardiothoracic Center, AIIMS, and former Director General ICMR. Dr. Shishendu Mukherjee, Director BIRAC. Dr. Shinjini Bhatnagar, Professor of Eminence, THSTI. Unfortunately, due to some unavoidable circumstances, Dr. Rajiv Behel will not be able to join us today for the panel discussion.
Sorry for this. I think we'll settle down in a minute. Good afternoon. It's going to be very difficult for us to match the lecture that we have just gone through, and for me to match the person who was actually supposed to conduct this. It's very difficult to step into the shoes of Professor Vijay Raghavan, who was supposed to conduct this. But let me try my best. You already heard about the panelists. We have a virtual who's who, people who have done research, people who have done innovations, people who are responsible for promoting innovation, and what have you. So we have Dr. Gokhale, who's Secretary, Department of Biotechnology. We have Dr. M. Srinivas, Director of this institute. Professor Balram Bhargav, Chief of Cardiothoracic Center at All India Institute, and he was previously the DG of ICMR and Secretary, Department of Health Research. We have Professor Chitra Sarkar, a Professor of Pathology, previously Dean Research at All India Institute. Dr. Shishendu Mukherjee, who is a microbiologist for training but has done a lot of innovation and has worked for promotion of innovation and is currently the Director of Biotechnology Innovation Research Assistance Council, or BIRAC. And we have Professor Shinjini Bhatnagar, a Professor of Eminence, a pediatrician, a pediatrics researcher who's been in this institute for several years and is now at Transnational Health Science Institute. So she's also into translation from medical research to products. So for the audience, we have 60 minutes, and I do intend coming to you and giving you an opportunity to ask questions and give your opinions. So we'll in between try and do that. I'll try and keep it simple, something that we people in medicine can understand, and I'm not going to go into too much of technology terms. That's actually because partly I don't understand many of those terms. So this panel discussion is supposed to be on biomedical innovation and entrepreneurship, how to take it forward. So to my panelists, and maybe Dr. Mukherjee can tell us this. So we are discussing about these phrases called innovation, entrepreneurship. These words are somewhat foreign to, I'm sure, many of us in the audience, and possibly to some people on the stage also, who knows. Can Dr. Mukherjee, you define these words for us? Innovation, entrepreneurship. And I might tell you, we all in this room seem to understand research, and that's what this day is about. So how are these words different from research? And then maybe we'll be able to relate to it.
Thank you, sir. And
always a pleasure to be here. And
Can we do something about the mic, please? Yeah.
Yeah. Thank you, sir. And it's always a pleasure to listen to Professor Balram, and great lecture and very educative, and a lot of new things came to us we never knew. Sir, your question to me is, what is innovation and entrepreneurship? So I would like to quote from Albert Einstein's one quote from him, and I'll read it out to all of you. Imagination is more important than knowledge. To raise new questions, new possibilities, to regard old problems from new angle requires creative imagination and marks real advances in science. I think this quote from Albert Einstein talks about research leading to new things. So innovation can't happen without research. Innovation follows research, and innovation is something which comes out in a novel way, which has to have a basis of research. It has to be novel. It has to have something which really creates an impact. It creates sort of a tangible benefit to the society. So that is per se innovation per se, and innovation can't happen without research. Research is fundamental to innovation, which is something new. Now, the second part of your question was, what is entrepreneurship? Now, converting innovation into a business model or something which can be exploited to societal benefit is converting science into entrepreneurship is converting science innovation into a business model is entrepreneurship. So, but it is not so easy per se. Research leads to innovation, innovation leads to entrepreneurship. There is a long way. I try to define it very simply, but there is a long channel. We start research with proof of concept development, then that proof of concepts needs to be validated. Once that is validated, that is called innovation. Then that needs to be translated, and that translation we have to cross what we call a valley of death, which is a very challenging part of the innovation cycle. Once we cross the valley of death and the innovation is validated, then we can think of entrepreneurship. Converting that innovation into a business model is entrepreneurship.
Thank you. But I thought innovation was everywhere. We in India, why we in India? Everybody in the world today talks about jugaad. I thought that was innovation without research. Would you disagree that that's innovation also? There is no research basis to it. So I'm trying to go away from how you defined. There are so many instruments being made by jugaad. So in biomedicine, it may be different, but isn't that innovation?
Absolutely, that is innovation. That we call it incremental innovation. So incremental innovation, or you call jugaad innovation, that is also needs science driven. You can't do an incremental innovation without understanding of what innovation has already been done. So that is very important, and that needs to be built into the system. So jugaad is the word which we use it, but that needs to have a full scientific understanding of the process.
I agree with you. Anyway, in biomedicine, I don't think we can have jugaad. We have to have research. So I'll agree with you. So, Dr. Balram Bhargav, so you've seen it all in the last few years, what is happening in the country all around. So what do you think is the current status of innovation and entrepreneurship in medical institutions? And as a country, how are we doing on innovations and entrepreneurship in biomedicine?
I think, first of all, congratulations to Dr. Balram for an excellent lecture. And on the basis of that lecture and the COVID pandemic that we have witnessed, we saw how Dr. Balram took us all around the world. But ultimately, he landed at ICMR. He landed in India. So ultimately, he landed at ICMR. So he took us from USA to Cambridge.
But he ended at ICMR of 1950s. Please understand that.
Let me finish. Let me finish. So he took us from Chicago to Washington to Route 66, from Flagstaff to Colorado to Cambridge to Salisbury. But ultimately, he landed in the presence of this whole 100, 200 meters that we are. So innovation in India is happening here. So that is what is to be remembered, whether it's All India Institute or the Indian Council of
Is that true?
Medical
I
Research.
think we'll be doing a wrong thing by saying so.
No.
Let me finish. Only recently we had
let me finish the
Professor
90
Mahalanobis passing away.
Exactly.
And I think he did innovation more than in this 100-meter circle. So please understand that.
I'm coming to that. Dr. Mahalanobis and his oral rehydration therapy, which Dr. Bhan took forward further, and then again it was taken to the clinics through. So all those things, innovation is happening in India, and it has been happening in India. But the major problem that we have is that India works against India. We don't glorify our own innovation. Whatever innovation we are doing, we do not glorify it. The 50 slides that we saw, only one slide was dedicated to India. So I think it deserved more than one slide, and there could have been much more slides about India. And a lot of work has happened in India in innovation. So you're coming back to your question, where is India in innovation? I think the direction is right. We are in the right direction. Coming back to the point of Jugaad. Jugaad is a quick fix innovation which was there, but it has to be robust. It has to be cheap. So we as Indians are genetically endowed for Jugaad innovations. We as Indians are gifted to be cost effective, frugal, low, thinking of working in lesser resources, and we should not lose this gene by mutation or becoming expensive westernized.
Okay, let me go back to Shirshendu. Shirshendu, you also have some international perspective of innovation. How have we done as compared to other countries? So I'm trying to extend from the question that we had. And why do you think today more than ever we need to focus in India on innovation and entrepreneurship?
So in the last 10 years, we have done exceptionally well in the innovation ecosystem. And thanks to organizations like DBT, ICMR, other funding agencies, BIRAC, and things like that, which really catalyzed and enabled the innovation ecosystem in this country. And if you see, there is a model called 360-degree innovation model, which we talk about, is 60% is science, but 40% of the innovation cycle is apart from science, which we call it, the IP issues, the regulatory issues, the clinical development plan, and of course the funding. Funding has improved exceptionally well in last 10 years. We have, if you see the BIRAC model itself, we have created incubation ecosystem across the country where you don't have to bother about instruments availability and things like that. We have funding, we have mentorships have developed, and thanks to organizations like AIIMS, which have ignited clinical researchers into innovation ecosystem, the Stanford India Biodesign or Biodesign system which has been created, that has been really pushing innovation ecosystem in this country, and that we are matching into the global standards now. And we have, working with, but innovation needs to have teams. It is not a one thing. You need to have science talking to all the other aspects of the place. Science needs to talk, basic science needs to talk to clinicians, the regulators, the IP, the funders, the business plan development, and the exit strategy. Needs to be built together. That we call 360-degree innovation cycle. That, if it's successful, it can convert to an entrepreneurship model, and that can literally fly off. So we have done literally well. We have not reached that peak, but we are literally touching that. We are going very fast there.
Let me move to you, Dr. Gokhale. You've been associated with a lot of biomedical research labs. The CSIR labs, the DBT labs, in the public sector have done very well in terms of science. In the last few years, they are publishing in Science, Nature, Cell, what have you. And they've also done well, better than the medical institution in terms of innovation and entrepreneurship. What Dr. Balram was referring to in COVID, most of that happened in those biology labs rather than in medical institutions. What do you think? I know you have very little experience of medical institutions, but what do you think is the difference in that ecosystem versus the medical institutions? Because if we understand that, we could make a difference. Thank you.
So let me try and reframe this little bit actually for me to understand. So innovation, as you rightly said, is there in all phases and all facets actually. It's there in our day-to-day lives, what we do actually. So we can't take away that because that becomes a Jugaad innovation actually. However, what is important from an entrepreneurship, what innovation is how to drive revenue for a model for a company actually. I mean, the focus of the discussion should be very clear actually. If you're talking about entrepreneurship, it's about how to be able to make revenue from what you have. From whatever science or whatever technology or whatever kind of business model that you come up with, how can you create a revenue? And I can give you a simple example. If you look at just yesterday, I was reading in newspaper actually, the Meta India, which is a Facebook of thing, Facebook, which is nothing but Facebook actually in India, the Meta India, has made about 16,000 crores from his ad model in 1 year. Now, you just imagine this, this is what not even the investment in our science and technology ministry actually, total of put together actually. Whereas the innovation model in Facebook is rather amazing. Right? It does not really require research. It really required a sense of understanding of how systems and business models and propositions of value creation will be managed in a systemic way, such that it creates excitement and creates a panel of societies of doing it. So it's an innovation of a different kind, actually. My point is that we've never understood correctly when we want to say something. When we want to say entrepreneurship, then we start talking about doing research. Then we start about, and entrepreneurship is not about doing research. It's about having done the research. Do you have a mechanism to productize it such that it can create value? And that's almost absent in biomedical institutions or medical institutions as of today, primarily because that's not the way it is looked at, actually. There's a different way of looking at it. Luckily, what happens, CSIR, which has an I in it, which is for industry, they had a better connect for the industry, and they therefore started thinking about it and initiated a program about that around. And because they had a connect, they set up some of these activities, which has kind of led to all other scientific institutions to take up. So it's very, very important to understand when you talk about entrepreneurship, it's about making interesting business propositions and which is of value. And that's how I look. And if we do that, I think medical institutions will also start covering. In fact, the capacity is so large today to be able to look at from different point of view. I mean, I gave an example of data right now, right?
Yeah,
the data in scientific medical
institutions. That way in India, we have Flipkart, we have, Ola, we have so many. So those are very different fields. We are talking about medicine and biomedicine and science.
Now, I use Meta because it's not a consumer market proposition. It's actually just a social
science. So those things work in India. The question right now is, how do we take our science? See, we have taken our science ahead. So we do launch satellites for others. So we have tried to commercialize. But again, we haven't been able to be as what has happened, say, with France doing it or other countries doing it. So there is, even though we have the Gujaratis and we've always been traders, businessmen, that's what we have said. But somehow that hasn't happened with our science. So, is it just that we haven't started doing it, or is there something more than that?
No, I wouldn't consider the social platforms are not science actually, right? I mean, it's a science that has involved tremendous amount of IT management and technological interventions. So it is scientific, but it is actually understanding what the business models would be such that you can make value proposition. And that's what I'm saying in a biomedical environment, we need to convert ourselves into that. I mean, for example, the data that actually collect from patients, that itself is such a valuable repository, actually, if you structure it in a form and use it.
We'll come to that.
So it's a thought process and idea that I'm putting to it.
Okay. So, Shinjini, so we are saying from science, from having a finding, a discovery, we need a jump into making it a product. So how does one translate this finding, this thought of a product into a product? Should one straightaway go to industry? Does it, because, what do you think we need to do next?
So I'm going to talk more basic, being a researcher myself. And I think the kind of things we were able to innovate was sitting here at All India Institute of Medical Sciences, and Balram already talked about the ORS innovation that happened here under Dr. Mahan's mentorship. But so the main thing is that I'll take the lead question later. I'm just talking about you have a product. you need to validate it. The validation should happen in a very unbiased manner. It should not be the inventor validating what he or she has done, number one. Number two, you do need to get the industry on board very quickly, because as a scientific researcher, you've got the idea, you've done all the clinical research, and you've done it with rigor, you've done it with all the methodology that you should do. But finally, if you want to see it going in to the market, to the people who it's meant for, you need to get the industry on board very quickly. And I'm going to give two examples which happened here. One was, of course, the ORS, and I'm not heard. Okay. Yeah. That's right. Okay. So when we had the ORS story sorted, it took us some time to convince the industry to actually change the formulation which was already there. And so we had to bring the industry as well as the government on board and then be able to change it. And that happened by academic researchers in All India Institute of Medical Sciences. So it's not that you need innovation sitting somewhere outside. The second thing was for a diagnostic test for celiac disease. Again, that was a partnership with a scientist and then an industry on board very quickly. The prototype was prepared, was validated again by a group of people who we were not involved with and taken to the market. So I think that's very important, that even just answering simple questions, you need to get the validation done quickly and get the industry on board quickly. I mean, larger questions we can look at.
We'll come to later. So Dr. Srinivas, as a head of a major medical institution, what do you think are the major challenges that institutions such as this and its faculty face in terms of innovation and entrepreneurship? Do you think there are legal challenges? Do you think there are financial challenges or something else?
I guess the challenges are there, but of course we need to address those things, the legal, financial, and all that is being talked about. Basically, what I think is basically what is required at the moment is probably, we work in silos. That is the big challenge, I guess. If you ask me, I'll just tell you that, when we were in Ireland, I was a senior research fellow at one of the children's hospital. We were looking at one of those, bio implant for this thing. We had to connect with everybody, industry were coming to the hospital rather than we struggle here to go to the people. There was a association between our hospital and a veterinary hospital where everything was there. The people were, ready for us to go and do the experiments on the animals. And then the research cell used to take care of all the IPR issues, speaking to the industry and everything. See, as a doctor, we don't run around the people, I think that is which is probably lacking with us. If I do something like that in India, I think I'll be struggling a lot. I'll be going from place to place, people to people, and then spending time, as a surgeon or some people who are on the clinical side, it becomes very painful for us to run around for all this, If it is a basic scientist is okay, to the certain extent that they can think of doing it because the part of their training program, they have that energy and the, enthusiasm to go around about it. But as a clinician, it becomes difficult for us to run around for all these people and do it, But that's possible in India also, if you look into that, COVID opened up our eyes, a lot of collaborations and cutting these borders happened because of COVID. We did not have the BSL-3 lab in our medical college at ESIC Medical College Hospital. Within 15 days, through the help of the Ministry of Defense.
We are talking about innovation and entrepreneurship. So let's look at innovations that have happened during COVID in medical institutions such as this. Let's forget about National Institute of Virology. Let's forget about those institutions.
Now, I'm just coming to the collaboration. What is important for us is that if we are a clinician or something like that, we need to be hand-holding from the institutions. CSIR is there, ICMR is there, DST, DBT, all these people, cutting across those borders and going beyond the so-called medicine and the basic sciences. We had a collaboration with the Ministry of Defense. We never entered those institutions. We were not permitted to enter those institutions, but we were asked to go and look into them. And the lockdown was a very blessing in disguise, because they were also without work those days. So we could get a BSL-3 done in 15 days. And the same way with the Electronic Corporation of India Limited, they were also without any work, because they said that, it's a lockdown period, but the scientists were willing to work. Because they were, if you ask a scientist to sit at home, I think it's the most painful thing. Unless what Dr. Bhargava sir was saying, that, you think of literature, poetry, art, and other things, and then compile and then come to a beautiful oration which you give it here today. It is very difficult for them. I think we need to cut across these borders and go beyond the medicine, science, biochemistry, and all that. And then being in a government institution like this, I think we need a lot of collaboration between the institutions, especially the defense ministry institutions, which we had in Hyderabad. Enormous amount of things. Artificial intelligence, we got the support. The Department of Atomic Energy through the ECL gave it for the electronic health records and electronic physiological monitors recording. Whole lot of things. And IIT Delhi helped, IIT Hyderabad helped for us. University of Hyderabad helped for us. So a lot of things happen through this kind of thing. I think we need to collaborate both those things. And the institutions like ICMR, DST, DBT, and everybody, and beyond those ministry, they should come out and make that atmosphere congenial for the people to collaborate and go about it. COVID was an exception because everybody was helping to us. A non-COVID area, I don't think we are most welcome to those institutions to go about it. I think we have a challenges. I think we need to go. Maybe the legal issues are also there. Those things also we need it. At the end of the day, I think we need a university sort of campus where we have legal support. There's a technology support. So basically bridging the gap. AIIMS will combine with IIT, and then maybe an ISB and all that, AIIMS and all that. That I think we should be doing it.
You've been sitting patiently. I'm sorry for that. But what do people in the audience think? Anyone wishes to say, what do you think are the barriers? Sir, we'll start with younger people. There is a young man there. We'll come to the seniors later. Yes. See, he can't even believe it that he could be asked to come and speak. You raised your hand, right? Yeah, so come. That's part of the problem in our medical institutions. Go ahead, dear. Shout. At your age, you can speak and we can hear.
So, I'm Viswas Kumar Anand, and I'm intern here. What I feel is that, like, we get to ideas, we get our research works done, we get the data, but the IPR, the intellectual property rights, which we are neither trained nor we know much about that, and that's the first step from where we move towards converting our ideas into product or company or whatever entrepreneur or like going to the society. So that's the point I'm.
So the Dean, Dr. Subrat Sinha, please have a course on IPR for your interns. Thank you. Okay. Thank you, dear. You made your point. Yeah. Dr. Makharia.
Just 2 points. One, that mindset. That we as a researcher publish a paper, get impact factor, that's end of it. Go forward. Second, infrastructure. That innovation gets a deal further. There's infrastructure support to go further. It means more collaboration, more legal framework, the IPR issues, and then a support system. So my idea taken forward by industry or by other co-collaborators. That's why.
Is there any lady who wants to say anything? Otherwise, I'll go to Professor Mahapatra. Yes, ma'am. Please come. No, ma'am, the younger person behind you. Yeah, you first and then madam also. No problem. We have time. Please come. You people are tomorrow's India, not we.
Thank you very much, sir, for giving the opportunity. Now that we're talking about
This
IPR,
is one thing I think we need to get out of. Please, next person, don't spend time saying this. You got your opportunity because you raised your hand.
since we're talking about IPR, I've worked in the ICMR for long and I could not understand when I'm innovating something, is that my intellectual property or is it ICMR's DG's intellectual property?
Thank you. Ma'am, please come. We don't have the current DG ICMR here. We'll come to you, so you better think about the answer. No, I'm sure he knows the answer. That's not a problem. Okay.
I will take from the silos part.
the question the intern asked, we have a patent incubator cell here. Dr. Vel Pandian is doing it. The only thing is that we need to have internship electives in things like IPR.
Open that and don't keep that cell as a silo.
Entrepreneurship, medical humanities. When they are interns, we can give them that choice of electives that can raise the level.
Thank you. Professor Mahapatra, please. Sorry, sir, to keep you waiting, but then that's the respect we have for elders.
I think Dr. Bhan had a meeting about 15 years back in Ashoka Hotel, and he was talking about industry and academia involved medical science. What you do when you did research in COVID, we took quickly the Indian biotech, the COVID vaccine development. So here the science and the product are going hand in hand. How quickly a medicine takes 10 to 15 years, but in 6 months time our COVID vaccine is ready, because here the industry, scientist and researcher all came together. So there is no coordination. There is no. This was Dr. Bhan when he was in DBT. He had a meeting in Ashoka Hotel and 100 industry came. They say the academia think industries are bad, and industries are academics are in the White House. They cannot come out from Crystal Palace. This is the problem in India.
So we don't have silos, we have palaces, and each one of us lives in our own palace. So let me go before you, Dr. Bhargav, to Dr. Sarkar. She hasn't spoken. So Dr. Sarkar, she asked in the context of ICMR, but let's say this had been one of your faculty members who had got up and said, when I do something, I don't know IPR belongs to her or to the All India Institute. What happens in All India Institute?
We also have intellectual property rights has been sanctioned in the institute, and the intellectual property rights, if I can say, if it originates from the institute, we have it. We have a document on it, which is vetoed by the Ministry of Law, and all the rules and regulations regarding how the to whom the IP will belong, and how the royalty will be distributed, etc., in collaborations, in research projects from AIIMS are all worked out in that document. But I have a few issues which I would like to raise, which is in context with the young person who talked about IP. What is very important in any medical institute, and that's ecosystem is lacking, is I have to file a patent. The patent belongs to AIIMS. I want to file the patent. Now, patent filing itself is a long procedure and a financial implication. Whether first I do a national and then go on to the PCT filing, which is an international filing, which may go on to anywhere about 2.5 lakhs. Do we have a policy at the moment in medical institutions, unlike in engineering institutions, where a faculty with an innovation which belongs to AIIMS will be supported for patent filing? If you have it with an organization under a research project like DBT, then or with IIT, then they help you to file the patent. But if it's entirely a patent which belongs to, you are the innovator, it has originated from the funds of AIIMS, we do not immediately have a policy that there is financial help for the patent filing, which not only takes a huge amount, but also may need support over years as an annual fees. The second point is, I may be an innovator, I go with IIT and a company is opened, suppose I'm able to commercialize it. So who will commercialize? AIIMS is not one who sells. So do I need somebody to commercialize my IP when it reaches that stage? We have no such policies in place about commercialization. Third, can a doctor be a part of a company? Can I hold an equity in a company? I don't have such rules and regulations where because of conflict of interest, I can do it. And last but not the least, a very important thing for innovation for our clinicians, many of them are interested. Do we have any protected time to do the innovation? It is a huge lot of time that one takes. So a suggestion which was put forth at one point of time is engineering institutions encourage the innovation and entrepreneurship, and they build an ecosystem like IIT. We have an MOU with IIT. The IIT has the FITS, and the FITS does all the IP management, the industry management. We have started a bioincubator here. It is just about a year old. But to put in medical institutions, this IP management, the industry management in place, will possibly need collaboration with third parties, and that like BCIL, Kalam Institute, the NHSRC, and all these may be helpful to medical institutes where people do not have the time or still the ecosystem has not come in. But it's very important that with this Atmanirbhar Bharat and with our younger faculty going towards innovation and entrepreneurship, that we build this ecosystem of IP management and industry management within medical institutions so that our faculty get the benefit of the innovation and just not get a small royalty being a mentor or an innovator. So that is a very important thing.
Thank you. And let me add a little bit of my view here. So I come from a similar institution, which is smaller, of course, but the institution funds the initial process of filing patent, whether it comes out of the institute funds, and sometimes people are coming up with something where actually no funding has been involved. But yes, there are challenges, because I think somebody needs to evaluate what to file patent for, because what is also happening is people come up with ideas which do not appear to be commercially viable, or they are so small. It seems to be that I filed 2 patents, and that seems to be the idea, which is not really useful, as was said by Dr. Shishendu Mukherjee in the beginning. Ultimately, the whole purpose of patent is either to encash it or to make it available, license it to somebody so that it will be available to public for free, which is how for institutions such as us it should be. And I agree with you that, and then the faculty member wants it to be protected forever. If it has not been commercialized in 4 years, why should I, head of the institution, go on doing so? So I think there are challenges on both sides. We'll go to Dr. Bhargav and just keep your answer short to that lady's answer question.
I just want to say one point in relevance to this. See, idea does not mean an innovation. Ideas everyone has. Every night I sleep, I have 100 ideas. I jot them down many times, but they're just ideas. Unless you're able to translate it through the whole process by getting a prototype, getting a product, getting a animal studies, getting a clinical study, getting a validation, patent and all that. The challenges which you talked about, patent filing, Office of Technology Licensing, those are minuscule in today's environment. There is so much funding available from all agencies, and policies are also there in place from the ICMR also, where all medical colleges can work like IITs. That policy has been launched by the minister, so that is not a problem. Important thing is the philosophical part, that once your patent is then it is sold, how much will I get? That is the important question for everyone. Here there are 2 philosophical questions. Again, I want to bring it. There is called one side of the spectrum is Edisonian innovation, and the other side of the spectrum is Gandhian innovation. Edisonian innovation is said that anything that won't sell, I don't want to invent. So he just wants to make money. Whether it is useful to society or not useful to society, I want to make money. And the Gandhian innovation says that there is a thin line between need and greed. So you have to balance it out. For a country like India, it has to be a balance between a Gandhian and an Edisonian innovation, and somewhere a middle path where, so how much will one get, the profit? Now, where an ecosystem where innovation is just starting, they give 80%, 90% to the inventor. An ecosystem where innovation has matured, they give 10%, 20% to the innovator. Stanford is a classic example. 80% is taken by, 90% is taken by Stanford University, where innovation has matured. while IIT is the other end of the spectrum. 70% is given to the inventor because innovation ecosystem is developing. So this is a broad spectrum where anywhere institutions can fit in.
Okay. Yeah, sure. Yeah. Please go ahead.
Exactly
Go ahead.
as we stand, AIIMS has, on some of these issues, a very clear policy. When I do something, my group, my students, whosoever is, we are inventors. The assignee of the, it is a patent held by AIIMS, but we are classified as inventors. AIIMS gives the inventors, I think, 60% of any royalty share that you get, which you can internally divide up depending on your contributions or equally. I mean, this is something which is standard. This is there on, it is written. It's been used before. And so on this issue, there is not, I mean, if you are able to sort of commercialize a patent for a royalty, this will be the distribution. Okay. The second thing is, through NRDC, Indian patents are filed free, provided they go through that process of vetting. Some amount of vetting, I am sure, is required. You cannot just pop up and say, Bhai, you do this. So there is some minimal vetting which goes on. As far as PCT and foreign filings are concerned, as of now, we are a bit scared for the same reasons, that they are very expensive to do and maintain. And very often, then it becomes a commercial issue, whether your investment is worth it or not. And for that, as Dr. Bhargav said, there must be many agencies which would help you in that process. So just wanted to put these facts on.
Thank you. Okay, now I'll come to you, Dr. Deepak. Of the people on the dais, how many of you have experience of setting up a company or a startup? Good. Well, that's good. People in the front row, any one of you? People in the audience, anyone? Is there, so how was your experience? You want to tell us? Easy, difficult? Because we are all talking about, and I also want somebody who tried but could not do it. Is there anybody who tried and could not do it? I know it will take it's very difficult to raise your hand. There you are. You are the intern who talked, right? No, the other person. So why don't you tell us what your experience was? Both of you? Ma'am, younger people first. They are the startup generation. I agree. Madam, we'll get the mic to you. Don't worry. Please, so you first him, then you, and after that, madam. Madam, we'll get you the mic. Can we get a mic? Please go ahead. Start here.
Yeah, so I was a MBBS student in JIPMER, Rakesh's student, and we were involved with a lot of innovation with IITs as well as MVITs there. At that point of time, it was quite difficult, is what I felt, collaborating with other institutes, being a part of JIPMER. But when I passed out, I started my own startup, a digital health startup, collaborating with private colleges. What I realized is that it's quite easier when you are outside a government college to collaborate with institutes, number one. And when I came to AIIMS, I had to give up all my equity because we signed an NPA when you join junior residency. And so I left it. One thing I want to request is that how do you guys think that you can encourage collaborations in the government setup by students and residents? Because I have seen a lot of talented residents and students here who have a lot of ideas, who want to collaborate. But because of the red tapes of these government institutes, they are unable to do so.
So what's your name, dear?
Dr. Shubhrankar Datta. I'm a junior resident in radiology.
Radiology. Yes, dear. Your name and then tell us what your experience was. Without the mic, with the mic, you are equally audible, so you might as well shout slightly.
I think I am audible now.
Yeah, now.
Okay. I am Dr. Dikshit. I am a PG resident, Community Health Administration at National Institute of Health and Family Welfare. So I am currently in my final year. And in my second year, actually, I stumbled upon one idea and being very passionate about the intersection of healthcare and IT. So I came across this idea and I think I thought that it could create a societal impact as what we are talking about here. And during that time, I researched on whether I am allowed to actually have a startup and whether I am allowed to open a company while being on roles on my current role. So I came across the ICMR DHR policy on that, and in that it's mentioned that the medical doctor associated with any medical institution cannot be in executive role. He can be in non-executive role. So that was, you can say, first barrier, and very big barrier, because I cannot go against my degree to open a startup, and being in a non-executive role, it puts me in a position where I won't be able to take decisions on my own. I would be always at a back seat, and maybe people who are not even in the center of that whole idea which started this process would take the front seat and would guide me throughout the process. So I think this was the biggest barrier, and at some point I thought that I should finish up my course and then I should pursue.
Right. So Dr. Dikshit, I think a part of what you said also illustrates our medical complex. We don't say I got an idea. We say I stumbled upon an idea. We somehow are somewhat shy in saying that we thought up of an idea. So I think what you said is absolutely right. So we'll see what we can do. People at the back, there are seats in front. Please come and sit in front. There's no problem. Yes, ma'am. There are lots of seats in front. Madam, you can speak from there. That's why we gave you the mic.
To prove a point, sir.
Sure.
Yeah. So we had this, thank you. We had this corporate social responsibility being introduced, and we had been offered projects, and funding from a company in Bangalore to see that whatever research we do, if that can be translated, like, impaired glucose tolerance, how many of them will be converted to diabetes or not, just by making a cognitive attention by eye tracking paradigm. And we were offered a lot of funding for that. But then in the institute, the international collaboration, a very euphemistic term just for a talk between Delhi AIIMS and the Bangalore company, it just got lost. And then the powers that be shifted in the Bangalore company. I waited 2 years. So then the other people came in the management.
And then powers to be here can shift and things can keep happening that way, I suppose.
So, I mean, what I'm saying is that in spite of everything, the first tranche of money was also to be there. And for something like they just wanted to put a board where we are working that our name should be displayed. And the thing was, CSR is a donation, so you cannot ask even for a board. So
no, but even in any Indian small town, if you go to a dharmshala, every bench says this has been placed in the memory of parents so and so. So even donations come with that much of the name, I suppose.
I am a truthful person. I'm telling you right here, the narrative, that
let's
was
hear
the
from
barrier.
A successful experience. Dr. Gokhale. So can you tell us truthfully what are the challenges you faced? I'm sure even being in your position, you face challenges. And if you have any advice for those young men there. And women.
Men and women. So, no, I think having heard some of the discussions that I've heard, I think what is very important and critical is to have a program on entrepreneurship actually. You need to have a structured program on entrepreneurship because there are very small aspects of things. For example, whether you should be, as he raised the point of non-executive director, executive directors, the liabilities that come with the non-executive director, non-executive directors actually, because they're all very different. And when you run a company, it's not about innovation only. It's about how well you run a business model actually. And I again re-emphasize the fact that until and unless we actually understand that making money is not a bad thing in life, which is what I was just talking to Balram actually, and we have this common thing. First in India, we think, isne to paisa bana liya karke. Right? That's the most amazing part of our life. We first think that way actually. So anybody who is going successful or making money is not an ideal Gandhian person or whatever maybe we called to look at it actually. So there are a lot of nitty-gritties that one has to understand when you make a company. It's about how to raise capital, I always tell, I've been told by one of the venture guy, the VC fellow, he said, what is it about making a company? It's about putting your hand in somebody else's pocket, taking out money from his pocket, putting it in your pocket, and still he is happy with you, actually. And that's a very different model that you can do this. So therefore, it's very important that it's not just ideas that sell, but it's ideas that have financial success stories, actually, and how you can spin it around that such that you can at least, create dreams that are very, very successful. But it requires understanding details. And I again tell, it's very important to have a systematic coursework if one wants to do an entrepreneurship. It's non-trivial problem. Believe me.
I agree. But the problem you talked about, entrepreneurship being putting your hand into somebody's pocket, getting money out and still making that other person happy. Many of us actually do that entrepreneurship, or people who pass out of the portals of this institution. They set up clinics. They do exactly what you talked about, except that they don't develop products as we are looking at it today.
But that is a part of entrepreneurship only.
So
right?
people do have entrepreneurship. There are people whom I have known who have set up, a person 2 years senior to me, center of site or whatever, center for site, whatever he has. That's entrepreneurship actually, and many others that I know. But we are here talking about slightly different entrepreneurship. So how many startups or companies is the All India Institute faculty a part of? Maybe the Dean Research can tell us, or the ex-Dean Research can tell us.
I don't think we are a part of a startup.
Or how many innovations have you been able to give to the companies?
I don't have the number, but we do have in the research section a kind of a corpus of which started off at about 40-50 lakhs, which is a part of that 40% AIIMS keeps back. Now, for so many years and this is since accounts have been, they were being accounted separate. We started accounting them separately. I think Vineet will know better about the figures if he's around. But otherwise, people have been, I can tell you incidents and of people being able to actually sell their patent share. That has happened. If you ask me how many, I'm not sure. It's not a very frequent phenomenon. That much I can assure. It's a rare phenomenon.
Okay, so, we've heard a lot about, sorry, please go ahead.
About a year back, the AIIMS has got a BIRAC grant under which we have a Center of Medical Innovation and Entrepreneurship set up here. It encourages incubators to come in, and about 9 startups have started within the course of about one and a half years.
Mm-hmm.
So these are being nurtured within AIIMS, and we have a whole sort of an administrative team under the research section looking at this center. So we hope that the ecosystem with this center will take it forward in the next few years as a model.
So that's where our radiology resident needs to go. The gentleman who was in black shirt and talked about red tape. Yeah. You're aware of it. Okay, good. So at least the center is advertising itself well. Now we heard about making money and money being bad. Why does that happen here with us? Because we train to be doctors. And we had this, whichever version of oath you take, it talks about not bothering about money. And other professionals don't take an oath. We somehow think that, our ethics are somewhat different. We somehow have conflict of interest in many things. Do people on the dais, anyone? Do you think that's a limitation in our getting into this entrepreneurship? Or people in the audience who may think so? First people on the dais. Dr. Deepak, please.
In recent years, there have been a very positive development at AIIMS. It's a long-term process. I've gone through 30 years of having an idea. My idea was used by the NASA. I couldn't do anything. The idea is still there. But the recent development here are taking good place. Let's do the micro-analysis. I have done an innovation, I have a product, a process. Who will do a validation for me? I don't find a person.
Other institutions around, other people?
Why should they take it?
Why not? So that's what we need to, that's what I'm trying to say, that we'll have to...
No, I think that's what the problem. Second, the industry. The industry situated very far. It should come to doorstep. By the time we go to the industry, it is already, the steam is already lost. That's what why fit it very useful, because we want to fit and we are going to end our NRDC. We still find NRDC, one product is lying in pipeline. The problem is the distance between medical sciences and industry is much, much far. And that's but that's happening.
Maybe it will happen in Jhajjar. If industry is set up in that place, you'll be closer to them.
I think
in Delhi, there is no space
maybe
for
this
industry.
biotech innovation has come up. It is bringing industry to us now. I think the first is permission to go to a private. Like if I want to apply to go to the private university, I said no. Private university like Manipal or even MBBS, no. Talk about industry? No.
But you are the leaders of the institution, dear.
Now this is a policy. Now this is what the people are sitting over here. Let's make a policy. That
I have lost the director AIIMS here, but the ex-director is there. The deans are all there. You are the policy makers. Who stops you?
Okay, so we'll do that. It's happening now.
Yeah. Okay, thank you. Shinjini, you wanted to say something. I saw you raise your hands.
To discuss with our younger colleagues here what are the solutions. I think we've been kind of going on and on about what the problems are. And honestly, I think the solutions are not all that unsurmountable. I mean, I know Alok knows, he's been looking after the equivalent of a bioness that we have at THSTI and at the cluster really. And I mean, this is, so I just want to take a minute to explain what and how the Institute innovation can really, so at THS, at the cluster, at the bioscience cluster, which has Regional Center for Biotechnology and THSTI and funded by BIRAC and with Rajesh's support there, we have what we call the BioNest, and which is exactly what AIIMS is starting now. We went through a lot of teething problems in the beginning, but it creates a platform of what you are saying, that you have an idea, you take that idea with a collaborator or without a collaborator. And Subrat, we have AIIMS TV, some of the TB group people there as part of startups in those things. So you have a collaborator, you go to them with the idea, you set up a platform for validation. They help you with getting an industry or people help to invest in your enterprise. There is an industry on board through that whole platform, and you take it cannot happen in isolation, and it cannot happen with individuals wanting to. So I think you people have a beautiful setup now, which can be a great platform for the younger people to take it forward. And the policies that DBT has, CSIR has, ICMR has that come out, everything is listed. Who is the IP owner? Who is the inventor? Where do you license? It's all clear. You don't really need to reinvent them.
Yeah, thank you. So, Dr. Shashendu, we have only a few minutes.
Yeah, I'll take only 2 minutes.
Okay, go ahead.
Firstly,
you say your piece, then I'll have a question.
Yeah, on the IP issue, I think a lot of people talked about it. It's a challenge. It's absolutely not a challenge.
Yeah.
What IP we should understand, firstly, when you begin to file IP, we need to do a potential commercial valuation of your innovation, which understands the prior art and then whether it is worth patenting, as Sir said. Now, to file a patent, initially it takes only 10,000 rupees. 4,000 is the patent office fee and 6,000 is the attorney fees. We at the BIRAC system have created facility for our funded projects to enable that to file it. You have 12 months to have as a provisional. Within that, we aggressively look for commercial partners. If you have it, extend it to the PCT phase. That takes around 3 lakh rupees, which is again funded. And then you have to have aggressively a commercial partner. If you don't have it within 12 and these 18 months of PCT, you should abandon your patent. It is not worth taking it forward. That is the first thing. But we have facility, as Madam said, we have a BioNest here. We have 75 such BioNest across the country, which is supporting it. We have established through BIRAC NBM facility around seven regional technology transfer offices, which is aggressively talking to all the innovators in all places to help you to transfer your connect with your industry and help you to transfer the technology. That is the second thing. Finally, on the industry built up on the research, we have programs where you can jointly support funding application, taking industry into your, you can support joint applications, where you are funded for your science and innovation, and industry is also funded for its scale up. I think we should all take benefit of all these things and work together. But ultimately, we have to build a team. It is not one thing, 360-degree team we need to have in place to take innovation to entrepreneurship. Thank you.
So, Dr. Shishendu, we heard that a lot of innovation happens in these 1 kilometer radius. But we also heard that Jugaad is everywhere in the country, so I'm sure. And you talked about there being centers elsewhere. So how many of those are in medical institutions outside Delhi? Or in, less endowed institutions within Delhi, for that matter?
What we look at as a cluster-based innovation model. It is not that it is housed within this system. For example, we have one in, say, the Regional Technology Transfer Office, IIT Delhi, and this is called the FIT, and its area is all around Delhi and some part of the northern region. So they need to go around and sit with the innovators there, understand their innovation, help them and handhold them to filing the IP. Similarly, we have, say, in west side in Pune. So they cover a belt there, so they are talking to all the medical schools, the engineering schools there, bringing in the collaborations together, help them to raise funding. So that is the enabling mechanism is there to build all, and I think we encourage all of them to really go out, do a strong outreach, connect with them to take it forward.
From the audience, yes, the gentleman there and then the person at the back behind him. Anybody else in the audience has any questions? Okay, we'll come to you. Anyone else? Okay, 4. Okay.
Sir, I'm Ashish
Otherwise,
Suri.
they'll throw us out after that. Yeah.
I'm Ashish Suri. I'm a neurosurgery professor here. I'm also the adjunct professor in IIT Delhi last 14 years. There are a couple of issues I would like to bring about. Number one, we have to understand that Indian industry is a service industry. It is not a manufacturing industry. Maximum, it is a service provider software industry. It is not a manufacturing industry. The change is coming, but I think it will take around another 10 to 20 years to become like Taiwan or like China or like places in which manufacturing is an important component. And we cannot do, the present medical industry is 90% imported, even in this hospital, and even in my super specialties in neurosurgery, 99% imported. We are relying on completely on US, Germany, and Japan. To completely replace that by an Indian manufacturing is creating a chaos that we are already witnessing. The GEM and the bypassing of the permissions for the global tenders, we're witnessing it every day. That is a very big challenge that we have. Medical production is not the same as industrial production for a bridge or a building or maybe a sanitary products or anything else. Second, we have to understand the difference between innovations and inventions. I think Dr. Bhargav beautifully brought it out. Innovations do not require patents. They require rapid production and hitting the market and before it changes. Inventions require patents for protection. And I think that is where institutions like AIIMS, IITs and many others have to protect the inventors. That is important because inventions take a long time. They take probably a series of 2 or 3 PhDs in series. Third thing we have to understand, and that is for the younger people who think that yes, you can study and you can become a professor, and then you can also have a company. We have to understand the difference between academics and entrepreneurship. 9 out of 10 startups fail. Please, this is the truth. The world over. No one has mentioned it till the last 1 hour. Out of that which one succeeds, 9 out of 10 success do not see the light of 5 years. That is also the truth world over. And this includes Netflix and it includes many other software programs as well. And plus, commercialization isn't simple. It takes a lot of high risk. I am here, I'm a professor. I'm a walking, talking company and many of the professors here are. But we took the challenge of remaining here, do academics, write papers, do some patents. But yes, we could have been doing the same thing like you mentioned. But running a company is a big risk. You can only succeed in running a company if your tail is on fire. Sir. If you lose everything and you have to make it succeed. That's the only way you can run a company. You cannot be in 2 boats and sail together. I'm very sorry.
Thank you, Dr. Ashish. I think that's a very important perspective. But then we can set our tails on fire. That's not a problem. When you have, for me, when there is a patient having a GI bleed, my tail is on fire when I'm doing an endoscopy, right? That's true for you in neurosurgery all the time. So that's doable. Yes, dear.
Good evening, everyone. My name is Chaitanya. I am we started last year with something called the Entrepreneurship Development Club. That is basically our undergraduate initiative to promote entrepreneurship amongst ourselves, since there is no formal program, and that's a need, like sir mentioned. And I wanted to share one or two experiences regarding it. The first was regarding the public perception. So we tried to inculcate financial literacy into undergraduates, and we wanted to do something related to stock education and, you know, having some leagues related to that. So one comment that we got was that this will look bad on AIIMS because it will seem like doctors want to start earning money. AIIMS के डॉक्टरों को पैसा कमाना है, and they're no more concerned about their education. That was a major concern in terms of public perception and how we fear that might happen.
But when you have pulse, nobody thinks that what people think about, right? Isn't it? I hear Pulse has changed a lot, because my students come with a lot of requests that they want to do things which are done at Pulse. And when we say that, those don't look nice on doctors. But that's okay.
It's fine, no comments. The second thing I wanted to add was that if you look, 9 undergraduate posters were presented in the research day. So, and a part of that is credits to the research section for starting the UG mentorship program, where a 2 lakh rupee research grant is given for research projects. In a similar manner, like Dr. Subrata mentioned, that there's a 70-30 distribution between inventor and AIIMS for any patents that come out of here. And the policy was also very retrogressive till 2017, where it explicitly stated that any invention done at AIIMS, with or without the support of AIIMS, is the sole intellectual property of AIIMS. So, but there was a revenue sharing agreement of 30-70. So I feel whatever 30% that AIIMS is getting, it should reinvest into entrepreneurship initiatives, and it can develop a parallel program similar to the UG mentorship program, where it can be a UG entrepreneurship program or even a residence entrepreneurship program. To that extent.
Dr. Subrat Sinha.
This 40-60 has been around for a long time, actually. Not started in 70.
Okay.
Right. Sorry.
Okay. Thank you. There was a gentleman here, and then we have somebody at the back, and then I think they'll drive us out if we don't stop anyway. So that will be okay.
Yeah, good evening, sir. It's a very interesting session. I am Dr. A. Chaudhary from...
Thank you.
I'm an additional professor in cardiac anesthesia department here in AIIMS. My question pertains to our our hands are tied because we have non-practicing allowance at one end. On the other hand, there is a talk of moonlighting these days, that being a 24-hour employee of this institution or any institution, and you are like catering your services to others. So in this light, how this panel discussion explains or assuage our...
Anyone wants to answer this question? Moonlighting in the IT industry versus this? You want to try, say something, Dr. Gokhale, on this?
I think a good question, but moonlighting is also not fraught with less debate even in the IT industry. It's facing a lot of flack even in the IT industry. And therefore, the next stage will be whether it will be considered with as a taboo in our industry as well, if you want to call this as an industry.
See, moonlighting, they are doing the same kind of work that they are doing for their primary employer. And the question is there a conflict of interest in that? And to me, they don't even get an NPA. You, for not doing moonlighting, you get NPA. I think the issue is of doing things other than what is your primary job that you are paid for. If you are, so we are we'll have to try and distinguish between that. And that's my question, what the student there said. If you develop something with or without the help of the institution where you are, if I can make a better cycle stand, which holds better, if I work here, does that mean that, that innovation also, the right belongs to the institute? So we'll have to try and distinguish between, how much related it is to my work. And that's what I think that we'll have to keep in mind. There was somebody at the back who raised hand.
Can I say something?
Yeah, sure.
Conflict? So, I wanted to bring this point that you raised a couple of times regarding conflict of interest. This is always going to happen when you're going to have an entrepreneurship. For example, Dr. Suri is not going to do entrepreneurship in non-neurology area because that's an area that you know. So I think we should not call that conflict of interest. We have to find mechanisms to mitigate conflict of interest actually. And in fact, find ways that it's a win-win for all of them. That's one of the major factors that we are all not progressing in our entrepreneurship programs and policies, because constantly we talk about conflict of interest. Arey Baba, that's an area I know. So people are going to fund me only in the area in which you know well actually, right? And so that's the critical part. The second I would request, looks like, because looks like medical sciences has coming up later on in this game. I think you've missed Startup 1.0. Believe me, it's not been fabulous in India in terms of making value chain and money. I think AIIMS should adopt Startup 2.0 policy, which is far better and interesting. And I can help you out drafting some of the points which I have in my mind actually, which is well ahead of itself.
So why I was raising this conflict of interest was exactly to hear what you said now. That's why I was raising that issue again and again, that conflicts of interest are everywhere, but they can be managed and the institutions will have to find mechanisms to manage. The lady there who wanted to say something, somebody there?
So I have a question.
Yeah, you have a mic. Okay.
So I am actually faculty in radiology and I work a lot in artificial intelligence. So my question really is, I'm not going to go as far as entrepreneurship, but it's a little bit behind that. So how can we, since there are so many policymakers here, make it easier for us to just collaborate with industry? So for example, if I was to collaborate with a Google or an HP or NVIDIA, there is a lot of difficulty in terms of, whether I'm allowed to share data with them, how do I go about it? Even in terms of grants, if I was to write a grant with an IIT, I'm likely to get it. But if I was to write with a Google, I'll probably not get it as far as most grant agencies are concerned. So how can we be helped out over there? Because most of the talent actually is in industry when it comes to collaborators, at least as far as AI is concerned. All the NLP, all the DL guys are sitting in industry, are sitting in Google or in Facebook. So how do we sort of come around?
So you want to take this, Dr. Mukherjee? I actually wanted to go to international collaborations, but it's come up there. Yeah.
A little bit answered this question.
Your mic is not working.
We in BIRAC actually encourage industry collaboration, and especially clinician with the industry collaboration. Literally is a very very encouraged proposition. So there are programs, there are initiatives where you can literally talk to the industry, jointly apply. Some of the milestones you do, some of the milestones industry do, and it's a joint collaborative proposal. And that is always encouraged when we have schemes and proposals coming out on a regular basis which supports this, and especially in artificial intelligence and robotics and everything, it's much, much encouraged.
One thing that I have learned in life. Okay, please go ahead.
See, listen, we are doing clinical trials. We are doing industry-funded clinical trials. Now, if you put in the same model, that model is there. So for you to collaborate or work using company money according to our rules is possible. Now comes the next issue is how, with clinical trials you are validating and all. Then comes the next issue of how you share equity, how you share profits. That is the gray area. But if for pure academic interest with IP written out, it is already happening.
So, but not as far as a clinical trial, maybe to co-develop something like,
So I'm saying the same models can be used for co-development in which perhaps I feel it is still possible to get a share of IP. The point is whether you will be holding equity in the company, that may not be. Share of IP, perhaps.
So I'm aware of your work. So we have had some people wanting to work with Google. And I can share with you what happened in the committee. There were people, including some comments from me, saying that when you share data with them, we all know what Google is. You search for a ticket from here to Bangalore and next 20 days you get, right? So that is what the issue was. And they said, no, we are going to have something on that device. They will not be able to find who the person is. Identifying data won't be there. And the questions was that they know which house you are living in once you have. So I think, but finally, I think we were able to, in a small group, able to resolve these problems. And the resolution was, Google anyway knows where you are, right? It's not going to be participating in this study. The problem was the investigator was intentionally trying to put in things to show as if data were not going to go to Google. And we had to say, why don't you say it up front that they would have this data, but they have this anyway, and why we do this? So I'm just trying to give you an alternative way. So it does happen in a committee, but somebody had to break the ice by saying, do you think without that Google doesn't know your data? So that was it. So one thing I've learned in life, in any session, a woman has to have the last word. And Shinjini has raised her hand, so I have to allow her to do that.
Okay.
Shinjini and I go a long way. I reminded her yesterday night that we go back to 1984 when we were junior residents together in C2 ward. So go ahead. From that time, she gets precedence.
Woman in our BioNest, 40% are women founders. So that's for the women. Okay, I just had one question for Dr. Suri. When you said that, it's difficult, and I don't want to leave, we shouldn't leave on a negative note here for the younger people. I know it's very difficult, it's not easy, and now not being an institute and being in another institute, I can't tell you how much we need medical people out there for giving ideas, for collaborating on these, even if it's not a company, collaborating on that innovation. There's a lot of industry there, a lot of other domains, but we really need medical people to provide those ideas. And I think that is something one feels hugely, that vacuum there. So, yeah.
Yes. Just to add to what Shinjini is saying, the need for medical people, for taking the innovations, for getting the ideas. If you look at the inventions that have happened in the world, medical devices and medical diagnostics, 50% have been led by doctors and 50% have been led by engineers. Whether it's CT scan or MRI or any of these inventions, 50% have been led by doctors. So there is great hope, and wish all the best.
So with that, let me first thank my panelists, Dr. Rajesh Gokhale, Dr. Balram Bhargav, Dr. Chitra Sarkar, Dr. Shirshendu Mukherjee, Dr. Shinjini Bhatnagar, and Dr. Srinivas. I suppose he got a call somewhere and he had to leave in between. Thanks a lot. You've all been wonderful. And the same is true for those from the audience who made interventions and gave us their experiences. And finally, I would like to thank Dr. Rama Chaudhary, the director of the institute, and Vineet for asking me to be here. You put me in a tight spot. I really was flustered when on Sunday evening I was told that I need to step into this role. But I think my panelists and the audience made the job easier. Of course, having a 44-year association, it's very difficult to say no to something from All India Institute of Medical Sciences, but I enjoyed it. Thank you.
That was really interesting and engaging. हम अपने मॉडरेटर्स का और पैनलिस्ट का उनके इतने उत्साहवर्धक, ज्ञानवर्धक और प्रेरणादायक संवाद सत्र के लिए हृदय की सीमाओं से आभार और अभिनंदन प्रकट करते हैं.
So thank you all for this fantastic, insightful and lively panel discussion. I request Professor Vaishali Suri to please felicitate the distinguished guests.
Dr. Vineet Ahuja. He is not listening.
Thank you, sir. Thank you, ma'am. So now I hand over the stage to my colleague, Dr. Sunita and Dr. Soumitra for the award distribution ceremony.
Thank you, Dr. Kamal. Good evening, everyone. So now we have arrived to the part of the Research Day celebrations, the part which is most eagerly waited for, not only by the students, but also by their mentors. I now request all the deans, Dean Academic, Dr. Subrata Sinha, sir, Dean Research, Ma'am Dr. Rama Chaudhary, and Dean Examination, Dr. K.K. Deepak.
I also request Professor Guleria to please be on the dais to encourage our students.
Thank you, ma'am.
I thought I will take this time to share something. We had a wonderful panel discussion, and for the younger people, I really want to share one book, which is on Jennifer Doudna, the Nobel laureate, and the book is The Code Breaker. And this is written by Walter Isaacson. I recommend all of you to read it. This is a real true story of success story, that how just a simple chemist can turn her dream into an entrepreneurship, and she was the one who got the Nobel Prize. The key to that was actually the teamwork and to dream big, to have big dreams in your life. I'm sure you can make it happen. Many of you may turn out to be Nobel laureate tomorrow. So please read this book. This I really recommend all of you.
Good evening, everyone. I know it's been a very long day, but it has been definitely worth the wait. So we have different categories of awards to be presented. So by the time we start, let me just take you through the different categories of awards that would be provided today. So we have MBBS, Nursing, MSc, Junior Residents, Senior Residents, Ph.D. students, Project Fellows, SDG, that is the Sustainability Development Group, and Goals, the Innovative Device, and last but not the least, the Selfie Award.
I just want to say that we had a very overwhelming response as far as the posters were concerned. We did select 3 oral presentations which were held today, and all the posters I think are going to be displayed for another 3-4 days, so that those who could not see can go and have a look. And even though the award is already given today, but still you have a chance if you want to really take a selfie with whatever poster you like the most. And it was a big challenge for the judges to really select the best one. I can say for sure that each poster, each work which you have displayed was really wonderful. And I don't know who will get the prize. I still don't know the result of today's evaluation. But I can say one thing, all of you are winner. So all the posters, people, I really want to commend your efforts, the teamwork, And every poster, every presentation, every abstract was really, really wonderful. At a very short notice, because we did not give you much time, especially to SDG posters. But I must say that it was a commendable job, and already director has told us to keep this exhibition for some more days. So please keep visiting those who could not, come and see this. And it is really encouraging to all of them, those who have spent so much of time in doing that research. A word of encouragement, it really which makes that person feel, the whole thing has been truly fruitful. So that way, I really want all of you to visit if you have not already visited. And with that, I again, once again, also want to thank my team, the research team. Whatever is to be said at the end, I will say in the beginning, just to save the time, that the whole, everyone in the research team and in the research section has worked very, very hard. And I think from last 3-4 days, many of them were spending their evenings and they went late. So that is really commendable. And thanks to each and every one of you. So big clap for them. So sir is here already. Director sir is here.
So in the MBBS oral presentations category, the second prize goes to Mr. Ishan Gupta. For the MBBS poster category, the third prize goes to For the MBBS posters, the third prize goes to Mr. Arunangshu. The second prize goes to Ms. Tamogna Ghosh, Mr. Madhav Mohata. We also have the Critics Awards for the posters. The third prize goes to Mr. Bhavik Bansal. The second prize goes to Ms. Vaishnavi Agnihotri. And the first prize goes to Ms. Tamogna Ghosh. Next, we move on to the nursing category. We begin with the Judges Award for the best posters. The third prize goes to Ms. Vasundhara. The second prize goes to Ms. Monica Tanwar. And the first prize goes to Ms. Ruby. In the Nursing Critics Award, the third prize goes to Miss Monica Tanwar. The second prize goes to Mr. Bibin Lal. And the first prize goes to Miss Ruby. We now move on to the MSc category. For the oral presentation, the first prize goes to Miss Anjali Gaur. For the MSc Judges Awards for the posters, there are two commendation prizes. The first commendation prize goes to Miss Nandana Devi. The second commendation prize goes to Miss Sweetie Rani. For the third position, there is a tie. Miss Shivi Khurana and Miss Millie Kaur. The second prize goes to Miss Bhanupriya Tanwar. And the first prize goes to Mr. Yogesh Sharma. Moving on to the MSc Poster Awards under the Critics category, the third prize goes to Miss Ananya Kar. The second prize goes to Miss Sneha Verma. And the first prize goes to Miss Preeti Singh. The next award for the oral category under MSc, Miss Shivani Srivastava, first prize. The next category is the junior resident. The oral presentations, the third prize goes to Mr. Shravan K. Raut. The second prize goes to Miss Anjusha Ranjit. And the first prize goes to Mr. Yellam Raju Saikostub. MSc oral paper, first prize, Ms. Shivani Srivastava. For the junior residents, the poster category, there are 3 commendation prizes. Dr. Naveen KS, Dr. Vivek Ghosh, and Dr. Ayush Agarwal. The third prize goes to Dr. Suraj Verma. Not that. The second prize goes to Dr. Amtesh Khare. And the first prize goes to Dr. Ajit Anthony. The Critics Awards for the poster presentation, JR category. Third prize goes to Dr. Sumna VM. The second prize goes to Dr. Naveen KS. And the first prize goes to Dr. Aishi Pal. Dr. Aishi Pal. So the next category, we move on to the senior residents. We begin with the oral category. The third prize goes to Dr. Nidhi Gupta. The second prize goes to Dr. Surabhi Jain. And the first prize goes to Dr. Jaya Biswas. Dr. Jaya Biswas. Now we move on to the Judges Awards for the poster category for the senior residents. There are 2 commendation prizes, Dr. Manisha Kalung and Dr. Nikhil Mehta. The 3rd prize goes to Dr. Rijuta. The 2nd prize goes to Dr. Salman Khan. The 1st prize goes to Dr. Mohak Narang. We now move to the next category, that is the Critics Award for the senior residents. The 3rd prize goes to Dr. Nikhil Mehta. The 2nd prize goes to Dr. Roktim Swarnakar. And the 1st prize goes to Dr. Saranya B. Gomati. We now move on to our next category, the PhD. Under the oral category, there is a tie for the 2nd position. Miss Apoorva Challa and Miss K.M. Renu both get the 2nd prize. And the 1st prize goes to Miss Srishty Raman. Moving on to the Judges Awards for the PhD poster category, there are 3 commendation prizes. Ms. Srishty Raman. So the poster awards under the PhD category, there are 3 commendation prizes. Miss Mahaiwan Shadang, Miss Jyotsna Singh, and Miss Christine Wilson. The third prize goes to Miss Leena Sapra. The second prize goes to Miss Shivani. And the first prize goes to Mr. Amit Kumar Yadav. Biophysics. Amit Kumar Yadav, Biophysics. Moving on to the Critics Awards for the posters, the third prize goes to Mr. Brijesh Kumar. The second prize goes to Miss Apoorva Bhargava. And the first prize goes to Miss Shweta Sharma. Next, we move to the other category, that is the Project Fellows. Again, we've got 2 commendation prizes for the posters. Miss Shruti Mahapatra and Dr. Vivek Dikshit. The third prize goes to Dr. Kriti Kaushik. The second prize goes to Miss Ruchi. And the first prize goes to Dr. Soumya Dash. Moving on to the Critics Awards for the best posters. The third prize goes to Miss Alia Mufti. The second prize goes to Miss Mamta Puraswani. And the first prize goes to Miss Prachi Tiwari. The next award category is the SDG Judges Poster Awards. The third prize goes to Miss Srishty Raman. Second prize goes to Mr. Vinay EV. And the first prize goes to Miss Apoorva Challa. Next, we move on to the SDG Awards for under the critics category. There are three second prizes. Mr. Masroor Anwar, Miss Indrani Mukherjee, and Dr. Leena Sapra. The first prize goes to Miss Sonakshi Gupta. We now move on to the Innovative Devices Award. The third prize goes to For the Innovative Devices Award, the third prize goes to Dr. Ashish Suri. The second prize goes to Dr. Nihar Ranjan Dash. And the first prize goes to Dr. Vikram Saini. We've now come to the last award, but that's not the least, the Selfie Award. And the Selfie Award goes to Professor Urvashi Singh from the Department of Microbiology for her poster on the SDG. So with this, we come to the end of the awards and presentations. Thank you, sir. Thank you, ma'am, for doing the honors. I now request Dr. Sunita to deliver the vote of thanks.
Thank you, sir. Thank you, sir. Thank you, ma'am. The crowd looks very excited, right? So now I'll just say thank you all, and thank you all. Thank you everyone who has contributed to this great festival. I call it as a festival, not a research day. As we head into Diwali, it's just a festival now. So I would, ma'am, would you please say
I
a few
just want
words?
to say one thing, that please have a photograph of all the winners before you leave. And thank you everyone. As I said earlier, all of you are winners. So please don't feel bad. The awards are meant actually for everyone and just representatives are getting it. That's all.
So here are some pictures for you if you want to revisit your last 1.5 days. Our sincere thanks to all our distinguished guests, speakers and chairpersons who spared their time to be with us today. We earnestly thank the director, Dr. Srinivas, especially for the support to our research and grants. Thank you, Dean ma'am. Thank you for your valuable inputs, without which we could not have organized this meeting at all. We thank Dr. Ahuja, who has been the backbone of this Research Day celebration. We thank all the participants who presented their research here on this platform. Every single contribution from you in the form of your research findings, experiences, and deliberations are vital to the mission of this institute. Congratulations again, and thank you very much. My vote of thanks will not be complete until I say something about the 4 facilities, the CMET, CMI, the CRU and the CCRF, who have worked endlessly, tirelessly over the last few weeks. So thanks a lot for making this event a huge success. Special thanks to Dr. Ahuja. Sir, would you like to say a few words? Sir, please. I think everybody wants to hear from you, sir. Okay.
So I just want to once again thank Dr. Vineet Ahuja. He is like behind the scene. He has been doing everything and the whole success goes to him. Please, big hand to Dr. Vineet Ahuja. Thanks to all the team members here. I think we should have a picture with all of you. Come quickly. Come.